¿Estás más cómodo a un lado o es quien así estiro la pierna? Good morning. No sé si se me oye, ¿no?
Good morning. Is it working?
Morning. Yeah, welcome to this second day of the conference. My name is Mircea Balasa, here from Barcelona, from Hospital Clinic, and together with Daniel Colea, who's here at Sant Pau Hospital, we're going to moderate these fluid biomarkers oral sessions. We have four talks today. Unfortunately, two of the speakers have not been able to come here in person, so we're going to have two live talks and two pre-recorded sessions. So for this table, we're going to have Julie Wish from the WashU, who is going to give a pre-recorded talk. Bo Anses from the same side, Washington University, who's here with us. Laura Ibanez, who we're going to have also a pre-recorded talk. And Aina Comas, here from Barcelona, from Hospital Clinic, who's with us today. So without further delay, first talk is Julie Wish, the interactive role of ApoE and sex in Down syndrome, Alzheimer's disease, from the Washington University School of Medicine.
Hi, thanks for having me. I'm sorry that I couldn't be with you in person, but I'm excited to share the results of a collaboration between ABCDS and DABNI looking at the roles of ApoE and sex in Down syndrome. I have no disclosures. It's been established in sporadic AD that ApoE haplotype dictates AD risk. You can see here, particularly in non-Hispanic white individuals, that the ApoE4 allele increases the risk of developing AD, while the ApoE2 allele is protective against developing AD. It's also been shown that ApoE interacts with sex, and particularly the ApoE allele seems to combine with the female sex to either increase the risk of developing AD in female ApoE4 carriers, or conversely, the ApoE2 allele seems to be more protective in females than in males. In Down syndrome, it's been shown that the ApoE4 allele does accelerate time to dementia. So in ApoE4 carriers develop Alzheimer's disease at younger ages. You can also see greater declines in cognitive performance on cognitive tests like the modified cued recall test, which is shown in figures E and F here in this manuscript that came from the DABNI cohort. This is shown to be particularly true in females, where it does appear that consistent within sporadic AD, the ApoE4 allele interacts with the female sex to be primarily deleterious in females. It's also been shown that the ApoE2 allele is protective in Down syndrome. This study demonstrated that individuals who were ApoE2 carriers and had Down syndrome developed Alzheimer's disease at older ages. In terms of biomarkers, it's been shown that the ApoE4 allele impacts both amyloid and neurodegeneration. You can see in figure A that ApoE4 carriers had lower levels of CSF beta 4240 earlier in life, and that's consistent with worse clearance of amyloid fibrils. What we would expect from that then is that those folks would have higher levels of amyloid PET, which we see in figure B, where the ApoE4 allele carriers have higher levels of cortical amyloid. And then you can see in figures G and H, those are imaging-derived markers of neurodegeneration, that the ApoE4 carriers have greater neurodegeneration starting around age 40. And so this suggests that in Down syndrome, the ApoE4 allele contributes to that, or leads to that earlier age of onset by impacting the pathological precursors to cognitive symptom development. There's been less work for the ApoE2 allele looking at biomarkers. I actually haven't been able to find any biomarker studies looking at the ApoE2 allele and biomarkers in Down syndrome, but there are some case studies out there. And so here's a case study of a woman who had actually participated in imaging studies over multiple decades, who lived into her 60s, remaining cognitively stable. You can see from her amyloid PET scans that she never really developed many amyloid plaques kind of throughout the lifespan, also minimal tau tangles. which is really remarkable for someone with Down syndrome in their 50s and 60s. And she remained cognitively stable. She was an ApoE2/3 carrier. This case study doesn't offer conclusive evidence that the ApoE2 allele is protective, but it certainly suggests kind of that the ApoE2 allele may be protective. Studying the ApoE allele is challenging just because of sample size. So when we start to try and divide up the data and look at people by ApoE allele, and then particularly by ApoE and sex, that can be really challenging to study. You just have to have large numbers. And so that brings us to this study, which I'm very excited to be sharing with you. So this is a collaboration between ABCDS and DABNY. These are two very large research consortia for individuals with Down syndrome. These people are deeply phenotyped, they're imaged, they do blood draws, many of them complete lumbar puncture. We also have extensive cognitive testing on them, and they all receive consensus diagnosis. And so you'll see here that although not all individuals completed all things, we have more than 1,200 people with Down syndrome who have completed at least some of these biomarkers. And I'm excited to be able to share with you kind of what we've been able to learn from this collaboration. To kind of just set the stage, these are the plots that I'll be showing. And so you'll see on the y-axis, I'll have the biomarker level. Along the x-axis, I'll have age. And then you'll see results by ApoE2, ApoE3, or ApoE4 based on colors. The ApoE2/4 carriers were classified as ApoE4 carriers, for those wondering. And we weren't able to split out and look at the ApoE4/4 carriers separately just because of sample size. To kind of keep a focus on the timeline, I just want to highlight that what's been shown previously is that the average age of amyloid positivity in Down syndrome is around age 40, and the average age of symptom onset in Down syndrome is in the early 50s. I'd like to highlight that the majority of the participants and the majority of the data that I'll be showing are from individuals with Down syndrome between the ages of about 40 and 60. We use generalized additive models for most of these analyses, and I just think it's helpful context to keep in mind that the bulk of the data is really in this key age range. This is when we expect pathology to be developing. This is when we expect cognitive symptoms to develop. So this is kind of the ideal place where we've got the bulk of the data. But sometimes we'll see statistically significant differences for individuals in their early 20s. And I'm not convinced that those are actually meaningful when we just think about model fit and the number of data points available in that age range. So just to kind of keep that in mind as an important caveat. Kind of on the backdrop of the rest of these plots, you'll see DABNY shown with circles and ABCDS participants shown with X's. For all of our analyses, we did control for cohort. We performed these analyses both separately and together. We've done data harmonization. And that's something that is covered in the manuscript that's currently under review at Lancet Neurology. You're also going to see these significance bars across the top. So we tested for differences at the group level. And so if there are statistically significant differences, you'll see them annotated at the top with these little bars. So first, CSF A-beta 42/40, we saw results consistent with the earlier DABNY publication where the ApoE4 carriers had lower levels of CSF A-beta 42/40 kind of in their 30s and 30s and 40s. This is suggestive of worse clearance. So we would expect that then to lead to greater amyloid plaque accumulation down the road. When we then we stratified it by sex. And we did not perform statistical tests because we're kind of limited in numbers here. But using the eyeball test, it does seem like the female ApoE4 carriers in particular are the ones with the lowest levels of CSF A-beta 42/40 in the kind of 30 to 40-year-old age range. Then when we looked at cortical amyloid burden, so now we're switching to amyloid PET, we saw that amyloid PET levels were significantly lower in the ApoE2 carriers between the ages of 40 and 55. And so that's suggestive of that protective effect of the ApoE2 allele. We also looked at inflammation and again saw the protective effect of the ApoE2 allele. You can see here that plasma GFAP was lower in the ApoE2 carriers in their 50s than in everybody else. In terms of cognitive performance, we saw the most dramatic effects of the ApoE4 allele, where ApoE4 carriers performed worse on this test starting around age 50 and then continuing throughout the rest of the study. When we stratified this by sex, again, we saw that big kind of sex by ApoE interaction, at least at the eyeball test level, where female ApoE2 carriers, which are shown in that light blue line, actually showed basically no decline in performance over the range of the test. Conversely, the female ApoE4 carriers, shown in that light pink, they had the worst performance starting around age 50 and had a faster rate of decline. So this kind of, again, points to that sex by ApoE interaction being really important. But again, we're still limited on numbers, even with this large cohort. We still need more to be able to rigorously investigate this. Finally, we looked at prevalence of cognitive impairment. So you'll see on this prevalence plot, we've got age along the x-axis. On the y, we have probability of cognitive impairment. And you can see that consistent with what the literature says about an average age of onset of maybe 52 or 53, the ApoE3 carrier, shown in green, hit that 50% probability right around age 52 or 53. The ApoE4 carriers, though, the 50% line hits right around actually about 5 years earlier. So this suggests that ApoE4 carriers develop AD on average almost 5 years earlier than the ApoE3 carriers. And then when we look at the ApoE2 carriers, we see that we actually never hit that 50% threshold for cognitive impairment. When we stratified it further by sex, we really see that female ApoE2 carrier protective effect again. So the male ApoE2 carriers actually don't look any different than the ApoE3 carriers when we looked at prevalence of cognitive impairment. But it's the female ApoE2 carriers who are driving that low prevalence of impairment and kind of that delayed onset. So what we can take away from this is that ApoE and sex are really important when we're thinking about timing of intervention, whether that's for treatment or for clinical trial. It does appear that the ApoE4 allele will kind of accelerates both some of the biomarker changes as well as observed cognitive impacts. And the ApoE2 allele is protective, and it also does seem that in both cases this may interact with sex, but that requires a little bit more study. We also want to, in the future, look at the effects of ApoE on tau pathology, looking both at MTBR243 and tau PET. This study, of course, would benefit from having longitudinal data, and we'd like to just continue to increase the sample size so that we can both more rigorously investigate that sex by ApoE interaction and also see if what that ApoE dose effect looks like. So with that, I'd like to thank, first of all, our participants. 1,200 people with Down syndrome have given a tremendous amount of time to allow us to investigate this, and we really appreciate them. I'd also like to thank our collaborators, both between ABCDS and DABNY. This is a huge effort across so many different sites, and I really appreciate everyone's contribution to this study. I'd also like to thank the people that support this study financially. Thank you so much.
Well, we thank Julie in the distance for this wonderful talk. And our next speaker is Bo Anses from Washington University too. And he's going to speak about NOLISA blood-based CNS biomarker panel across the adult lifespan of Down syndrome.
I hope you can you hear me?
Yeah.
Okay, great. It is really a pleasure to be here today. Just wanted to highlight for a second that last talk that we had. That all came out of this conference last year. So the ability to have these kinds of collaborations, these kinds of links is immensely beneficial and has really allowed us to explore some of these different areas. Today, I'm going to be talking about an integrated lifespan analysis of Down syndrome and euploid controls using blood-based biomarkers using this test called NELISA, which is this nucleic acid-linked immunosandwich assay. This is really came out of a different conference that I was at, and really the new data. This has never been seen before. This is brand new data that I wanted to kind of show you, and it's really a collaboration between University of Colorado, in particular Joaquin Espinosa, as well as Kelly Sullivan at University of Colorado, and my team, Sarah Cooley and myself and others at WashU. So what we're going to be talking about today is how does NELISA work? What is measured in this panel? We'll talk about effects of sex and age on these plasma biomarkers. We'll also look at effects of aging across the lifespan. And what we're going to be really looking at are these kinds of windows and moving time frames for looking at the lifespan. Then we'll do kinds of computational approaches to identify these clusters and then compare them across the lifespan. And then I'm going to give you a hint at these curves of Jack, but it's very simplistic, I'm going to tell you that, but across the lifespan. And I knew kinds of ideas of how we can use these blood-based biomarkers. And then at the end, I'm going to show you not just looking at a single biomarker, but their interactions as well. So what is NELISA? So it's a DNA conjugated antibodies that form these immunocomplexes that target the proteins. And then what they do is there's a double binding that occurs for this. This immunocomplex is then purified first with oligo-DT, and then further with streptavidin beads binding to biotin for detection. Then there's multiple washes that occur, and then this DNA reporter is generated by proximity ligation and amplified by PCR. And this DNA oligonucleotide contains these barcodes, so then we can see and demultiplex these targeted proteins. And so this idea is that we can look at multiple different proteins within a very small sample of blood. And when I'm talking very small, we're talking 25 microliters at most for the sample that we can be looking at. What's also interesting is that we can then, this complex does much better in looking at this more dynamic range as well as this lower sensitivities compared to other complexes that are out. So in red is our typical measures, and then in NELISA you can see there's significant decreases that you can already see that it can measure at, and a much more longer window that can be seen. And then same even for HIV, for P24, you can see that it measures at a little bit lower concentrations and has this full dynamic compared to the Samoa, which kind of stops halfway or 3/4 of the way through. Using this, we can then look at different measures of inflammation, neuronal death, synaptic, cytokines, and altered hemostasis. And so the idea is this almost gives us a liquid biopsy of what's going on in an individual, and we can look at multiple different values compared to any one single value for this individual. So what we did was we took 188 individuals with Down syndrome. Those are individuals that I see in my clinic at WashU, as well as those that were seen at the University of Colorado. And those ages were from 1 to 68, with 55% being female. And then we have 161 euploid controls, ages from 2.7 to 81 years of age, with 49%. Here you can see the different distributions, and you can see we have a very nice distribution in our controls. And then in our Down syndrome in green, and you'll see that consistently throughout. They'll always be green in these figures. You can see that we cover a large age range, but then, as you were hearing, we heard and talked about yesterday, into the 50s and 60s, but especially into the 70s, not many are living to those ages, so we have very few of those individuals. We then looked at different areas, so amyloid and tau, neurodegeneration, inflammation, synaptic function, and vascular. These are these kinds of different panels or different groupings that are used in ELISA, so we'll look at that. And then we looked at this bioinformatics analysis, so we did these volcano plots. You'll see the sliding window for looking at over 20-year age bins, and then we did these kind of correlation matrices looking at how different markers kind of interact as well too. So first on this volcano plot, so many of you may have seen these, but if you don't, though, to the right are those that are elevated in T21. To the left are those that are down in T21, so in Down syndrome. And what you can see is that in multiple different areas of all of these panels, you see some increases, but also you see some decreases. In particular, with A-betas, you already see increases, and this is just the abundance of this, is 40, 42, as well as our p-tau species that are present. You can also see that in neurodegeneration, heavy chain NFL, so NFL heavy chain, as well as decreases in certain measures, and I've given you some of those, so like ENO2, which is, or also NSE, which is a glycolytic enzyme, a neurotrophic factor, ANXA5, which is a protector factor for amyloid. So all these protective factors are being downregulated in Down syndrome. You can also see large increases in certain inflammatory proteins like CCL17 or C-reactive protein, but also decreases in specifically in CRH, which is a corticotropin-releasing hormone, again, something that protects against amyloid toxicity and tau phosphorylation. And then in synuclein, you can see some increases in SOD1, but also increases in vascular metabolics, or SAA1, which promotes neuroinflammation and amyloid deposition. So you can see a lot of this pro-inflammatory kinds of changes that are occurring, increases in amyloid that are present in these, as well as increases in neurodegeneration, and then these protective factors are also being downregulated in these individuals. And so we can then do these plots and just looking at overall differences of abundance between the two. Here are different measures of amyloid, so A-beta 40, p-tau 181, and we'll then look at all the other tau species of this as well. NFL is all elevated in these individuals, as well as inflammatory markers seem to be increase in these individuals. And then in some with Down syndrome, there's actually these decreases in certain of those kinds of protective factors that are present. So multiple different pathways are being affected across the lifespan. And then we can also look at the effects of sex. And so here, what is up or increased in males versus down in both the euploid controls as well as in trisomy 21. The big point out of this is biological sex seems to be affecting both in Down syndrome and controls, but it seems to be greater, so there's more upregulation in the controls than there is for the Down syndrome. You see both increases and decreases that are present in trisomy 21. Now you also look at the features that are common and see how many of them, of these top features or bottom features, of those that are downregulated, and you can start to see that there are differences. So the features with the largest changes in each of the karyotypes are not shared between the karyotypes. When we look at the effects of aging, you can see that there are some similarities. So again, what is upregulated in controls versus downregulated, same thing in trisomy 21, what's upregulated, what's downregulated. You quickly see that NFL, so this is neurofilament light chain that is also there, is increasing. We see that typically with aging in general, and then other ones like MME is also upregulated for degrading enzymes, and then CRH in both are also downregulated in both. And then we can see that there's some similarities, but there's also some differences. So few of the features from the largest fold changes in both of the karyotypes are shared between the different karyotypes. All right, so that just tells you an abundance, so how much is present and what is up or down. We can also look at this sliding windows and look at overall over the lifespan, what do we see? And so I'm going to use first the ELISA different breakdowns. So this is amyloid and tau, neurodegeneration or inflammation. And what you can start to see is that these species of A-beta 38, they're elevated very early on. Same thing with 40. Same thing with many of these in 42. They're elevated and they remain elevated throughout the lifespan. In contrast, what we see is this p-tau, specifically here's p-tau 181, but we can also see this with 217. And what you start to see is that early on, there is a large increase. And in fact, in cognitive, so in the euploid controls, you can see that it's actually higher. Then there's this kind of decrease over aging, and then it kind of remains flat, and then it starts to increase back later in life, suggesting that early on these changes are synaptic pruning, or we wash my hands on this, but we think that there's some kinds of changes that are occurring in these individuals very early on, but then these later on changes are most likely from Alzheimer's disease. In contrast, in Down syndrome, there is an increase, but it is not as high as those that are seen. But you can see the confidence intervals closely overlap. But in general, they're lower in Down syndrome. And that means that neurodevelopmentally, so how many of those connections are actually diminished. So there's not as much synaptic pruning. It decreases over time. But then there's this sharp rise. That is that amyloid deposition that we're seeing. And so here we can kind of see the amyloid production. Here we can kind of see those amyloid depositions that are present within these markers. We're looking at neurodegeneration. You can start to see heavy chain is increased throughout the lifetime. SMOC1, which co-localizes with amyloid plaques, is also increased throughout or a little bit later on in the lifetime. Inflammatory markers, S100, CRP, are elevated throughout the lifetime of individuals with Down syndrome. When we looked at different effects of Down syndrome with regards to synuclein and synaptic or vascular and metabolic, you can see SOD1 is elevated throughout the lifetime, and we know SOD1 is an antioxidant that protects free radicals, and NGF, which maintains nerve growth factor for the basal forebrain and is one of the earliest markers of cholinergic loss, is already diminished and continues to diminish across the lifespan of adults with Down syndrome. And in regards to vascular changes, PDGFR, which is a measure of this blood-brain barrier, it seems to be already diminished across the lifespan, and hemoglobin A1, it seems to be a little bit elevated. But then we also see other markers like KDR, which is another marker of this blood-brain barrier maintenance. It seems to be also affected when that amyloid deposition is starting to occur. So there may be changes that are occurring throughout the lifetime in the blood-brain barrier, and then specific changes, and we talked about this yesterday, that may be occurring when amyloid is being deposited. We can take that, so that was all the NELISA categories. We can then take our own clustering and then just see how do all of these panels, if we just put them all out, how do they kind of cluster? And we can kind of see, are there ones that are increasing throughout the lifetime? Are there ones that are decreasing? Are there ones that are changing at certain periods? And we can develop these kinds of clusters. So clusters 1 and 2 are elevated across the lifespan, and you can see one being YKL40, which is increased throughout the lifespan, as well as NFL, and then A-beta40 we already talked about, and IL-2 being increased throughout the lifespan. What you see is that these are measures of not just amyloid, but also neurodegeneration and inflammation that are occurring throughout. So this is not just the disease of amyloid, it is multiple different complexes that are occurring. With regards to cluster 3, we see those that are only occurring later in life, and that's where all these, the p-tau 181, 217 are all kind of clumping, and 231 are all clumping together, and that's usually around the ages of 40, and you can see, again, these large increases that are present there. Then there's a cluster that is decreased throughout the lifespan, and these are a lot of those protective factors, so changes in the blood-brain barrier, changes in kind of neuroprotection that are occurring, and then you can see some that are occurring later on, and then some that are occurring throughout the lifespan for this. Then the fourth one is some that are elevated just slightly across the lifespan, and this includes all the biomarkers from all the 5 different categories. And then cluster 6 are these light blue that are decreased throughout the lifespan, and that can be even VEGF as a neuroprotective factor, but they cover multiple different categories that are present. So I just showed you then looking at the differences between like abundances, but what are the overall kind of timelines, or what are the overall curves looking like in these 2 different groups? So in orange are euploids and blue are Down syndrome, and you can see there are some, we're now just looking at the overall shapes of these that look like they're flat throughout, some where there's an increase in Down syndrome later on, some where there's an increase and then there's kind of just a flattening out for both, some where there's an increase and then flattening, and others that are going straight up. and then some that are kind of start out and then in Down syndrome seem to decrease later on. And you can start to see that they follow these different patterns. So like in NFL is going up versus A beta 42 and 40. They also have these same kinds of overall similar trajectories versus in p-tau 217, you see wide differences between the two groups. And then KDR, this breakdown of the blood-brain barrier. So they're not only differences in abundance, but also the timing that can be important. From this, we can kind of start to generate these hypothesized. Now, these are early. We're going to be making some changes to this, but we can look at either controls or T21, so those with Down syndrome. And you can start to see with those with Down syndrome, there's a decrease in A beta 42 that's occurring. But very interesting is that p-tau, which starts up a little bit, kind of normalizes for a period, and then in the 40s really takes off. That's followed by changes in like GFAP, which we talked about yesterday, and then also changes in NFL. So that kind of gives you the curves, and now that's the timing. And I've shown it for each of the different kinds of proteins that we can look at. What about those interactions between the proteins? And so we can look at these by correlation matrices, and you can see that when we looked at either the euploid or the trisomy 21 or the differences, you can see that there's about 227 differences between the groups, and it's highly really conserved in these p-tau species. And so when we do these volcano plots of these interactions, we can kind of plot out those changes, or we can also look at it by graph theory of these kinds of changes. And what I'm going to kind of point to is one example is CXCL1. You can see that it's with a large amounts of changes with amyloid, so neprilysin, p-tau 231, 181, and CXCL1 is the signaling molecule for microglia. So there seems to be this interaction between amyloid and microglia that we can kind of start to see. And then you can also see with the GDP or GD1, that also seems to be downregulated in Down syndrome, so it's weaker versus in the euploid controls, and this ENO2 seems to be also these kinds of interactions. So how do we summarize this? Well, really, we're at this unique starting to be stages for these blood-based biomarkers, specifically some with NELISA. This serves as a sensitive liquid biopsy that looks at multiple different pathways that we can do, and this really makes it available to everyone. I am an imaging person who loves imaging, but I can't scan everybody. I can't do PET scans for everybody. I can, though, get blood-based markers that are more accessible, and then that allows us this to also do it for even for clinical trials. When we look at these markers, there are effects of aging on these markers. Some are elevated, some are diminished, and then some increase later on or earlier on in life. We can even generate up these curves. But really what this is starting to tell us is that Down syndrome, because of the great work, and we've seen this with Alzheimer's disease and the new criteria, is stage 0, where there is already an overproduction of amyloid occurring at birth, and then later on we're starting to see these changes in deposition, and specifically p-tau 217 in infancy may reflect these synaptic changes and axonal, neurite, and pruning, and that then later on in life are those pathological changes that we see due to decreased clearance that are present. With regards to the inflammation, there are some pro-inflammatory markers that are elevated at birth, while others are elevated later on, like the GFAP, and then we see some of those regulators, so those protective factors, are also diminished throughout life. Synaptic transmission, such as neurogranin or other ones, are reduced throughout life and continue to be reduced throughout the lifespan. Blood-brain barrier, there's some that are occurring earlier, and then some that are more accentuated with amyloid deposition. And then plasma markers overall can then point us to these new interactions, these new pathways, and then lead to new therapeutics for more precision medicine. So I'm very fortunate enough to work with this great group, as well as really thank our funding organizations. But most importantly, I just want to say it is really a pleasure to take care of so many of these amazing people and their families. So thank you so much.
Thank you. The next talk is also a pre-recorded one. It will be presented by Laura Ibanez from Washington University School of Medicine, and it's titled Cerebrospinal Fluid Proteomic Profiling and Biomarker Discovery in Autosomal Dominant Alzheimer's Disease.
Good morning, bon dia. I'm Laura Ibanez fluid biomarker core leader of the DIAN study. And today I have the pleasure to show you the latest results on CSF biomarkers using the NULISA platform in the DIAN population. Before I start, I want to thank everybody in DIAN who is involved, collecting samples, collecting data, admin, everybody, but also want to highlight the role of these three people in the results I'm going to show you. Bryce, Sashka, and Wenjing, they were crucial to generate the data, analyze it, and generate the plots. Also, this QR code is going to go, it will get you to the preprint that it's already available. So I like to start this presentation going over the NULISA technology, but I know that Dr. Anses already did that, so I'm going to skip it and go straight to what we have done. So we have included a longitudinal sample of about 1,000 CSF samples corresponding to 500 participants from DIAN, which are more or less distributed 2-to-1 between mutation carriers and non-carriers. Demographically, as you can see in this table, there are no big difference between those. Before going into the analysis, and because NULISA it's sort of a newish technology, we wanted to benchmark it similar to what we did in sporadic AD in collaboration with Dr. Cruchaga. So the first thing we did was a correlation between the values that were available in DIAN from the same samples with the NULISA measurements. And as you can see, we had mass spec, Lumipulse, and a sort of ELISA or proximity assays, and all of them have high correlations. The x-axis is the Spearman correlation value with small errors. The exception might be neurogranin, SNAP25, and BILIB1, but those are homebrew assays, so that might explain this higher variability. But we were very happy with these results. Also, because it's a new technology, we want to know how stable this assay is, so we scattered about 10 samples around plates. NULISA run in groups of 86, so we wanted to know that if we measure in different plates or in the same plate, the same sample, we get the same measurement. And as you can see in this plot in which we correlate replicates, intraplate on your left and interplate in your right, they are highly correlated. Before moving into analysis, it is important for us to check that there is no batch effect or methodological artifacts going on, again, because this runs in batches of 86. And we were happy to see in our principal component analysis that there is a nice and tight cloud of samples. There are some outliers which are outside of these light dotted lines, which is 2 standard deviations. Those were excluded from the analysis. In the diagonal line, there is a little bit of biological stratification that is explained by not being a carrier, being a non-symptomatic carrier, or being a symptomatic carrier. We didn't consider that a batch effect since we can explain it with biology, and we know this is what we expect. So we moved straight to the analysis, and the first thing we did was differential abundance analysis using a cross-sectional sample. We leveraged the last visit of each participant, so sample size is about 500 participants, and we compared the carriers versus the non-carriers, and what we observed was that we were getting the top hits were the known usual suspects, which again supports the use of this technology. This model was adjusted by expected age at onset and sex. And if we disregard the known biomarkers, as you can see, there are a lot of proteins coming up in this region that are highly interesting, and then we will take a look at them later on. But because DIAN, we know we have people that is asymptomatic, but we know they are getting closer to symptomatology, we wanted to compare among those that are mutation carriers, those that are symptomatic versus those that are non-symptomatic. And what we observe is that the usual suspects, p-tau217, A-betas, they are still important, but the importance is reduced, and other proteins are coming up as important in the symptomatology stage, NFL, synuclein, FAP-P3, etc. However, DIAN enrolls people that have mutation in 3 genes, presenilin-1, presenilin-2, and APP. So we wanted to know if any of those proteins was, the abundance was different due to any of those genes and not in the others. So we compare each of the carriers for each gene to the group of the non-carriers. And I just want to highlight that presenilin-1 are the vast majority of those. So when we did that, as you can see, there are no big differences. P-values are different probably because of the sample size, but we see the same. And I can further convince you showing the correlation between the effect size of each of those comparisons that you see in this slide to the general comparison that I showed you before. And with precedent 1, we have almost a perfect line, probably due to the sample size. But for the other two, we are not far off of this correlation, and the results are pretty much the same, suggesting that putting them together, it's okay methodologically, because the big changes are the same. I'm sure that if we can get larger sample sizes, we will find specific proteins for each of the mutations. So now that we can go back to these proteins that were coming up in symptomatology and try to chronologically model when they become abnormal in this population. To do that, we leveraged the longitudinal sample again, and we calculated the probability of protein abnormality as a function of estimated years of onset. We did that as previously published using the percentiles and then fitted a logistic mixed effect model. And this is what we obtain. We know that the amyloid pathway is what gets abnormal first. But if we look at symptom onset or the expected year onset at EYO0, there is a lot going on here. And so we were interested in that section. And the goal is, well, we have these pre-symptomatic time in DIAN can we leverage to develop protein models to predict age at onset that potentially can be applied to other forms of Alzheimer's disease. So to do so, we went back to a cross-sectional sampling and we created bins of 2.5 years. And we used Lasso to rank our proteins and see what was more informative and created a multi-protein model. And then we calculated the mean absolute error of this model and the known biomarkers and the published biomarkers using the mean absolute error by bootstrapping. So on your left, you have a panel with the proteins that we included in our multi-protein model and how informative they are to predict time to onset. GFAP coming up with 25% of this prediction. We also included p-tau217 and the A-beta ratio, but others were also playing a big role. On your right, you have a summary of our comparisons. On the y-axis is the mean average error, and in the x-axis is the estimated years to onset, and we plotted only when that error is the smallest. And as you can see, the multi-protein model is the one that has the lowest mean average error, especially close to onset, whereas p-tau181, it's better when we go farther away from age at onset, similar to p-tau217 and A-beta. And at the same age at onset, those are the comparisons. But this is just a snapshot of when they are the smallest. So I wanted to show you the overall for those comparisons. And this is the the predictive value, it's in the solid line, and the shadows are showing you the mean average errors at each time point. And also on the bar plot, you have a reference of what was the sample size for each time bin. Remember, each time bin contains, there are no participants in two bins. So, and we selected the sample in a way that maximizes all the bins. So as you can see, this is the bottom right, this is our multi-protein model, and this is the smallest error that we get, especially close to zero. P-tau217 does great, also -5, -15, -5, whereas others have like big errors, especially A-beta 40-40 has big errors, and imaging has a stable error. So what this suggests is that maybe single analyte is not enough to predict onset, and we need to combine several proteins to get to a good prediction of when this individual, or how far is this individual from developing symptomatology. So in conclusion, we have replicated previous results using this novel mid-throughput multiplex low-volume platform. We have shown that inflammation, potentially neuroinflammation, is important in mutation carriers regardless of symptomatology. We have shown that there are slight differences in protein abundance depending on which ADAD gene is affected, but in general, they look very similar, supporting the analysis as a group. We have proposed a cascade of events for the DSAD population, which suggests that inflammation is the last pathway to get dysregulated and amyloid first, as we all know. We also have generated preliminary models to predict age at onset ranges. What we are doing now is the full DIAN and plasma OPS collection and the CNS 120 panel is being generated, and we are hoping to be available soon. The paper is on draft status. Again, I put here the QR code for you to, if you want to check the preprint, and we are getting ready to submit to Lancet Neurology soon. And with that, I want to thank again the people in the lab, the DIAN family, but also the funding, the patients, the families, the caregivers, everybody. I'm sorry that I cannot be there to take questions, but I am available via email if you have any. And I'm sorry that I'm missing my city, so please enjoy Barcelona, drink Estrella, and get some Fuet on my behalf. Thank you very much, and I hope you enjoy your time in my city.
Well, we thank Laura for her presentation and also for her advice too. And I think we can move on to the last speaker, who is Aina Comas Alberti from Hospital Clinic here in Barcelona, and she's going to talk again about proteomics in cerebrospinal fluid and plasma in autosomal dominant Alzheimer's disease.
Thank you, Daniel, for the introduction. I'm so happy to be here as I presented in the first edition of this Congress 2 years ago as a master's student, and today I'm happy to present my work as a second-year PhD student. Today, I'm going to talk about proteomic profiling of CSF and plasma in autosomal dominant Alzheimer's disease. I have nothing to disclose. So, as you may know, proteomic profiling of CSF and plasma can reveal molecular changes associated with Alzheimer's disease pathology. Nowadays, most proteomic studies have focused on sporadic AD and with limited work done in autosomal dominant Alzheimer's disease and usually in one single biofluid, although the number of studies is steadily increasing. Existing ADAD studies usually use conventional proteomics, but ultrasensitive multiplex methods like NULISA-seq have been barely unexplored. And for the detection and progression of disease monitoring, we require biomarkers that can be measurable in accessible fluids. So our main objective was to characterize the proteomic profile of proteins related to central nervous system disease in autosomal dominant Alzheimer's disease in 2 different biofluids, in order to identify proteins that can be associated with disease stage, indicating early or preclinical changes, and that could serve also as potential biomarkers or provide insight into the biological mechanisms of ADAD and even extrapolate to sporadic AD. And finally, we wanted to assess also the relationship between CSF and plasma protein levels to evaluate their concordance. This is our cohort. We included participants of the Picogen genetic counseling program of the Hospital Clinic de Barcelona. And we divided these 3 groups, the healthy controls, which were non-mutation carriers, the asymptomatic mutation carriers, and the symptomatic mutation carriers. The CSF cohort comprised 47 individuals divided in these three groups, and the median age was slightly higher in the symptomatic group compared to asymptomatic and healthy controls, and sex and ApoE4 allele carrier frequency were similar across the 3 groups, so that's why we decided to correct just for age our analysis. And then our plasma cohort comprised 39 individuals, of which 26 of them had matched samples of CSF. To achieve our goals, we decided to perform the NULISA-seq CNS disease panel, which is a panel that targets approximately 120 proteins related to central nervous system disease, and we measured it in CSF and plasma. I'm not going to explain the technology, as Bo did explain it really well, but it's ultrasensitive multiplex technology that only requires 20 microliters per sample. And then we obtained the NPQ values, which are transformed to a logarithmic scale, and we analyze the differential expression using Lima linear models, which are implemented in our software. So here you can see the results of our differential expression analysis. In the upper part, you can see the CSF, and at the bottom, you can see the plasma ones. If we take a look at the symptomatic mutation carrier versus healthy control comparison, we have 23 upregulated proteins in the symptomatics and 1 downregulated protein. And if we look at the plasma, we have 9 upregulated proteins in the symptomatic group. Unfortunately, when we looked at asymptomatics versus healthy controls, we did not obtain differential proteins significant, but we did look at the symptomatic versus asymptomatic, and we obtained dysregulated proteins both in CSF and plasma. We next examine which hallmarks of neurodegenerative disease were associated with the significant proteins in the CSF comparison of symptomatic versus healthy controls, as here is where we obtain the most significant results. And we see that the most prominent proteins are involved in pathological protein aggregation, which are represented by the p-tau species. And then the next most represented pathway would be inflammation, which is alongside the protein aggregation, can indicate early disease changes. Then we looked at the similarities between the CSF and plasma of the symptomatic mutation carriers versus healthy controls. And what we saw is that all dysregulated proteins in plasma are also dysregulated in the CSF. And additionally, 12 proteins were also dysregulated specifically just in CSF. This means that CSF shows a high sensitivity to capture CNS-related changes as it is in closer proximity to brain. And at the same time, the overlap between both fluids support the potential of certain plasma proteins to reflect central processes. So, a total of 11 out of 130 proteins, which corresponds approximately to 8.5%, showed concordant levels between CSF and plasma. In this plot, we see the significant associations of these proteins, which are mainly represented by p-tau species and brain-derived p-taus, and followed by neurofilaments. In the red dots, with the red dot, you can see the significance, which is represented as in a logarithmic scale as adjusted p-values, and then the blue bars represent the strength of the association of the correlation coefficient of Spearman correlation. And we had 26 samples matched. Altogether, these results reinforce the robustness and reliability of these markers across CSF and plasma, supporting their utility as consistent biomarkers of CNS pathology. So, since no significant proteins emerged in the asymptomatic mutation carrier versus healthy control comparison that could indicate early disease changes, we decided to examine the overlap between the symptomatics versus asymptomatics and the symptomatics versus healthy control comparison. And we found out 3 specific proteins that were altered between the asymptomatic phase and the symptomatic phase, which were ENO2, neurogranin, and SFTPD. Here you can see a clearer visual representation of these 3 protein levels. And what we saw is that in the asymptomatic phase, these 3 proteins are decreased, and then at the symptomatic stage, they increase. We next wanted to investigate whether these proteins were associated with measures of disease progression, such as estimated years to symptom onset in the asymptomatics, or estimated or years since the symptom onset in symptomatics. And the only proteins that showed significant correlations were neurogranin, which were positively correlated with AO, and SFTPD, which were negatively correlated with AO. And unfortunately, ENO2 protein did not show any significant correlations. It could be that a possible limitation is that we have a small sample size per each subgroup, and this can reduce the likelihood of finding significant associations between the protein levels and the time disease variable. Moving on to the discussion, we searched for literature of these three proteins, and we found out that neurogranin is a synaptic plasticity and is involved in synaptic plasticity and memory formation, and several studies have found this protein elevated in CSF. However, other studies found out this protein to be lower in the plasma exosomes derived from Alzheimer's disease and mild cognitive impairment patients. This could mean that neurogranin has different dynamics depending on which biofluid are you looking, and maybe that's the reason that we did not find neurogranin significant in our plasma results, maybe. Then the SFTPD is involved in innate immunity and inflammatory responses, and although is less studied in Alzheimer's disease, a recent plasma biomarker study correlates it positively with age. And another study highlights that is higher in serum levels, SFTPD is higher in serum, and this is associated with an increased risk of dementia and mortality, which suggests a link between innate immunity and neurodegeneration. And finally, ENO2 is involved in glucose metabolism in neurons and regulates the GSK3 beta pathway, which has an important role in tau phosphorylation and A-beta deposition. Literature found lower levels of this protein in Alzheimer's disease, although in our cohort, the levels of this protein are higher in symptomatic mutation carriers. So our results seem to suggest a protein level pattern in U shape, as the proteins are decreased in the asymptomatic phase, but then increase in the symptomatic phase. Together, these proteins point towards a combination of synaptic plasticity, innate immunity, and glucose metabolism that can be key mechanisms underlying transition from the asymptomatic stage to a symptomatic one. And this fits with other stress or early pathology markers, which often show an initial decline before a compensatory response emerges. Finally, I would like to conclude our study saying that we identified proteomic alterations in autosomal dominant Alzheimer's disease, both in CSF and in peripheral blood, that are associated with early neurodegeneration. Almost all proteins we found are upregulated, and in CSF we found a greater number of differentially expressed proteins than in plasma, although I found really valuable studying both fluids. We described potential biomarkers of clinical conversion in CSF, which were neurogranin, SFTPD, and ENO2, which seem to show a U-shaped pattern. And finally, most protein levels are compartment-specific, and only a few markers reliably reflect CNS pathology in blood. Just to finish, I would like to thank the Center for Human Genetics of University of Oxford for performing the experiments and analyzing the raw data, the Alamar Biosciences support to resolve our methodological questions, and to all of my colleagues of the Unitat d'Alzheimer y altres trastorns cognitius from the Hospital Clínic de Barcelona, especially to my two PhD supervisors, which were Dra. Sánchez Valle and Dra. Antonell. And thank you very much for your attention.
Thank you, Aina. So I think we can open for debate now. Are there any questions in the audience? There's one here. I think we have a mic somewhere. Do we? Yes, coming.
So now that we have these different methods of analyzing these biofluids, is NuLISA that we should be using, or Olink, or so how are we going to handle this?
Maybe I'll start. First of all, I have no stock or equity or anything else in NuLISA or anything else, so I will say that, or Alamar specifically. So I think I don't know. It depends on the question that you want to be asking. So that's number one. And number two, I think you're starting to see a revolution that's occurring with these proteomics. And I would just take the step back is that I think these proteomics can really characterize, and with now small amounts of blood, whatever way you want to be doing it, but a large panel that, you saw that it is similar to a lot of the mass spec. You've also seen that it gets a multiplex. Now there's actually even a 220 panel for ELISA and doing this. So there are constant new changes that are going to be. I don't know if ELISA is going to be the one, 6 months from now, something else could be the next one that could be there. But I think these proteomics really give you a much clearer picture. There are differences, as you were seeing with the CSF and the plasma, but it gives you a first approximation, specifically in the plasma, if you can't get a lumbar puncture or it's difficult in certain populations. So I do see that as the new mean. That also takes out of the equation, and again, I can't believe I'm saying this as a person who's did his PhD in imaging, is that you don't need to do PETs. I mean, you'll probably need to still do PET scans or other things for confirmation or following that. We don't know how to follow these markers with an intervention. They're so new in doing that. But you don't have to do repeated PET scans. I mean, like you wouldn't have to do, to get all these kinds of variables, you'd have to do an amyloid scan, you'd have to do a tau scan, you'd have to do S1P, or sorry, some other neuroinflammatory TSPO scan. Here you have it in one quantity. So I think that's where the real gains are from this. What's going to play out? I don't know. I mean, it could be different ones depending upon the question you're asking.
Yeah, and I think that it can, with this type of analysis, as they are so sensitive, you can get a more precise picture also of the biological mechanisms, because it is true that we already have well-established biomarkers, as I showed the correlations between CSF and plasma of the p-taus, neurofilaments. These markers already work very well for disease progression and disease diagnosis, but in the field of more basic bioscience research, it's really important that we can have these specific technologies, because this way we can detect proteins that were not detectable with other technologies, the concentrations, for example, with Simoa or with ELISA. So I think that we are in a good pathway, to try to find new markers that can help us uncover the disease pathogenesis.
I think there's another question here.
Or should I?
Okay.
What, Sam?
All right.
You had a question, Sam?
Yeah, sorry, I didn't see you there.
All right. And sorry, it's a long one. Bo, really cool stuff. Two related questions. For the Down syndrome NULISA panel, did you exclude patients with non-Down syndrome comorbidities, compromised iron status, autoinflammatory conditions, stuff like that? And if you included them, did you see differences from those conditions?
Yeah, so great question. So this is a patient population. So some of those individuals do have other conditions than when they're coming to see me in the clinic. I did not exclude any individuals. Anybody who was coming to my clinic who was then asked to be, if they wanted to, they'd be willing to have us collect blood and do some very simple tests like a DSMSE and some other kinds of tests on them. So it was very, very, very simple. We tried to make it within actually like a 30 to 40 minute kind of period of time so that we would be able to capture those individuals. That is across the spectrum. We did not exclude or did not include those as variables into some of our analysis. We did look, I didn't show this, we did look at some BMI effects. There are some BMI effects. There are other kinds of things that probably need to be considered, but this was a first pass on some of this kinds of stuff. But no, we did not exclude those.
So you're hitting a lot of acute phase biomarkers, for example. So
Correct, but that's true. But I would say, on the contrast, these were all stable individuals. But you're right; there could be some blips that are occurring in individuals that could be present. Yes.
Do you have standard lab panels for the same visits? White cell counts, hematocrit, that kind of thing?
We do on some, but not all. And then our colleagues in the University of Colorado, that was from their collection. So I don't know if they have all of those values.
Yeah, because those might explain some of the variability. Thank you very much.
No.
We had here in the front, maybe later there in the back.
Thank you all for those talks and the discussion. This is a question for you, Bo, because I'm trying to think through your presentation and then the discussion yesterday in the first session with what Natalie had presented around GFAP maybe driving the amyloid to tau, and then looking at kind of your timeline of when you see GFAP elevation in the context of tau, and it doesn't look like you necessarily are seeing it preceding the increases, the upswing of the tau later. And so can you talk a little bit about how kind of what Natalie had presented, and I don't know if she's here, but maybe she could weigh into kind of what she's seeing and then what you are seeing and I realize they're done very differently, but.
No, great question, Heather. I don't think we're saying things that are different. We're actually saying things that are more similar. So we first have to understand that first of all, the p-tau 217 is actually a marker of amyloid and tau. And when we look at the relationship specifically in Down syndrome, it associates with both. And that's actually been done in studies with Oscar and other groups of looking at that. So it actually correlates with both PET amyloid and PET tau. So the 217 is kind of this marker of in between. I would say that we're still seeing the GFAP is going up at a little bit of a later phase compared to when some of the other amyloid changes, like the A-beta 42s and 40s, and those kinds of changes that are present. I would say that it's probably, we didn't look at the temporal dynamics of that. That's what we're going to be looking more at. I don't think it's still saying that it is going up in the 40s, and that's probably when those same kind of changes are occurring. So I actually think that there's more similarity. And then we've done work literally looking at that, looking at how GFAP kind of mediates that relationship. What we don't have in this panel is MTBR. So the measures that we have are the 181s, the 217s, the other kinds of measures. We don't have a specific tau measure. I think there's a move to try to develop that in ELISA, but that's not yet present on that. So really definitive markers of tau we don't have in that panel. I hope I answered your question, Heather.
In the back first.
Yep.
Okay. We can talk more.
Thank you for great talks. I'm interested in your thoughts on the dynamics and exchange of the different protein levels between different compartments. Extracellular space, of course, we don't measure, CSF plasma, and not only cross-sectionally but longitudinally. To give an example, there is, let's say, p-tau 217 can be going up in plasma while it's going down in CSF. We were talking about clearance. But in general, what are your thoughts on that? Because, of course, a single cross-sectional sample in one or the other compartment may not be that informative on its own.
Yeah, unfortunately.
And what might be driving those variations, right?
Yeah. That's a really good point. I would love to have the longitudinal data of my patients because this is a really interesting thing to study. Unfortunately, I don't have it, or my N sample size would be so small and I cannot look at it. But it's really interesting because I think that it could change, longitudinally, but I don't know really how. I don't know, Bo, if you have any clue of how could it change with time, the disease dynamics between CSF and plasma.
Yeah, I still, we're in an infancy, and you're hitting a really good point. I mean, invite me back next year, and we'll get you the longitudinal. We're just starting to collect that, and you did see though these are reliable, but we also want to look at the ideal situation, and Julie, Dr. Wish, and I have had these long conversations. Is there are these dynamics, even in the mass spec and even in any of our measures. And the key thing to be kind of looking at this, kind of separate this out, is to repeatedly sample individuals over a short period of time to understand the stability of that measure and then understand the dynamics that are going on. And that would be wonderful. I don't know if we can do that, but I would charge anybody, if we can get repeated blood samples from individuals, or if we can do the CSF. I do lumbar punctures, but I don't think most people would want to have, weekly LPs or anything else. But if they were willing to do that, then we could start to answer those kinds of questions, because I do think that there are some dynamics that we're not capturing on just the cross-sectional analysis.
I think there was a question here in the front.
Hi. So really nice talks. And I just wanted to go back to Heather's comment on this interaction with GFAP and tau, because I think it's really interesting. I love Natalie's talk yesterday. And I think on the one hand is that GFAP in our data in DAPNI starts to diverge from euploid like in the very late 20s, early 30s, so before tau. But the thing is that it has like a huge dynamic range, and so the initial elevation is not huge, and then it gets a really large dynamic range. And the other interesting thing, and maybe Danny can comment on that, is that we have another work under review on the different phospho-tau species in plasma and in the CSF, and it seems like it's very compressed. So this thing that we're seeing in autosomal dominant AD, where you could kind of stage with 205 being later than 231 and 217 in the middle, in our data is kind of more compressed. And I think that it fits nicely with prior work by Bo, no? That says that it's like more compressed. After amyloid positivity, you have an earlier tau. And the other interesting thing is that we're seeing more phospho-tau in Down syndrome than in sporadic AD. So it seems like, I think it fits together with this story in that GFAP starts early. I think this is something that is emerging now. I know that Tariq Pascual is collecting now a huge data set with thousands of individuals, multi-cohort, and this interaction between GFAP kind of facilitating in amyloid positivity the tau. I think that is emerging and converging across populations, and I think that in Downs this is particularly strong with higher phospho-tau levels and a more compressed story in Down syndrome. I think this is really interesting.
Just a quick question, maybe to you and to you, is so then would you think that the GFAP dynamic range, you said it's quite broad, do you think very, very subtle changes in it that may not be detectable or maybe very minor on something like what you shared, Bo, could be driving to that, driving the mechanism?
I don't know, but I think it might be sample size. I mean, you have 180, and so probably in that age range is and the study I was referring to, I can't remember how many we had, Danny, but it was like, I don't know, 400, 600. So it was, I think sample size probably had something to do with it. So these early elevations when the dynamic range is not huge, is probably the reason. I don't know.
I would agree, but I think there are different inflammatory markers that are changing. So like the GFAP is not elevated early on. It is some kind of measure that's starting to change, before or during when amyloid's occurring, or really this uptake, versus other inflammatory markers that are already elevated very early on, which is a little different than what we were seeing in the autosomal dominant. And so there are some similarities, but there are also some differences between these two genetic forms of Alzheimer's disease.
Do you have any thoughts on the basis for high levels early life and then lower levels and then going back up again across several proteins that you guys showed, where that midlife period, there's a decrease in levels of certain proteins that you found, both CSF and plasma. Why do you think we see this pattern?
I think that it can be more easy to explain maybe for inflammatory markers like the SFTPD that I was talking, because sometimes these inflammatory stress markers, before the onset of symptoms, they tend to decrease, the body loses their protection, and then in this more symptomatic phase, the body reflects a more compensatory response. I think that is more explainable in inflammatory markers because it is reported that there are some proteins that follows this path. But for example, for neurogranin, which is more involved in synaptic plasticity, I really couldn't ask why in a symptomatic phase there are a loss of these protein levels, and then in the symptomatic phase it increases. I think that we should get deeper in these proteins, first validate them, and then to study like a more functional way and try to understand why they follow this U-shaped pattern. But I think that the inflammatory markers could act that way.
I want to thank the panelists for presenting on this really exciting emerging work. I think it also really highlights the biological complexity that we're looking and allows us to kind of expand out past just our very traditional A-betas, some of the p-taus, to kind of look at more of these nuanced changes. I'm wondering, again, for the panel, were there any markers in particular that you felt like were emerging or maybe we should be looking more into? I think, for so long we've really honed in on the 181s, the 217s, but was there anything through what you're seeing that maybe we should start focusing on or doing a little more kind of digging into based on what you're seeing in your work?
And that's the question that I also wanted to ask Bo. I saw that, for example, the ENO2 protein, it's really interesting because I think that he saw it downregulated in Down syndrome, but in my results in ADAD, it's upregulated in the symptomatic phase, no? And also in the literature, ENO2 has been reported in sporadic AD, also high in plasma, I think. And it could be a really interesting protein to study because it's a glycolytic enzyme in neurometabolism. But I'm really curious about its dynamics because why is it in Down syndrome, downregulated and sporadic, and my results in symptomatic seem to be the opposite pattern? And that's maybe one protein to focus on because several studies report it. And yeah, I don't know. What do you think, Bo?
I don't have a good answer. I mean, we're still trying to figure out what all these proteins mean and where they're pointing us to. And I would love others to also comment. I don't have, I mean, that's why we need to do more. And then we need to do, and what this is also suggesting is how to compare this versus sporadic versus autosomal dominant. And if we have them all collected, and then we can kind of do those kinds of comparisons to really tease out if there are differences and why there are differences between them. And then we need it in larger samples, as Juan was talking about. I would say that there are other inflammatory measures. If you looked, I was talking about YKL40, so microglial markers seem to be very important. So the GFAP is more of an astrocytic, watch my hands, air quoting that, but it is more of an astrocytic marker. But you saw other inflammatory with microglia having to seem to be important. And in fact, that microglia could be sometimes upregulated as a protective factor, but then downregulated later, downregulated, trying to control stuff, but then gets too much and now starts to be upregulated and actually is a toxic mechanism that's present. So I'd say there's inflammatory. You also saw some different cytokines that were very important that we're starting to see. Not all of them are interferons that are not all on chromosome 21, but on other chromosomes that are there. And then you also saw, which was very interesting, is breakdowns in the blood-brain barrier. And you saw a lot of these other factors that we don't have yet great measures, at least on the MRI kinds of findings to do that. But this is suggestive that there is this cross-exchange and this measures of why there's CSF could or could not look like what you're seeing in the plasma. So I think this gives you more food for thought. And then really looking at some of the work that you were presenting and what also Laura was presenting is that I don't think, and I completely agree with her, I don't think it's going to be just one measure, but if we could create a panel of 4 or 5 and we take that together, then we can have a better staging of an individual than any one marker. And we've been, because 217 is great, and I love 217, but the point is that it gives us one picture, and it's kind of looking at the elephant in one way, and it's a much more that we want to capture of that individual. And so this combination of ones is probably going to be the way that we do it. Same thing how we're thinking about combination therapies. We're not going to just probably be giving an individual a single therapy. We are probably going to be doing a combination. And I go back from my other role when I do infectious disease, when I take care of HIV patients, we don't give you one drug, we give you a combination of drugs and we stage you at those different periods. And so looking at these markers, we'll be able to kind of give you a more personalized therapy. So I see that as the wave of the future.
Thank you.
And if I can just go on in this line, do you see these proteomic platforms such as NOLISA or others like Olink or SomaScan as platforms for discovery of target candidates of biomarkers, or maybe a platform that we will use in clinical applications to detect patterns or hallmarks of different pathways that we can measure or detect the effect of clinical trials? How do you see it?
I hope it was the second one, but I don't know. I think that nowadays we are most close to the first thing you said. As I said before, it can help us to uncover new targets for research and biology experiments, no? To try to uncover the biological mechanisms of these diseases. But I think that we are still far away from implement these new technologies in a more clinical setting. Yeah, I think it's still far.
Yeah, I think I agree completely. I mean, we're in a discovery phase of this. If you also looked at all of our slides on the NOLISA, it's MPQ-2, right? It's not an actual measurement. It's the NOLISA value that is there. They are working right now to get it so that it would be to those kinds of measurements that we could have at that. We're still in an infancy, but it does allow you to start thinking about how different pathways or different interactions and then different targeted focuses. But I would still, and I'll defer to Mike, I don't think we're yet at the plasma marker for this, but I'd love to hear his comments.
I was going to say low-hanging fruit here with this new platform would be, particularly for the Down syndrome population in clinical trials, is to look at correlations in the NOLISA platform and microhemorrhages and ARIA. There's a high incidence of this in this population. And if you're able to avoid the use of MRIs to do the monitoring, which is a huge burden for this population in particular, I feel like that would be an easier goal to reach than understanding inflection points across so many of these different mechanisms. I mean, Bo, you had everything in there: inflammation, neurodegeneration, the entire ATN. That's great, but in terms of clinical trials efficiency and safety and reducing burden on this population, I would look for simple correlations in predicting microhemorrhages, ARIA-E, ARIA-H, just as part of the natural progression of AD in this group that has high incidence of CAA, while you're working out the fundamentals of inflection points across biomarkers.
I just had a quick question for Bo, because I was curious.
You can ask me about the other APOE4. I can try to cover Julie or Laura.
I just wanted to know if have you replicated your results on sporadic AD, or have you performed any ELISA on sporadic AD? Because we did, and what we found out is that comparing to ADAD, no? That all ADAD significant proteins were also dysregulated in sporadic AD, but in sporadic AD we saw a greater number of dysregulated proteins that we did not capture in ADAD. And I just wanted to know if you have performed this comparison and why it seems to work better in sporadic AD than in autosomal dominant Alzheimer's disease.
We haven't done that yet. It's great.
Okay.
Carlos Cruchaga has the sporadic, and that's been one of our next topics, is actually compare these three different but similar diseases and what is up or what is down and what we can see. There's a large sample that he has. I mean, it's in the numbers, and if we get more and more, and if there are people who are willing to do that, this is this other shameless plug. I'm going to follow you, Jasmine. And if you have those samples, we would love to work with you or work with others to do that. And we'll make all of this publicly available. It's part of Include. So this is all going to be available to others as well to utilize. So my answer to you is it's still early phases. A lot of work needs to be done. And Mike, with regards to imaging, I love the idea. We've just been comparing it in like 20 or 30 individuals who also have PET scans. It does seem to be highly correlated. We don't have as much MR on some of these people, but I can start to do those kinds of things. But if you have suggestions for us, we'll glad to do that. Glad to help in running those kinds of studies. Do that.
I have one question. Do you have time?
Yeah, one last question.
Oh. So one comment about discovery. So, I mean, these in the ELISA you find these markers because they were already predefined in the panel as important for Alzheimer's disease, and there are some markers that are surprisingly for example, microglial markers that you would expect really to be important in Alzheimer's disease. They are not really very relevant, but I would like to comment on that. And the question is, do you think we still there is room in unbiased analysis, such as mass spec, for example, that really the true discovery method to find new elements? And I think was in your presentation, you had some mass spec data like that, where you could find like new markers to be included later in these panels. And so that's questions and comment, and also if you can comment on the microglial markers that perhaps I missed that you said they were important.
Yeah, so great question. So let me see if I can tackle that in two different parts. So for the roles of microglia, we did see measures like YKL-40 that seemed to be elevated and then really went up. It was already elevated at a younger age and continued to go up. There are other microglial markers. I can't remember all of them, but there are some, and I'm glad to show you or walk through the slides with you of other measures of microglia that are there. There was also CXCL8, which is another microglia or involved with microglia. It seemed to be, when we looked at the correlation analysis, that seemed to be very important with other amyloid species that were there. So I do think that there is a role of microglia on that. With regard to the second part, yeah, there are definitely new discoveries. The panel is what they've generated. They're going to generate more inflammatory. You can do a whole inflammatory panel. I feel like I'm a huckster for ELISA, and I'm not, all right? This is full disclosure. But there is an inflammatory panel. So this is the CNS panel, and they're increasing the number of inflammatory markers. And what I have been told, now again, this is what they've told to me, but we'll see if that really works, is that if you have a new measure, they're willing to develop it and really are willing to do that. It will cost you some money, but that can be added and you could be looking at that. So I think that there's still a role for mass spec. I'm not saying that there isn't. I think it is great potential. However, the marketability of doing mass spec is really, really tough, and you need over like an ml or more of blood to do mass spec. And the mass spec is really when we're focusing, like you'll get the 217 or you'll get an ApoE and you'll get a couple, but you won't get the full panel that you often get when you're looking at the ELISA. So it's, what do you want to do? Do you want to do depth or do you want to do breadth? You know, and this is this kind of question. And so you can do both, and you can be kind of looking and kind of targeting, and then you could go deeper if you wanted to figure out, and then, all right, I want to cover as much as possible too. So I think they each have their own places. The one other thing with the Alamar is that it's much more available than, you know, you really need places that know how to use the mass spec and do that. It's a really intensive procedure. So I definitely see a role for it. And I don't also get money. C2N is at WashU, so full disclosure. And my former chair created it, but needless to say, I have no more money or royalties in that too, full disclosure. But I think there's a role for both.
Okay, thank you very much. With that, probably we have come to the end of the
I
session.
think we can close the session now.
Thank you. Thank you all for your
Thank you very much.
Great job.
Us poso aigua.
Així jo vaig a ficar-me aigua també.
Oh, it's so bright.
Yeah.
Our mics are on. Yeah.
Ara se sent, ja? No.
Well, hello, everyone. We're just going to get everyone some water, get us settled. The lights are so bright up here. Wow. Now I know why Bo looks so starstruck. You were like... Well, welcome everyone. I'm Sarah Walter from the University of Southern California, Epstein Family Alzheimer's Therapeutic Research Institute. That's a mouthful. I'm so pleased to be here today, and I want to thank the organizers for making space in the agenda to hear from our families living with genetic causes of Alzheimer's disease. So this panel is really focused on their experience in clinical trials. It's so important for us as researchers to understand what the experience of being in a study is like. I know they have a lot of wisdom to share with us that we can all learn from to make our studies more meaningful, more impactful, and more supportive of not just the individual that's in the study, but the entire network that supports them in their lives. I want to thank the organizers, Bea Bosch Capdevila and Raquel Sanchez de Valle for organizing the session. I want to thank Neus Falgas Martinez for doing the live translations. This is no easy feat, but it's wonderful for those of us that don't speak Spanish to be able to hear what your family's members are sharing with us. So our discussion over the past few days is really focused on some very exciting advancements in the field. We've got new trials beginning. We've got results from ongoing studies, longitudinal studies, new protein biomarkers. But for this session, we're going to focus on what's at the center of what makes advancing science possible, the people who take part and their families. Taking part in research is more than signing that consent form and giving a blood sample. Our families and our participants are exchanging a very personal sense of trust in each of us as the primary researcher that's connecting with them, but also the broader research community. Some of you in this room may not interact with families, and we know that this is really valuable for you as well to hear directly from them. So our goal is to develop studies not only that are on the cutting edge of science, but that are built in partnership with the participants and our families, because ultimately the quality of our research depends on how well we care for our participants, both while they're in our study and then afterwards. So we will start with listening to our panel around the first step, and I'll turn it over to Bea, who will be asking questions first in English and then in Spanish. So Bea, take it away. Thank you.
Thank you, Sarah, for this introduction. And building on what Sarah just shared, today we'd like to focus on something really important, what it actually feels like to take part in a clinical trial. And beyond the medical side, there's a lot more to it, and the decisions people have to make, the doubts to come up, and the impact on family, and what day-to-day life looks like. To start, I'd like to ask each of you to briefly introduce yourselves and who you are and what your connection is it to clinical trials or with the young clinical trial. Buenos días y muchas gracias por acompañarnos. Si os parece, ¿no?, lo que comentábamos, primero haremos las preguntas en inglés y luego me pasaré al castellano para que todos nos sintamos, ¿no?, podamos ir siguiendo la conversación y nos sintamos cómodos. Siguiendo un poquito la introducción que nos hacía Sara, ¿no?, pues hoy queríamos centrarnos sobre todo en algo muy importante, ¿no?, que es cómo se vive realmente la participación en un ensayo clínico. Y más allá de la parte médica, ¿no?, hay muchos otros aspectos que nos acompañan en estos ensayos clínicos, ¿no? Hay muchas decisiones que tomar, las dudas que surgen, el impacto que causa en la familia y cómo es
el día a día, ¿no? en la convivencia con todo este ensayo clínico. Y para empezar me gustaría pediros que si os podéis presentar brevemente y quiénes sois, qué relación habéis tenido con el ensayo de Diane, ¿no? Y si estáis aquí, pues bueno, hasta donde vosotros os sintáis cómodos, ¿no? Si estáis aquí como pacientes, familiares, como acompañantes de alguien cercano y en qué momento estáis de este proceso o qué momento habéis vivido. Así que nada, el que... Ángel, sí, no, Santi, venga, va. Santi.
Soy Santi y soy el acompañante de Alba en el proceso del estudio.
He is Santi, I will be translating. He is Santi and is the partner of Alba.
Yeah. In life and in the study. Alba, ¿sí, igual?
¿No?
Sí.
Yo soy Alba y bueno, y estoy aquí porque tengo la mutación, tengo la enfermedad, estoy con un deterioro cognitivo y luchando con el tratamiento para que esto vaya lo mejor posible.
Her name is Alba. She is a mutation carrier and also started with cognitive symptoms recently. And she is also in the clinical trial to try to improve the progression of the disease.
A ver, yo soy Ángel, sí llevo muchos años luchando con esta enfermedad. Soy nieta, hija, sobrina, hermana y tía del Alzheimer. No tengo la enfermedad, pero sí que, bueno, pues la llevo en la vida.
Her name is Angel. She has battled a lot with the disease because she has several family members suffering from it.
Nada, muchas gracias. Y una vez hechas las presentaciones, si os parece, empezaremos por el principio, ¿no? La decisión de participar en un ensayo clínico. Salto un momento al inglés. Once we've done the introductions, if that's okay, let's start at the beginning, the decision to take part in a clinical trial. When the option of a clinical trial comes up, what tends to weigh most of the moment in that moment when deciding whether to participate or not? Una vez, ¿no? Empezamos con la decisión de participar en un ensayo clínico, cuando aparece esta opción encima de la mesa, ¿no? ¿Qué es lo que más os pesa en ese momento para decidir si participáis o no? ¿O qué es lo que os llevó a participar en el ensayo clínico, ¿no? Si fue una decisión más emocional, más racional, algo que compartisteis a nivel de familia, que se tomó entre todos, y qué fue lo más difícil de decidir, ¿no? En aquel momento.
Bueno, sí, vale. Yo me tiré a la piscina directamente, no hay otra cosa que hacer. No había otra opción para mí.
Fue una decisión.
Sí.
She didn't have to think about it a lot because it was for her like the only option to try to move forward.
Yo lo hablamos en casa y apoyo 100%. Era decisión de ella y apoyarla.
They discussed the options and he just supported her on her decision.
Y nosotros, pues, fue con Amladulos, con mi hermana pequeña, y pues fue una gran decisión a nivel para ella, de forma particular, y a nivel familiar pensamos que era, bueno, muy beneficioso.
Her little sister was first involved and she also supported her and the rest of the family members.
Thank you for sharing that. Bueno, gracias por compartirlo, ¿no? Ayuda mucho a entender lo importante que es esta decisión, ¿no? No es fácil, pero ¿no? Todos los que estáis...
No, es una decisión importante. No es fácil porque ya hay muchos cambios, entrar en la dinámica del ensayo, pero bueno, yo creo que lo más importante es que la persona que está enferma, si lo vive con mucha positividad y mucha fuerza, pues los demás hemos de acompañar en todos los momentos y en todas las horas que podamos. O sea que es una esperanza No.
It was a bit difficult at the beginning because it's a difficult decision to make because it has implications for you on how you organize your life on the next days. But if the patient stays very positive about it and wants to do it, the family can do anything else that give their support.
Bueno, una vez tomada esta decisión, ¿no? Y una vez adentro, o en el momento que os fuisteis a enfrentar, ¿cuáles fueron los miedos o las principales dudas que os surgieron? Y si echasteis algo en falta o algo que habría hecho la decisión más fácil en aquel momento.
¿Contesto yo?
Sí.
Bueno, nosotros la verdad es que el equipo que tenemos ha sido...
Perdona que te corte un segundo, lo comparto, disculpad. Thinking back to before joining, what were the main doubts or fears and what helped you feel ready or not to take that step? And looking back now, was there anything you felt was missing or that would have made it easier to decide it? Sorry.
Bueno, en nuestro caso, el equipo médico que teníamos para mí fue fantástico, a nivel explicación, a nivel coordinación, a nivel acompañamiento, a nivel amor, y para nosotros fue, bueno, fue difícil por situaciones y bueno, porque no siempre el humor es el mismo y por tanto la fuerza también decae, ¿vale? Pero para mí fue básico entenderlo, que nuestros doctores nos lo explicaban de forma que podían entenderlo y por tanto seguíamos. No, o sea, ¿cambiaron cosas? Sí, cambiaron cosas a bien.
Es la siguiente pregunta.
Vale, pues ya te la he contestado. Muy malamente.
No, súper bien, no.
Even though the decision may be a little bit difficult, they have a lot of support from the medical team and they explain very well how the clinical trial works, the disease, etc. And all this information facilitated the decision and all the things that they had to do along the way. So they are grateful for that.
Miedo al principio, pero después, una vez vas viendo la forma de trabajar, comunicas con los doctores y eso, todo perfecto. Excelente.
At the beginning, they feel a little bit of fear because of the unknown, what was going to happen, but because, again, of the support of the medical team surrounding them, it kind of got easier.
And once you actually made the decision and entered in the trial, how did it compare that what you expected on what's been the impact on your day-to-day life and your family? Una vez tomada la decisión y adentro del ensayo clínico, ¿en qué se ha parecido diferenciado de lo que os esperabais o de la idea del principio ha cambiado mucho a lo largo del tiempo, ¿no? Durante el ensayo? ¿Y qué impacto ha tenido en vuestro día a día? ¿No? ¿Cómo se ha introducido el ensayo en vuestras rutinas o cómo ha modificado vuestro día a día y lo veis, ¿no?
A ver, modificar en parte pues tampoco mucho porque él sigue... laboralmente él se tiene que organizar más para cuando pues tengo las visitas, pero...
Y, por suerte vivimos...
Sí, estamos en pueblo y tenemos la familia que, pues, por ejemplo, hoy que veníamos aquí, pues está en casa de los
niñas, tiene dónde quedarse.
Sí, tiene...
Alba y Santi tienen una niña pequeña, vive fuera de Cataluña y las visitas se están haciendo a nivel domiciliario en casa. No, Alba has a little child and she's receiving the treatment at home. And well, she has a extensive family who covered them when they need it, for example, today to assist to the meeting.
I think that you translated.
Perdón.
No, that's fine.
No sé, Ángel, si quieres añadir algo más de cómo tuvisteis que modificar el día a día, ¿no? En el momento que los participó en el ensayo clínico.
Bueno, para mí como era tan importante, yo tenía la libertad que quería en el trabajo. Cumplía mi trabajo, mis objetivos y, por tanto, mi vida de mi familia era lo primero. Sí, que modifiqué, sí, pero estoy encantada de haber modificado.
She got to adapt the job schedules and all the daily routine, but she's happy to have done that, and she felt that it was necessary.
Prioridades.
Priorities.
Thank you. That really helped us understand how much everyday life can change once you are in the trial. And that naturally brings us to something many people talk about in these situations, that it's living with uncertainty. How do you deal with the uncertainty of not knowing whether there will be a personal benefit? And as time goes on, do you feel it becomes easier to live with, or does it change in some way? Gracias porque, ¿no? vuestras experiencias nos están ayudando a entender mejor, ¿no? Cómo se va adaptando el día a día, ¿no? En la aparición del ensayo en las rutinas y cómo va minando esto, cómo nos vamos adaptando, ¿no? Y eso nos lleva a algo que muchas personas, bueno, y aparte de esta primera parte, ¿no? Eso también nos lleva a algo que muchas personas comparten en este proceso, que es la incertidumbre, ¿no? De saber cómo convivís con la incertidumbre, de no saber si este esfuerzo, esta participación, recibir este tratamiento, ¿no? Nos va a comportar un beneficio personal. Y cómo habéis vivido esta incertidumbre, cómo la estáis viviendo, y con el tiempo si sentís que esta incertidumbre se aprende a llevar mejor o va variando con el tiempo.
Bueno, con los años varía, porque con los años ves las cosas diferentes. Pero, bueno, yo creo que uno empieza el estudio con el ADAD con mucha esperanza. Y en mi caso es una esperanza presente y de futuro. Por tanto, bueno, creo que hay que vivirla y toda la comunidad que estáis aquí a nivel médico-científico habéis de saber que para los enfermos y para las familias estas enfermedades genéticas que encadenan generaciones es importantísimo vuestro trabajo y nuestra fuerza.
How the participants live the clinical trial, it changes along the time, but especially at the beginning, they live the experience with a lot of hope for the present and for the future. And she highlights that everybody that is working in the community and the clinical trials and developing these, the investigators are really, really important, and they're doing a really important job. So she wanted to transfer that to you.
Lo mismo,
Sí,
es
lo mismo.
esperanza para hoy y sobre todo para el futuro.
He echoes Angel's in regarding how hopeful it is in the present and in the future, especially.
And if we stay with this moment, no, because this uncertainty, this is something that stays present through the whole journey. Just the last question, but I'd like to move to something very practical. Thinking about other people or families who are right now unsure about whether to take part, what do you think researchers or medical team, maybe you in part answering the previous question, could do better to help them feel more informed, supported, and resolute? And if you could change just one thing from that very first contact, what would it be? Bueno, enlazando un poquito con la respuesta anterior, ¿no? Gracias porque nos ayuda mucho a entender cómo se convive, ¿no? Con esta incertidumbre a lo largo del tiempo y transmitirnos a la comunidad científica, ¿no? esta importancia que tiene para vosotros. Yo creo que todos los que estamos aquí es lo que nos mueve, ¿no? A estar aquí, pero sí que es verdad que, bueno, poderlo poner en común yo creo que es muy importante, ¿no? Que haya voz de las familias, este mix. Y la importancia, ¿no? De esta incertidumbre de darle peso. Y como último, para haceros una pregunta como más práctica, ¿no? Pensando en otras personas o familias que ahora puedan estar en este momento dudando, ¿no? De si participar en un ensayo clínico, cómo esto les va a minar en su día a día, o los miedos, ¿no? Todo esto que hemos ido comentando. ¿Qué deberían hacer mejor los investigadores o qué deberíamos hacer, ¿no? El equipo médico para que se sientan más informados, acompañados o más tranquilos? Y si pudierais cambiar alguna cosa desde el primer contacto o algo que hayáis echado en falta o algo que, no sé, incluso aquí abiertos a los procedimientos del ensayo, información que os hubiera gustado tener, bueno, malo, qué es lo que os gustaría, si tuvierais que rediseñarlo, ¿no? Hay algo que...
En principio, yo todo bien, no sé, a ver, sí. Hacéis demasiado ya. Sí. Yo creo. Por mi parte, sí.
Sí, yo creo que la información es muy importante a nivel de las visitas y la implicación, así aceptar, ¿vale? ¿Cómo podéis hacer que las personas acepten que están en este proceso? Pues es complicado, pero yo creo que es muy básica la información y después que la persona que esté delante esté receptiva y lo que hacéis a nivel familiar y a nivel enfermo. Quiero decir que yo creo que el núcleo es importante y yo creo que dais información y brazos para poder seguir, ¿no? Incluso sitios y personas que también te pueden ayudar fuera. Bueno, yo creo que al menos yo saqué bien que mi equipo nos ha dado muy buena información y ha respuesto a las preguntas. Sí. O sea, que no sabría que tendrías que mejorar. Yo creo que ya aquí sería la propia persona enferma o la familia que pueda reconocer. Es que no es fácil reconocer esta enfermedad. Os prometo que es muy difícil. O sea, a veces yo creo que las familias casi lo esconden. Es complicado, cuesta mucho. ¿Vale?
In general, they don't have complaints about the medical teams or the clinical trial itself. They think that the most important part is have the full picture of all the information about how this is going to work out in the next few years. And generally, they got that from the medical team. And this is very important to help them move forward into the disease, and also to help them to acknowledge the disease, because there is some tendency sometimes to try to hide the disease to others, to the society, and having the information helps for the patients and families to understand better, and therefore being more open to share or live this with some sort of normality.
También a lo mejor estaría bien haber algún departamento en que pudiera haber algún contacto entre familias. Sé que es difícil, ¿vale? Pero que no todo el mundo lo aceptaría, pero no está mal que entre nosotros hablemos un poquito o nos escuchemos, ¿no?
Angel says that it would be great if they have more chances like today to get together, patients and families, so they can interchange experiences and share the vision on the disease. And this is something that barely happens. I agree, and we have to find better activities or try to find the moments to get together and discuss more.
Intercambiar un poco experiencias y bueno, y la opinión que tenemos del equipo, de los ensayos.
No, y que está bien, que hay más gente que está como tú y eso, que no estás solo.
Insisto, es difícil, ¿eh? Simplemente es abrir a lo mejor un camino y la persona que está receptiva o preparada, porque también para poder hablar o bueno, simplemente encontrarse más que hablar, ¿no?
Sí, tengo que decir que es algo que hemos hablado varias veces y está en construcción, pero aún no está organizado. It's the thing that they are claiming. It's something that we have discussed as a team many times. Or sometimes it's difficult to find the resources or find a way to put together people from different areas of Spain. But now, also with the online opportunities, probably we can set this up at least locally.
Yo me ofrezco a montar también.
I will call you.
And just volunteers to organize it.
I think that Sarah
I have a
has
question that what I'm hearing is so consistent with what we hear from our clinical trial participants too. They really want to connect with each other. They have this shared interest in science, this interest in living well and fighting back. And then connecting with each other is one way that they know they can be empowered as part of the study. So just wanted to reflect that. So there's two kind of points of research that we hear from our participants can be sensitive and quite difficult. The first is when you get your hopes up that there's going to be a study for you, and then you find out that you're not eligible or you're not a good fit for that study. People talk about that as feeling that they were rejected, they didn't want me. Even when researchers say it's not a good fit, that's not how it feels, right? It feels very personal. So that's one thing, when you're not a good fit for a study. And then the other challenge that we've really encountered is at the end of a study. How do we take good care of our participants at the end of the study? So I know that's two questions, but I wondered if we could add that to the panel.
Sí, ell comenta que hi ha dos situacions, comenta que hay dos situaciones también que es importante, que es cuando alguien tiene la esperanza de que va a entrar en un ensayo clínico porque hay opción, pero después no es elegible y finalmente pues no acaba siendo. Y el otro escenario es qué pasa cuando se acaba el ensayo clínico, ¿no? Cómo cuidamos a partir de entonces del paciente y de las familias, que entonces se podría perder un poquito más el contacto, ¿no? Tan diario que se tiene cuando se participa en un ensayo clínico.
Sí, pero como la realidad de las familias es tan distinta entre ellas, y bueno, a mí una cosa que me abrió muchos caminos fue cuando vaig estar en Londres, al Dian Tu. Y el momento de las familias, sobre todo cuando fue el momento del lunch que me fer, yo solo escuchaba, porque habían tantas historias dentro de una historia que por eso digo que és molt important aquest contacte.
Sí.
Sí.
She was sharing that she was in the meeting of Diane 2 in London a few time ago, and she had the opportunity to meet other families, and she was like very attentive of what they were explaining, because they were sharing so many stories that she could relate and learn from, that she reinforces this idea that we have to meet more and discuss these kind of things.
Vale, o sea que no es fácil para los equipos médicos, porque cada uno vive la realidad de una forma muy distinta. Y claro, hay un sistema genérico, pero es difícil.
That some things might be difficult to approach in the medical consultation because, of course, you have some standards of care, but each family and each person is different, and this is something that can be more addressed in the community or having these small meetings.
Me dirijo a ella un momento en español, respondiendo un poquito a lo de Sara. Sara nos comentaba cuando alguien no puede entrar en un ensayo clínico, ¿no?
Bueno, en
Esto
mi caso,
sucedió
claro, en mi caso habían mi hermano, claro, tú estabas ahí. Sí.
In her family, there was two siblings that tried to enter to the trial, and one of them couldn't. And well, no, the situation, it's just to answer a little the Sarah questions. I don't know if what's the situation in that moment.
Bueno, estaba mi hermano y mi hermana, y a mi hermano pues ya no entró, y mi hermana sí que entró. Pues bueno, mi hermana le decía a mi hermano que ella no estaba enferma y mi hermano estaba enfermo. O sea, entre ellos sí que hubo un poco como de rivalidad, de que ella estaba como más enfermo en vez de pensar más en el ensayo, ¿vale? Y a nivel familiar, pues no había nada más a hacer, o sea que estábamos por los dos igual y con una teníamos más visitas médicas y con Xavi menos visitas médicas. Lo normalizamos, no teníamos, lo normalizamos.
Her sister participated in a clinical trial, but his brother was not eligible for the clinical trial, and this created some tension between them, some sort of rivalry. But in general, the rest of the family was supportive because there were no other option. They just have to show support in both cases.
Y a nivel familiar, fuera del ensayo, como lo hacíamos todo juntos, pues yo hacía y me llevaba los dos, o sea, no, por tanto, la vida siguió normal.
Outside of the clinical trial, he was continuing her normal life, but always with the two siblings.
No podían hacer res, ¿no?
Yeah.
And trying to answer the second question, Sarah, our experience is that patients feel not so good when they finish the clinical trial, no? And we try to follow them every 2 years. It's not exactly in all the patients, but for the reason we are encouraged to try to perform or prepare more support for them, because the feeling is not so good, no? I think that yesterday Dr. David Aguillon explained us the same situation with their population, his population.
I wanted just to mention that today we should have another participant that couldn't make it because she underwent a lumbar puncture a couple of days ago and had this horrible headache post the procedure, and just we wanted to thank her also for the availability even if Diane couldn't came.
Does anyone want to say anything else?
Yeah.
Maybe the
should we open the floor for questions?
Do you want to? I
No,
don't know
no.
how much time we have.
Pusemos el vídeo.
15 minutes, I
Yeah.
don't know.
Is there anything else that any of you would want to say to the researchers?
¿Alguna cosa que vulgueu demanar als investigadors?
Bueno, yo
que
quiero añadir que os pido, por favor, que no dejéis de luchar para estas enfermedades que son difíciles para las personas que quieren seguir viviendo un día a día. A todos los médicos, científicos y personas que puedan ayudar a todos los niveles a dar voz de lo complicado que es para las familias lo que vivimos, y que vosotros sois nuestra esperanza.
Sí. She asked the scientific community and the medical teams to keep fighting this disease, try to find solutions, and also to give voice to those who are not available, they cannot express by themselves, and make advance on this field.
Pensar que las personas que tienen esta mutación, ahora no me surt en castellà, héroes y heroínas. Héroes y heroínas. Y os lo digo porque lo he vivido y lo vivo.
She says that the persons who are suffering from this disease or carrying the mutation, they are heroes.
They are.
¿Puedo posar el vídeo? ¿Podrían poner el vídeo, por favor? We will.
Hello, my name is Alicia, and I carry the PSEN1 mutation. I learned about my genetic mutation by happenstance, as my mother had been misdiagnosed with Lewy bodies dementia. 5 years ago, I was in cooking school to become a holistic chef, and my final project was on what diet would be supportive to maintain brain health. During this research, I learned about ApoE4, which is an increased risk factor for late onset Alzheimer's disease, but not deterministic like PSEN1. Through the process, I learned that I carried this, as I had asked my primary care physician to get me tested, and instead they directed me towards a genetic counselor. There were little resources at the time of learning what I carried, and I had been following the research of Lewy body's dementia. So my mother began showing symptoms when I was 13 years old, and she passed away when I was 27. As a young person, my mom would forget to pick me up from school, and I would be left for hours waiting for her. This forgetfulness played a role in my development as a young person, as I didn't develop a strong foundational piece. As my mom was getting worse, my dad was focused on my mom, and both were preoccupied in the chaos of life. As my mother's mind increasingly began to change, her disorganization and confusion grew to the point where she would forget where she parked the car, couldn't find her way home, and would leave the stove on. While she was able to function for a while without too much help, her memory would glitch out, and she could easily lose her sense of direction and drive on the wrong side of the road, being a danger to everyone. For my father, he was challenged by all aspects of caregiving, as she would have hallucinations that he didn't even understand, strong emotional outbursts, a Parkinson's-like jerk that would make her fall, as well as being forgetful and vacant. I have 3 children, 18, 15, and 11 are their ages. My children never got to meet my mother or anyone else with Alzheimer's disease, yet they're all at risk for carrying the genetic mutation. While my eldest daughter is of an age where she could participate in drug trials, she has no lived experience with this disease to motivate her to engage in taking part in a drug trial. While she's aware that she has a 50% chance of carrying this gene mutation, it is not enough for her to engage in a drug trial itself. Additionally, dissecting the meaning of what being a gene mutation carrier is and what life could look like for her is a large burden to hold. Drug studies are also demanding and time-consuming for a young person who might be attending university and holding a job. Typically, young people don't have the flexibility to take time off from school or work to partake in research. The nuances of being in a drug research program is also mentally demanding for a young person who rightfully should be focusing their attention on growing into the person they want to be. By having access to drugs which have shown promise to prevent Alzheimer's would allow my children to pursue their dreams and live a full life. I partake in drug research to give the next generation the opportunity to live a fuller life with the capacities of a full mind. I will add here that if the drug trial went away, I would desperately plead for these drugs to be made available to me. Without these drugs, it would potentially allow me several more years to be with my loved ones. These extra years would be wonderful for me to experience, but even more so for my partner and especially for my children. When a child loses their parent to dementia, it is extremely complicated, especially if that young person is under the age of 25 years old, when it is said that their mind is not fully developed. The maturity of a young person progresses during these younger years, and their mother and father play a pivotal role in the person they become. The sense of knowing who they are in this world is reflected in their parents' eyes. But when a parent has dementia, that reflection can no longer exist. There would be nothing more I would want in my life than to reflect back to my children the beauty they possess in this world for as long as possible. These drugs would not just benefit me, but also for my children to fully express who they are and will be in this lifetime. I'd also like to comment on my brother's case as well before I close. My brother is 53 years old and has 2 young children. He wonders if he has started the decline, but does not want to pursue testing. he did not partake in the recent drug trials, as to dig deeper into familial Alzheimer's disease could mean was too much to emotionally process. Even though there is a drug trial he could have participated in, he is the primary breadwinner of the family, and it takes time to partake in drug infusions and baseline visit trips. He is unable to take time off of work for these studies, as he needs the income for his family. But again, by having access to drugs that would help this slow decline, it would allow him to work longer and provide for his family. Most importantly, to allow time with his loved ones. As someone who is dedicated to the DIAN-TU drug trial and looking forward to contributing to research, if there is a drug trial that has proven benefits for preventing Alzheimer's disease, I would wholeheartedly ask to be made available to anybody who is at risk of early onset Alzheimer's dementia. Thank you. Hello.
Well, after this, just say thank you so much for openness and for sharing your experience so honestly. What we've told us today really helps bring us closer to that what actually means to take part in a clinical trial, and not just from a medical perspective, but from a human one. And I think what stays with us is how personal each journey is and how important it is to listen to every perspective involved. And so thank you again for being here and for helping us to understand this much better. Muchas gracias por vuestra participación y por compartir con nosotros y vuestra experiencia con tanta sinceridad y naturalidad como lo habéis hecho, ¿no? Durante este panel. Lo que nos habéis contado nos ayuda a acercarnos mucho más a lo que realmente significa participar en un ensayo clínico, y no solo desde la parte más médica, sino desde una parte humana. Y creo que nos quedamos con algo muy importante, que cada experiencia es única, que es fundamental, ¿no? Escuchar las voces implicadas en todo esto. Así que muchas gracias por estar aquí para ayudar a entenderlo mejor.
Gracias, Amanda.
Gracias, Amanda. Gracias. Bueno, gracias. Al final sí que vamos a llorar.
Yo me estoy como Tamona.
No. Hi, guys.
Good morning, everyone. Can you hear me? No? Hi. Good morning, everyone. Welcome back. And welcome to this session about neuroimaging and emerging biomarkers. I'm Alexandre Bejanin from the São Paulo Memory Unit, and my co-chair is Bo Hans from the Washington University, St. Louis. So we have a very exciting program in front of us, and we will start with the first talk from Mariola Zapater-Fajari, that will speak to us about a distinct temporal pattern of tau PET and plasma biomarker in autosomal dominant Alzheimer's disease. She is from the Karolinska Institute. The floor is yours.
Hi, everyone, and thanks, first of all, the organizers for this amazing opportunity. Today, I'm going to talk about regional tau spread and plasma biomarkers in autosomal dominant Alzheimer's disease. As you might know, PET imaging allows us to track AD pathology with existing biomarker tracers for amyloid, tau, astroglia, and also glucose metabolism, among others. And here we can see how we can track AD pathology from pre-symptomatic to symptomatic stages of the disease with these different tracers. One of the main goals of AD research is to track AD pathology years before or like in early stages of the disease. And in this sense, autosomal dominant Alzheimer's disease allow us to track AD pathology even years before symptoms appear. Here we can see several papers of our group by using multiple imaging biomarkers and also plasma biomarkers in these autosomal dominant Alzheimer's disease individuals. Specifically, my colleagues found that by using amyloid PET, there were significant increases 17 years before expected symptom onset in this amyloid. At this time, dopamine levels, a marker of inflammation, were high and progressively decreased across time. And also they found at minus 10 years before expected symptom onset, significant decreases in FDG metabolism, and also at this time, significant increases in plasma GFAP. We went farther on and we saw differences in biomarker trajectories, specifically in FDG patterns among different mutations, APP and PSEN1, and also we saw different cognitive trajectories in these different mutations. Also, we saw that the relationship between FDG and plasma GFAP was only significant in APP mutation carriers, suggesting that early differences in underlying disease mechanism between these two mutations. Regarding tau PET imaging, there has been developed several tracers for track in vivo tau pathology in the brain with first and second generation tau PET tracers, and specifically in autosomal dominant Alzheimer's disease, by using fluotaucipir tau PET, they have found in the DIAN cohort significant increases in tau PET binding close to symptom onset. And also, they talked about yesterday, in the Colombian kindred cohort of preselinin-1 mutation carriers, they found significant increases in these tau PET levels 6 years before expected symptom onset. However, second-generation tau PET tracers like RO948 have shown subcortical tau PET binding in MCI stages, and here we can see in a paper that is now under review, and you can check the preprint that we found significant increases in tau PET binding in subcortical regions. Comparing to fluotaucipir as well, RO948 has shown less of target binding in critical areas of tau accumulation, early tau accumulation, making this tracer more suitable to track AD tau pathology early on the disease. With all of this, the main aim of our study was to characterize the temporal dynamics of tau PET and plasma biomarkers in a sample of autosomal dominant Alzheimer's disease individuals. To do so, we had 19 individuals belonging from families with known autosomal dominant Alzheimer's disease mutations, and we had 10 non-carriers and 9 carriers of different mutations, APP Swedish, APP Arctic, and also preselenium 1. And we had 6 presymptomatic mutation carriers and 3 symptomatic mutation carriers, spanning between -25 and 20 years across expected symptom onset. And then we measured the second generation tau PET tracer RO948. We also had measures of MRI, and we measured plasma biomarkers of tau, and also we had a huge neuropsychological testing battery. As we can see here, the groups were quite even in terms of age and educational level. However, symptomatic mutation carriers had a higher percentage of APOE4 carriership, and also we were all male individuals. And also, as expected, these symptomatic mutation carriers had lower levels of mini-mental. Regarding tau PET, first of all, we wanted to check the regional tau involvement frequency by meaning higher frequency earlier appearance of abnormal tau accumulation, and we did so by calculating how many individuals were abnormal in were abnormal 2 standard deviations above the mean of the non-carriers in all the regions of interest. And we saw a significant higher percentage of tau abnormality in subcortical regions like amygdala and hippocampus, followed by precuneus and posterior cingulate, suggesting that tau spread in autosomal dominant Alzheimer's disease does not follow the typical Braak staging suggested by sporadic Alzheimer's disease individuals. When we looked across expected years to symptom onset, and this is really interesting, we found that significant binding in amygdala around -8 years before expected symptom onset, whereas tau binding in the precuneus was found close to symptom onset. And also we saw that one individual that was presymptomatic but had past expected years to symptom onset started to had a high binding in amygdala, but not in the precuneus. We also saw a high variation in this tau uptake between symptomatic mutation carriers, suggesting heterogeneity in the disease. When we looked at the PET images again, we can see that this individual that was 8 years before expected symptom onset started to show high binding in amygdala and hippocampus, and then when we see at 4 years past expected symptom onset, we started to see binding in parietal cortex and lately in the temporal cortex. And again, this individual that had past expected years to symptom onset but was presymptomatic started to show binding in this amygdala and hippocampus areas. When we looked at the group level, we found a higher variation in amygdala region in presymptomatic mutation carriers that was not found in later regions like precuneus and occipital cortex. And also when we looked at the voxel-wise level comparing symptomatic mutation carriers against non-carriers, we show a higher binding in occipitoparietal regions in these symptomatic mutation carriers. When we compare this with a subsample that we had with early onset and late onset Alzheimer's disease sporadic individuals, we saw that the tau spread pattern in symptomatic mutation carriers was similar to the ones in early onset Alzheimer's disease and was more different with the late onset Alzheimer's disease. Again, these symptomatic mutation carriers had higher binding in parietal and occipital cortices compared to late onset Alzheimer's disease. Regarding plasma biomarkers, tau plasma biomarkers, we found significant increases close to symptom onset in all tau plasma biomarkers, and also this presymptomatic mutation carrier that had past AO started to have similar levels as these symptomatic mutation carriers. And also we saw lower variation in these plasma levels in the symptomatic mutation carriers. when we correlated tau PET against plasma p-tau217, we saw that the plasma p-tau217 correlated with precuneus but not amygdala, and also as tau burden increased, it seemed that plasma levels seemed to plateau at some point, suggesting that tau PET is more precise in tracking tau pathology at symptomatic stages of the disease. Regarding cognition, we saw significant decreases in episodic memory around 10 years before expected symptom onset, and this was corroborated with our longitudinal data. Regarding the correlation between the biomarkers and cognitive scores, we saw that neocortical tau PET, specifically areas like frontal, occipital, or parietal cortices, were correlated with all cognitive domains, whereas plasma p-tau217 only correlated with executive function, and amygdala, interestingly, did not correlate with any cognitive domain. So in summary, we can say that tau spatiotemporal spread is starting in limbic regions like amygdala, followed by neocortical regions like parietal or precuneus, and is similar to what we see in early onset Alzheimer's disease individuals. Also, the high variability in this tau PET against the lowest variability in plasma biomarkers suggested a ceiling effect in these plasma levels. And lately and interestingly, we found similar plasma p-tau217 levels in APP and also preselinin-1, even though APP Arctic mutation carriers do not have the typical amyloid plaques, thus suggesting that the p-tau217 reflects processes mainly related to soluble amyloid and protofibrils. Lately, I would like to thank all my colleagues and also, and especially my supervisor, Agneta Norberg, for all the support, and also the founders founding this project. Thank you so much.
So our next talk is concordance of biological and clinical staging in Down syndrome-associated Alzheimer's disease. It's going to be given by Jason Russell from the Epstein Family Alzheimer's Therapeutic Research Institute at USC.
Thank you, and thank you to the organizers for the opportunity to talk today. So yes, I'm going to be discussing the concordance of biological and clinical staging in Down syndrome-associated Alzheimer's disease. So, as I'm sure many of you are aware, the Alzheimer's Association released the revised criteria for diagnosis and staging of Alzheimer's disease approximately 2 years ago now, and this integrated the clinical and biological staging for Alzheimer's disease. And Down syndrome, individuals with Down syndrome are classified as stage 0 from birth, and this means that they are essentially genetically determined to develop Alzheimer's disease due to the presence of the amyloid precursor protein on the triplicated chromosome 21. There are 6 different clinical stages, starting with clinical stage 1, and this would be cognitively stable or cognitively normal. Clinical stage 2 is subjective cognitive decline, clinical stage 3, mild cognitive impairment, and clinical stages 4 to 6 are increasingly severe stages of dementia. It's important to note that in Down syndrome, subjective cognitive decline is not well defined. And so for the studies that I'm going to be showing you that we've performed, clinical stage 1 and 2 are collapsed together in our analyses. For the different biological staging, we start with biological stage A, which is amyloid positive, tau negative, and then progress through increasing tau burden through biological stages B, C, and D. So in this table, you can see the different grayed out boxes indicate essentially the concordant trajectory that we would expect an individual to progress through as they develop increasing pathology and therefore increasing clinical symptomatology. So this manuscript was published last year in Sporadic Alzheimer's Disease, looking at the concordance of clinical and biological staging. So this figure is laid out the same way as the table that I just showed you. So in the different rows, we have the different biological staging with amyloid positive, tau negative at the top, progressing through increasing tau burden as we move through the different rows. And then the different columns, the different clinical stages, starting with stage 1, cognitively normal on the left, and then dementia on the far right. And the different colors indicate the different concordant groupings. So the blue is biological in advance of clinical. The gray boxes are the concordant individuals. And then the red are the clinical in advance of biological. And what was observed in sporadic Alzheimer's disease is that individuals tend to show a relatively high level of discordance, with the majority of these individuals displaying a clinical stage in advance of biological stage. And this was hypothesized to be due to co-pathologies in this population, driving the clinical symptomatology beyond what we would expect based on just the amyloid and tau burden alone. So we sought to investigate this in individuals with Down syndrome, and we utilized 241 participants from the Alzheimer's Biomarker Consortium for Down syndrome. We selected participants' last visits that had complete amyloid, tau, consensus diagnosis, and modified queued recall test data. For the individuals with Down syndrome, we determined clinical stage in two ways. First, we determined this based on consensus diagnosis, and then we repeated the analysis using previously determined cutoffs on the total recall score from the modified queued recall test. In the table, you can see the different imaging processing that we used. So for amyloid, we used the standard Centiloid pipeline, defining amyloid positivity as greater than or equal to 18 centiloids. For tau, we assessed the standard uptake values in different BRAC-defined regions. So BRAC-defined region 1 and 2 would be mild tau, 3, 4 moderate tau, and 5, 6 severe tau. And we determined positivity as 2 standard deviations above the mean of the amyloid-negative cognitively stable group. And then for some of the later data that I'm going to show you, we used free surfer-derived hippocampal volumes and cortical thicknesses to look at some of the differences between the different concordant and discordant groups. So these are the demographics of our population, and you can see in the different columns we progress through increasing dementia stage, and then also in the cognitively stable we separate that out by amyloid negative and tau positive. And what we can see is we have the expected increase in age with increasing dementia diagnosis. There's no differences observed in sex, intellectual disability, or race or ApoE4 status across these groups. We see the expected increase in centiloid value, expected increases in tau SUVRs across the different BRAC-defined regions, and we observe decreases in the modified queued recall test total recall score as we see increasing dementia diagnosis. So these graphs just demonstrate the different cutoffs for the different tau SUVRs. And what we have here is the mild, moderate, and severe tau in the different rows, and then determining in the different columns, and then in the different rows we determine cognitive stability based on either consensus diagnosis on the top row and modified queued recall test total recall score cutoffs on the bottom row. And we can see that whichever method we use to determine the cutoffs for the tau SUVRs, we derive very similar values, which we can see with the sort of dotted blue line on these different graphs. So this is the concordance that we see between the biological and clinical staging when we use consensus diagnosis to determine clinical stage. And this is laid out in the same way as the previous manuscript and as the table. So rows are the biological stage, columns are the consensus diagnosis or clinical stage. And we see that individuals with Down syndrome display a higher concordance between biological and clinical stage at 63% than was observed in sporadic Alzheimer's disease in the original publication at 38%. And then the individuals that are discordant, interestingly, tend to display a biological stage that is in advance of their clinical stage, potentially indicating a form of resilience in this population. So we repeated the analysis using the modified queued recall test total recall score cutoffs that had previously been determined, which you can see here on the right. And what we observed was that concordance actually increased from 63 to 70% when we used modified queued recall test total recall score. So next, we wanted to look to see if premorbid intellectual disability differed by the different concordant and discordant groupings. And whether we looked based on consensus diagnosis or modified queued recall test-based clinical staging, we saw no difference in the distribution of premorbid intellectual disability between these groups. So I want to talk briefly now about the potential implications of this for clinical trials, and then some of the biology that is underlying the cognitively resilient group. So first, the implications for clinical trials. So this data suggests that Down syndrome-associated Alzheimer's disease represents a population that display increased concordance, and therefore cognitive decline is less impacted by concurrent non-Alzheimer's disease neurodegenerative conditions, suggesting a population that's ideally suited for AD pathology targeting clinical trials. This also suggests there's a more predictable progression in association between the biological and clinical staging. And so by having early initiation of treatment, we would expect that we would see more predictable effects of these early treatment initiation. And then secondly, as modified queued recall tests provided a higher concordance between biological and clinical stage than consensus diagnosis, we would suggest that modified queued recall test be used as, or could be considered as, an alternative for clinical trial inclusion criteria. And this additionally may allow the inclusion of participants where consensus diagnosis is unable to be determined. So next now, I just want to talk about the biology that underlies the cognitive resilience that's observed. So this analysis was performed on individuals that were discordant or resilient based on both their consensus diagnosis and their modified queued recall test total recall scores, so taking into account both of those different staging methods. So the resilient group that I'm presenting here would have a biological stage of 4, and then the consensus diagnosis would be cognitively stable, and they'd have a modified queued recall test total recall score greater than 28. And compared these individuals to the individuals that were concordant dementia, again, based on both the consensus diagnosis and modified queued recall test score. And what we observe is that the individuals that are resilient tend to display a slightly younger age, slightly lower centiloid values while still crossing that threshold for positivity. And same in the tau SUVRs across the different BRAC-defined regions, slightly lower values but still crossing the threshold for positivity. And then this group also displays larger hippocampal volumes. We observe no difference in sex between the groups, and we observed no difference in APOE4 status between the groups. We also looked at the different cortical thicknesses between these two groups and saw that the resilient group showed thicker cortical thickness in the left and right hemispheres. And then when we looked at some sort of more AD-specific regions, we saw that the resilient group displayed thicker cortices in the left and right inferior parietal regions, but observed no difference in the entorhinal cortices. So current work is ongoing to further understand the biology that underlies the resilient group. And we've been using machine learning approaches to model the brain age compared to the chronological age based on different AD-related pathology and cognitive symptomatology. So essentially, this is looking at sort of the whole brain amyloid across all the different regions, whole brain tau across all the different regions, and same for neurodegeneration. And using machine learning approaches to model the different ages that the individuals appear to be based on this biology and comparing that to their actual chronological age. And we're still working through that data now, and we'll be presenting that at AIC in a couple of months. And then secondly, we're looking to assess some of the genetic factors that may be underlying the cognitive resilience in this group that we've identified, and that is work that's currently ongoing. With that, I'd like to thank my mentor, Michael Raffi, and also our collaborators at ATRI and Washington University St. Louis, Bo. And then also thank the ADAD-DSAD study team, who obviously collected all the data that I've analyzed for this study, and their funding sources. And of course, a big thank you to all the participants and their families for taking part in these studies.
Thank you for this very nice talk. It is my pleasure now to introduce our next speaker, Zina Szlaetsky, from the USC Epstein Family Alzheimer Therapeutic Research Institute in California. And she will be talking about plasma p-tau217 correlates with amyloid and tau PET measure in people with Down syndrome.
Thank you so much for the kind introduction. I'm very pleased to be here today and talk about our data on plasma p-tau217 in adults with Down syndrome and correlation with amyloid and tau PET. I have nothing to disclose. So why are we focused on p-tau217? Why is it interesting for us? We know that it's one of the most robust plasma biomarkers, and it is correlating highly with Alzheimer's disease pathology, both with amyloid and with tau pathology in the brain. We heard a lot about it already. And there are at least 2 tests that have been successfully tested in people with Down syndrome, including the ALZPES-CMO technology-based and Lilly MSD-based technology that shown to distinguish A-beta positive participants from amyloid negative participants in people like people with Down syndrome. And we know also that different platforms for measuring p-tau217 show variable performance. So it's very important to actually have specific cutoffs for different platforms and test each single test separately in different populations, because in, for example, in dominant conditions and in Down syndrome populations, the cutoffs for the test may be very different. So, so today we will be focusing on 2 of these platforms that are really frequently used in sporadic AD population, general population, but have not been tested yet in Down syndrome to our knowledge. One of them is recently, or relatively recently, less than a year ago, FDA-approved test, which is a Lumipulse p-tau217 over A-beta 42 plasma ratio that shows high correlation with amyloid pathology as measured by PET or CSF. This test has been approved based on data on about 500 participants that had already data on CSF related to amyloid positivity in the brain or PET. And it has been replicated in several studies with 2 cutoffs showing better positive predictive value and negative predictive value as compared to a single cutoff for this measure. And also the ratio was taken primarily because it was better in distinguishing different, in distinguishing positive from negative individuals in different cohorts, but also because it allowed for classification of participants who showed intermediate results with a single cutoff. Another test that we were interested in how it performs, or the platform, how it performs in people with Down syndrome, is the C2N expressivity AD2 test, which is a laboratory-developed test. So it's also pretty widely used in the clinic, and it's clear, if the lab is CLIA certified, it can be also used for clinical care for helping in aiding diagnosis of Alzheimer's disease. However, also we don't know how applicable it is in Down syndrome. And in this figure on the left, you can see on the y-axis is APS2 score. This is a primary measure of that test. And APS2 score is derived from amyloid beta 42/40 ratio, p-tau217 and non-p-tau217. So it's a complex measure. With the cutoff that's seen by the blue line over there, that basically separates those which are amyloid positive with amyloid negative. However, we see the measures here are also for centiloids that are less than 25 or higher than 25, which is a typical sort of visual read related cutoff for positivity in the brain in sporadic AD. And we know that in Down syndrome, we prefer now to use the 18 centiloid cutoff. So that is also an additional difference what we see. And if you look at the specific analytes in this test, and we see that APS2 and % of p-tau217 perform pretty similarly over there. P-tau217 alone also performs really highly, but A-beta 42/40 ratio has slightly or quite significantly lower AUC as compared to the other composite measures and p-tau217 alone. So what are the challenges? We know that there are limited data on plasma biomarkers overall in DSAD, although like most studies are coming as we see. And also there, like how do they fit within the ATN framework is also like our aim probably, or in the end to discover how do we, in order to create successful clinical trials, we also need to understand at which stage this plasma biomarker raises, which stage they, or like when they go down. So when, for example, when amyloid starts to rise, right? Like where does p-tau fit in here, p-tau 217, or other biomarkers as well? How long does it remain elevated, where it stops? But also how well it correlates with our gold standard test. Also, like within the ATN biological stage, and we also see that there are like certain standards that we have to follow, and how these biomarkers follow the staging is another question. So the goals of this study was to explore the correlation of plasma biomarkers with amyloid and tau PET in people with Down syndrome. Compare the performance of two platforms, Fujirobio Lumipulse G assay-based assay and Precivity AD. And then the last one is to investigate accuracy of these plasma biomarkers in detecting amyloid PET positivity in people with Down syndrome. However, I need to say that this is a relatively small cohort, so this needs to be taken, all the results have to be taken with caution, or interpreted with caution. So we have 39 participants, 25 to 55 years old, who were non-demented, so they either were cognitively stable or potentially had MCI. From the NIH trial-ready cohort, TRACK-DS, that we already heard also about, and confirmed diagnosis of trisomy 21. And of course, the main also inclusion criteria in here was that they should have received amyloid PET. 21 of those participants had also received a tau PET, so it's a subgroup for the analysis. We measured only plasma p-tau 217 with Lumipulse G, so we don't have the ratio data for the assay, but I will present the p-tau 217 alone, which was actually performing great already. And the C2Ns, it's all the analytes that are present in APS2 score, were actually also included in here. So we did like a differential analysis, or like analysis for each single of them separately as well. And positive PET was considered if it was more than 18 centiloids for amyloid, and universal SUVR was used for tau PET correlation analysis. This is participants' demographics. We had about 20% of participants who were amyloid positive. Mean age was about 38 years with a range of 25 to 55. Female participants were about 40%, and we had about 30%, I believe, 25% of Hispanics, and about 30%, which is not actually that many, were ApoE4 carriers. So here, this slide shows the concordance of p-tau 217 measures and amyloid beta measures with amyloid PET. This is based on Sperman-Ranks correlation, and those participants that were amyloid PET positive are shown as orange dots, and those which are amyloid negative are blue. And we can see on the top panel, and also like actually the Lois curves shown also to address trend or to show the trend. In black. So on the top part of the slide, you can see those that correlated well. And this includes p-tau 217, both from Lumipulse and C2N, but also APS2 score, NP non-phospho-tau 217, and the ratio of p-tau 217 to non-p-tau. So all plasma measures that included p-tau 217, but also NP-tau 217 also showed quite good correlation. Whereas all the measures that were either amyloid beta 42, amyloid beta 40, or the ratio did not show any correlation at all. And looking at how well, or like how statistically significant that was between the groups, amyloid negative and amyloid positive, it's basically showing the same results, but showing the statistical significance that all measures, again, that included p-tau 217 were statistically significant, whereas amyloid beta measures were not. This is a ROC analysis with AUC evaluation for performance of those blood tests. And the higher, for those who are not familiar, the closer this curve is to the top left, the better the performance is. And the cutoffs were calculated, or the thresholds were calculated using the maximized student index, so that is the classical statistic method. However, there are also some drawbacks in using that one. We can see again that plasma p-tau to 17 and non-p-tau to 17 ratio, as in green over here, and p-tau to 17 alone show really great concordance there, so they are pretty similar in performance, and all of them perform pretty high, whereas a beta 42/40 ratio falls really below of the other metrics. This is to go a little bit into detail. The thresholds were calculated, as I mentioned, with a maximized student index. And we can see that accuracies are pretty high for both p-tau to 17 and C2N and Lumipulse assays, but also the p-tau to 17 composite measures from the C2N assay. Specificities and sensitivities were also pretty high, with sensitivities reaching there almost like there for the threshold that is advisable, like the currently advisable threshold is 0.9 for the test to be applied in the clinic. So we are close there. So I think more participants will be needed to understand how well it actually can be detected. But at this time, we are hopeful. And again, AB42/40 ratio was actually useless in this population at this time. Okay. And this is also for the other metrics, just to briefly show that there was no good correlation, or like those plasma measures, sorry, did not perform that well as compared to the p-tau to 17 measures. That includes non-phospho-tau and A-beta-42 and 40 separately. Again, these are just to go a little bit more in detail. For those that didn't perform well, the accuracy were like 0.90, 0.59 to ranging to 0.79. Sensitivities and specificities were not great for these measures as well. Then we looked at how well the plasma measures for just p-tau to 17, since we saw that they actually, p-tau to 17 alone performed really well in this cohort in both tests. So we thought, how well do they correlate? And we saw that there was a really high correlation in there, and that was also confirmed or consistent with a similar performance in detecting amyloid PET positivity. There was also no statistical significance between these two tests, AUCs. Plasma biomarker in regard to tau SUVR, this is really like exploratory analysis since we only had very few participants, but still we also could see that there was a pretty good correlation for p-tau to 17 in both tests, and p-tau to 17 ratio with APS2 also did really well. However, again, this I didn't show, but plasma non-p-tau to 17, AB42 and 40, they didn't do a great job there. And with that, I want to go to summary. Lumipulse and C2N plasma p-tau to 17 levels are associated with amyloid and tau burden as measured by PET. Plasma p-tau to 17 measure may be applied to determine amyloid status in people with Down syndrome. However, again, this is exploratory study, more data is needed, but this is a start. And Lumipulse and C2N's both assays did really well, and they have really high correlation and perform similarly in determining amyloid burden. I already said that, so the results should be interpreted with caution again. And I would like to thank again all the participants and their families foremost and first, and all the personnel that supports the clinical trials in general, especially with this population, since you need much more patients working with them. So it's great that we have this study, and it's great to have collaborations with C2N, with Joel Braunstein, and also with Matthew Zammit, who performed PET analysis. And all the people from USC with my mentor, also Dr. Rafi. Thank you so much.
Thank you, Zina. And our final talk is by Dr. Lita Bianchi from King's College in the UK. And she'll be talking about resting state fMRI in individuals with Down syndrome with and without Alzheimer's disease, a systematic review.
So, good morning, everybody. My name is Leda, and I am a PhD student based at King's. And thank you for inviting me to present some preliminary findings about my systematic review, talking about resting state ephemera in people with Down syndrome and without Alzheimer's disease. So I'm going to keep this short, as we all know. So as we all know, trisomy 21 triples the APP, causing the near universal risk of amyloid deposition by the mid-30s, and over 90% of our DS population will have neuropathology by 60. What is important for this presentation is that the DS brains are structurally and functionally atypical from birth. So when we see functional connectivity changes, we always need to ask whether they are from neurodevelopmental baseline or if they are for Alzheimer's disease development. Briefly, just for context, so when we are at rest, the brain remains active, and brain areas that coactivate together, so rise and fall together, they are functionally connected and form large-scale brain networks. And functional connectivity can reveal how those networks are organized. In the context of Alzheimer's disease, as we all know, the default mode network is the one that is most studied. Resting state is very suitable to our Down syndrome population because no task is required, so there is no performance confound, and it's visible across a full range of cognitive ability in Down syndrome. So the research question that the systematic review addressed are what are the resting state connectivity patterns in DS with and without Alzheimer's disease, and how do those patterns correlate with Alzheimer's disease biomarkers and cognition. Briefly on the method, so this is a Prospero-registered systematic review and is PRISM-aligned. I forgot to add the registration number, but I can give it if anybody's interested. We searched 3 databases from inception to December 2025, because the results were very heterogeneous, a meta-analysis was not possible. So the results, 13 studies met the inclusion criteria. Our pool sample was 300 people with Down syndrome and 283 controls. The age range varied from 7 years old to 56 years old. One study focused specifically on kids, young children and adolescents. 8 studies focused on young adults in the pre-amyloid window, so below the age of 35. 2 studies focused on the age of 35 and older, and 2 studies had a very wide range of inclusion with no differentiation of age. 2 studies reported also PET amyloid association, and 4 studies reported plasma biomarker association, but only 2 reported ApoE status and 2 related markers. 10 studies reported neuropsychological association. So moving on to the results, I divided them in pre-amyloid and amyloid window. So under the age of 35, we can see there is a frontal DMN coupling, which was preserved and enhanced within the DMN. And complexity was increased. So basically, the network are less specialized and less organized. This seemed to be a neurodevelopmental baseline, where there is a network disorganization before the AD disconnection that comes later on. Over the age of 35 years old, we see the anterior-posterior DMN dissociation, long-range connectivity between posterior cingulate cortex and prefrontal region, which were reduced, so the front and the back region, which are more metabolically expensive, they were compromised, while the short range, so the closer one, were spared. This simply mirrored the hallmark of the DMN disconnection that we see in the sporadic Alzheimer's disease. So it means that despite there is a different neurodevelopmental baseline, we do have a shared vulnerability with the sporadic AD. And then a few studies, I forgot to say that most of the study obviously is focused on the DMN for obvious reason, because it's the most studied for Alzheimer's disease, as it's implicated with memory. And so beyond the DMN, we can see a widespread increase in functional connectivity, where excessive between network coupling. So networks that are supposed to be working independently, they actually don't. And we have a ventral limbic and dorsal executive dissociation, meaning that the emotional network and the emotional network is overconnected, whereas we have the dorsal executive dissociation, which is the planning and working function that is less functioning. So this simply means that we have a functional connectivity disorganization that extends beyond the DMN. So it's not only about the DMN. Those studies beyond the DMN were mainly focused on young populations. So moving on to the functional connectivity biomarkers. So the PET amyloid positive studies found that a reduced long-range DMN connectivity, as expected. So the front and the back of the DMN was compromised. One study found that long-range connectivity seemed to predict transition to dementia. For plasma biomarker, only one study is really focused on this, and we found that functional connectivity was negatively associated with pro-inflammatory marker, but was actually positively correlated with t-tau. No association with amyloid beta was found, but this is because that study focused on very young population, so I guess it makes sense. And no association with the ApoE stages. The key limitation for this is that the samples were very small and there was no longitudinal observations. Moving on to the cognitive association, so we found that language and communication was correlated with language performance. One study found that ventral functional connectivity explained the 86% of variation in communication. Memory was also associated with higher ACC frontal connectivity, and it was associated with lower memory scores. The thalamus DMN connectivity was also correlated with IQ scores, and the whole brain connectivity patterns predicted IQ in youth. Again, this was a study only in young people. Greater network complexity was also associated with worse quality of life. And the key limitation for this was that despite 10 studies reported neuropsychological measure, there was not a single study that reported the same, so they all used different batteries, and it was very difficult to compare. This is just to summarize the findings. So at the top, we have our timeline from birth up to 60 years old, with the red line representing the amyloid positivity threshold. So the blue box represents the neurodevelopmental baseline, where we have a loss of network selectivity, excessive between network coupling, ventral limbic dorsal dissociation, limbic hyperconnectivity, and decrease in executive connectivity. We also have the increased DMN complexity and altered dynamics, but we have the preserved DMN coupling. Frontal DMN is intact or enhanced. And at the bottom, we will have what happens when the Alzheimer's disease comes. So we will see the anterior-posterior DMN disconnection that we see also in sporadic AD. The selective long-range connectivity is lost, but the short-range pairs are spared. And we see that functional connectivity tracked with amyloid and can predict decline. So the key messages for this is that DS show a neurodevelopmental baseline, which is characterized by this network disorganization and increase between network coupling and DMN complexity. The AD-related changes, they do mirror very much the sporadic AD. And the functional connectivity marker evidence is very limited because we only have 4 studies out of 14. And there is little focus on a population over the 35 years old. So the young population seem to be overstudied. Well, not overstudied. Conclusion and future direction. So what we would need is larger longitudinal cohorts, which span across the full lifetime, systematic evaluation of all the canonical large-scale brain networks, both for pre-amyloid and post-amyloid groups, because as we see, it's not all about the DMN. And ideally, we would like to do integration with plasma biomarkers, as we discussed, like p-tau and NFL, and harmonize neuropsychological assessment so that we can compare the data. Thank you.
Thank you very much for this very nice presentation. We are now open for discussion, if anyone has a question.
There's one up on the top. Is there something?
Oh, yeah.
Really excellent series of talks. Thank you. Treatments like donanemab clear the amyloid below the tau PET detection threshold, but they only reduce p-tau217 from 500% of normal down to 400% of normal. So it's a statistically significant decline, but it's still in the hyper-elevated range. How do those associate? I mean, as a practical question, you might continue these analyses at some point in patients who had been treated, but how does that result inform ongoing work and interpretation of the amyloid p-tau associations? Thank you.
I'll probably take that. I think that we need to consider that p-tau217 does not necessarily reflect the clearance of amyloid plaques in the brain, but rather the pathological processes that are still ongoing in the brain potentially. So they are still being formed potentially, the amyloid plaques. We still probably have some soluble amyloid in there, likely, that also results in p-tau217 elevation. So you cannot probably remove that fully. On the other hand, there may be also the delay in what we see in plasma as compared to what is happening in the brain. So that may be another contributing factor that I can imagine may also lead to that you don't see it right away, but maybe with time, as you clear the pathway probably, for amyloid to actually be cleared even post when you terminate the study. So maybe there is still a delay where you can still measure it a bit later and it goes even more down. But I agree that we are not targeting everything with anti-amyloid therapies, and maybe going earlier, using BACE inhibitors, for example. That may be another option to start the pathological, to address the pathomechanism at even earlier stage, potentially. So that might be helpful. In our study, it's basically a TRACK-DS trial-ready cohort Down syndrome, so they are not treated currently. We definitely will look at those measures in ALADDIN study that we are starting, and probably in all the other studies that we will have that address the pathological mechanism of the disease. I hope that answers.
It does. Thank you.
Mm-hmm.
Do you want to start?
Yeah, while the room is sinking in questions, maybe I can start and ask also Jason. I really appreciated your presentation. I found very interesting to see these concordant cases that are more important in the context of Down syndrome, to also see that the discordant cases tend to be more resilient, which I found surprising, and I was trying to give sense why in this population you may have more resilient case, especially because over the last day we have been speaking about copathology in this population, and even though some age-related copathology like advanced TDP-43 is not as present in this population as it can be in sporadic AD, there is CAA, there is sometimes alpha-synuclein, and sometimes also TDP-43. So I thought about the discordance between your results and those of the BioFinder cohort. And I was wondering whether some methodological aspect could also influence the distribution of the case. Among those, I thought in the different proportion of cases among the different stages. So I noticed that you did not have a lot of clinical stage, patient with MCI or patient with dementia. And I also assume that in a Down syndrome population, you are enriched in preclinical, by definition, you are enriched in preclinical Alzheimer's disease, which enrich you of people that have pathology but not yet symptoms. So I wanted to have your thought whether this can influence, and also what could be the influence of the intellectual disability on this result.
Yeah, thank you. Good questions. So I think if we just with regards to the distribution, you know, that was somewhat limited by what we have available there. But I think it's something we certainly want to continue to track as we get more data from ABCDS. We'll have more individuals that are progressing through the disease. So we'll be able to look to see as that progresses, how that starts to change. As we think about the different co-pathologies that, you know, we've spoken about yesterday particularly, I do think it's interesting that we saw really very, very few individuals that showed clinical symptomatology in advance of biological stage, given it's likely that there is some co-pathology in there. So it suggests that co-pathology is not driving at least the development of the clinical symptomatology up to clinical stage 4, right? So 4 to 6 is combined. So that's not to say that the co-pathology couldn't accelerate through stages 4 to 6. But it also suggests to me that the co-pathologies are likely downstream of amyloid. So you've got amyloid developing, and then with these pathological processes, you then start to see additional co-pathologies on top of that, whether that's due to changes in clearance mechanisms or something else that potentially cause this. I think that in itself leads to some sort of interesting questions as we start to think about treatments, you know. So we're seeing this really nice concordance as pathology develops. But if we're to remove amyloid, remove tau, how much will that affect the clinical symptomatology if we have co-pathologies? But then secondly, if we're to remove amyloid early, will we prevent the development of these co-pathologies? So I think that's some things that we need to think about more. And then just lastly about why are we seeing the resilient individuals? And I think that's a very interesting question that we're really trying to grapple with at the moment. And so one thing I've been looking at with the machine learning approaches is sort of, if you remember, there were very few individuals that were biological stage C and mild cognitive impairment, which would be as you would expect with the progression. So I'm trying to understand, is the progression what you would expect, sort of typically from top left to bottom right through the gray boxes, or is the typical progression more you see amyloid and tau accumulate and then clinical symptomatology in Down syndrome? So you end up with concordance at both ends, but the progression is more like this. And that's what we don't understand at the moment. So I'm looking to see if there's reasons for resilience specific in these individuals versus this is just the progression we see in Down syndrome because the pathology is so accelerated that, you know, you get the pathology and then pretty rapidly afterwards you get the clinical symptomatology. But in this snapshot, we're able to pick up people that have pathology but no symptomatology. And I don't have the answer to that right now, but hopefully soon.
And regarding the intellectual disability?
Yes.
How did you account this for the memory, for instance, measurement?
Yeah. So we looked to see if intellectual disability differed between the different concordant and discordant groups, and saw no effect of that. You know, may it affect how well MCRT is predicting the concordance? Potentially that might underlie some of the discordance, but again, I would expect that we would see their individuals to have worse symptomatology based on MCRT rather than better, and we were really seeing better clinical symptomatology. So it didn't seem to have at least an effect on the concordance in this study.
Okay, I have a question. Very nice job, Mariola. That was a very nice presentation. One of my things was we've been talking, and we saw in the sessions here and previous sessions that p-tau217 is actually a marker of both amyloid and tau. And so I was interested to know what was the amyloid and what was that relationship with some of the tau changes and the tau spread? And did you also look at other markers? I know it's in a small group. There are other tau measurements like MTBR that are more reflective of the tau PET in doing that. So any thoughts about that, especially since we see in these subcortical areas a large amount of amyloid that's also depositing, often early on in the disease?
Really interesting question. We do not have MTBR tau, and I would love to have it because I think it's more closely to tau PET than it is like p-tau 217. And we did not measure amyloid, so I would also like to know how it's working with plasma p-tau 217. So it's a really good question, actually. And we saw this. What I want to point is that we had this APPR tick that they might not be following the typical progression, and we have to take this into account. So maybe we are seeing these plasma p-tau levels because of this typical sample, and with other type of mutations, we would see another trajectories. But what we see is this tau accumulation in limbic regions, and I think it is important to take that into account.
Do they have any clinical symptomatology? Because we see heterogeneity in many of these cases, and we've talked about mutations and where there's heterogeneity in that. There can be also differences, and you did a very nice job of explaining the spread of the tau. Are there differences that then translates into clinical symptomatology?
We did show this decline in episodic memory that starts 10 years before expected symptom onset, and I think that also a nice thing that tau can tell us is that different patterns of tau accumulation will tell us different patterns of cognitive decline. So I think this is something that we have to take into account and that plasma does not tell us. And we saw this correlation between plasma p-tau and executive function that I think that is an early symptom. Executive function is a really early symptom of the disease, together with episodic memory, of course, but then maybe it's later on. So,
maybe to jump on the involvement of the medial temporal lobe, I saw that you were insisting on the amygdala results. And I wanted to have your thought about this topography and whether in other cohort, like Diane, whether it is a result that has been consistently reported, and whether there can be sometimes some results that are specific to one specific tracer, no? For instance, we had the case of the floor-to-CPR with the off-target binding from the choroid plexus, which limited the ability to see the hippocampus. So, is this amygdala, in your opinion, something like we should look deeper at, or?
I do think so, and actually in this paper that we have written and is now under review, we also saw that in the early onset Alzheimer's disease, subcortical regions start to accumulate early on, and then it follows more like typical Bragg staging, like entorhinal, and then middle temporal, and also spreading through parietal and frontal cortices. So I do think that the amygdala is an important region to look at, and I don't know if other tracers could corroborate these results, because as you said, flow-tau CPR is not able to capture this because of the off-target binding. So I would also love to see if other tracers can corroborate what we see with RO948. But I think we have to start to look into subcortical regions more closely if we can, because they are important. And also some other studies point out locus coeruleus as a really early tau accumulation region. So I would also like to look at locus coeruleus, but we didn't have the resolution in the MRI to look at, so we couldn't. But it would be a really nice also regions to take into account. Thank you.
Okay, I'll go to Lita. I give you a free degree, so you're going to have to tell your advisors ahead of time. Very interesting results. One of the things that we've noticed when we're scanning any kinds of individuals, but especially those with Down syndrome, is there's a lot of movement that occurs, especially, and that can really affect your resting state collections. Yeah. And so, And then also it's the time of the collection, so how much resting state you have, and that really helps you in understanding that. So in the studies that you did in looking at the review, how did they account for movement? How do they account for the duration of the scan? And then the second question is, do you think those increases that are occurring, is that some kind, early on, is that some kind of compensatory mechanism that's then failing or not? So love to hear your thoughts on both of those.
So on the motion, it was like a very common issue. It's a common issue also on our sample that we are doing fMRIs as well. I think the pre-processing, so when the sensor for motion is quite lenient, because otherwise you risk cutting out half of the sample. And the samples included in the systematic review, they were actually very small samples. They range from, I think, on the top of my head, from 10 participants to 22 maximum. So it's quite a small sample. And what was the second question? Sorry.
The compensation. Is there early changes, some kind of compensatory mechanism or something?
I think some of them must be like some compensatory mechanisms. For example, when we saw like in the tau correlation, the plasma tau correlation, it was on a younger sample. It was on a sample that had like a average age of 27 years old. So I think it's a compensatory mechanism rather than AD-related tau insurgence, or whatever it is.
Right.
Yeah.
I can add a question about the resting state. I found very fascinating to see all this study, but it's still a literature that is very at the beginning, and there is still a lot that we need to learn from it. I was curious about like how to interpret difference between the Down syndrome population and upload controls, and thinking especially at what could be their rest activity, because we're often referring to the resting state fMRI as like an activity without thinking, but actually subjects are protecting themselves in the future. There's a lot of brain activity during this 10 minutes of the scanning. And I wanted to know if all your reading, you noticed some group did some post resting state fMRI interview to know about the activity of the subject under the scanner?
Yeah, that's a very interesting topic, and it's a question I've been asking myself a lot. And I also read a paper that said that before taking your MRI scan, the resting state, it's very important what you're doing. So for example, there was one study where they were watching movies during the resting state. So that wouldn't be a true resting state, right? Yeah. Because the brain is looking at that and is having emotions. But it's like we don't know exactly what's going on in the brain while it's at rest. So I think there are some new techniques that they use, like dynamic analysis method that kind of helps mitigate for that or something like that.
Autism part, yes, maybe. In other case, we can also do a post-scanning interview, but it's true that maybe the recollection of the activity during the scan in the Down syndrome population may be challenging, especially if you're thinking in advanced stage.
Yeah.
And based on this exhaustive reading you did, what do you think are the next steps for us as a field to take?
It would be nice to have larger sample sizes and maybe correlation, so multimodal association with plasma markers and other, either plasma or PET amyloids or something like that, because it's alone like this, it's a bit difficult to interpret. And also maybe because functional MRI is measured with many different methods, so sometimes it's difficult to understand the results and apply them to another study. So maybe something more standardized and longitudinal measurement to see like changes so from the young to the older populations.
Okay. We can further discuss, but in Dabney we have been collecting resting states since many years. So we have some data for it.
Sina, let me ask you. So we had some questions about, and we talked about a little bit of these new plasma biomarkers in a number of sessions. One of my questions was in the sample that you had, was the C2N or the LumiPulse how was the temporal relationship between the centiloid value? So did amyloid change before the plasma markers or at the same time? Because when we've used the Lilly assay, we've actually seen it's about 3 or 4 years after the changes in this. I know you have a small sample,
exactly.
But I didn't know if you could see that.
No, we didn't get those analyses because we only had, like, if you notice, we only had 8 amyloid positive participants.
Yeah, like 3 or 4, yeah.
So that's too little, we thought, to do that.
Okay. And then the second question was, with regards to some of the changes that you showed, I didn't see, but maybe I wasn't, there wasn't really that much of a difference between the LumiPulse versus the C2N, either for their sensitivity, specificity, or the AUC. So where do you think we should be going with these as markers for the field for us?
So for LumiPulse, actually, AUC was even slightly higher than for C2N, but then other sensitivity metrics, I think, or that was a specificity metric. I think that was slightly higher in C2N. So I think it's difficult to tell. I think we really need more participants, and we need more plasma samples that have also amyloid PET to actually also establish the threshold better. We also need other measures to actually going into front or like forward, like something like positive predictive value and negative predictive value will be more important in terms of what we apply in clinical trials. So yeah, to answer your question, we just need more participants. I think that's good. I don't think that we can tell now whether C2N or LumiPulse is better. I think that we also didn't measure other analytes with LumiPulse, so we just focused on p-tau 217.
You didn't do the quad. You didn't do like a quad or anything else. Right? Okay.
So therefore, I think like we just need more data first before we can answer that. But I feel like in our hands, they performed really similar, and I would just exchange one for another right away.
Do we have other questions here? I think Mike has a question. Or...
I just want to add something.
Good. Please.
I just wanted to add that the C2N and the LumiPulse are the only two FDA approved. And there are families that, even though there isn't a treatment available right now, want to know. They want to rule out Alzheimer's as a cause of symptoms. We just had a poster at this meeting on p-tau 217 in DS regression disorder. And a lot of families bring in their loved one for changes in cognition and functioning in, between the ages of 20 and 40. And some physicians are saying, oh, it's Alzheimer's.
Right.
And it's not. And it may be something treatable. And so for that reason, both C2N and LumiPulse, Fujiribio, are interested in showing the utility of those two in this those two assays in this population. So we wanted to get that out quickly, particularly for the clinical trials as cut points, and see how far apart they would be for eligibility. So we wish we had a larger sample size, but there's an urgency right now in the field to get these out for both clinical use and for clinical trials.
No, I just want to add, maybe Lucia Pertierra is here. Okay. She's got a poster on LumiPulse because we had published the results with ALZPAD, but she has a poster. We ran all the cohort with LumiPulse, and I don't know if Lucia, I'm calling for you, if you want to comment something.
Thank you for a presentation. It was very interesting to see that we are working in a similar topic. And yes, as Juan said, we analyzed our cohort in a cross-sectional study with LumiPulse for tau 217, and we also found very good areas under the curve. But it's true that when it comes to the clinical application of it, and you need to think about a post-test diagnostic accuracy and so on, you need to take into account also prevalence, and for positive predictive values and negative predictive values, you need to take this into account, and that at that point, perhaps at the age of regression disorder and so on, the test accuracy is not as high, or well, we need to take into account all those things in the context of population based and in a posteriori results. So the areas under the curve are really promising, as you showed, and we found almost the same cutoff. It was, for amyloid, it was 0.26, I think, and you showed 0.21.
Correct.
So it's really similar and encouraging to see that everything looks the same.
It's
so,
great to
well, I invite you to the poster if you want to talk a little bit further about this.
So on that point, have people been doing repeated sampling to look at if there's any differences in the stability of either the Lumipulse or the C2N in this population? Do we have to worry about that? And, as Mike, you're bringing up here, I think at the group level, we're kind of do this. Do we think we're at the individual level? I mean, yeah, I do think we're getting closer to it. The question is, and I have that in my clinic all the time, of somebody's coming in with DSRD, and it's DSRD, not AD, but they want to have rule-outs. So are we at that level, or where we're thinking of that? Okay. He said we're running those assays now for those who were upstairs who couldn't hear. Other questions you have? I can ask another.
Oh,
yeah.
Sorry.
Yeah, please go ahead. Oh, please, Dr. Kuhn.
So I just have a comment to make about the functional connectivity as the author of a few of those papers. I really think that a really good thing to do, take this idea from me, please, is to look at neurovascular coupling in people with Downs, because we know that affects functional MRI and definitely affects connectivity, and we don't know that neurovascular coupling is the same and in people with Downs as it is in the typical population. I think, usually you would do MRI EEG, which would be really hard, and part of the reason I've never done it. But you could also do something like an auditory oddball task in the scanner and then EEG out of the scanner. Do it that way. But yeah, I don't know that neurovascular coupling is the same, and it's always kind of bothered me, even though I've also compared people with Downs to controls. It's always really bothered me. Like, are we really measuring the same thing? So it's just a comment. Yeah. Take that idea, run with it, someone.
I had one more question to Jason. Beautiful presentation and watching the concordance or not. When we're in the clinical setting, one of the hardest things, especially in ABCDS, is differentiating MCI versus dementia. We're always sitting there, and in my hands, it's maybe MCI, and then, or I'm waiting, and then it's actually dementia, and it was probably MCI. And so this differentiation, and it's always very hard to separate. Do you have any ways, or do you think we can start to separate that out a little bit more, or using your machine learning algorithms of doing that? Or is it really even important? Is this just, if they're really symptomatic, that's more important?
Right. So I think, if I'm remembering my data correctly, using MCRT did produce slightly more individuals that were in the MCI group.
Right.
So sort of using those cutoffs may enrich that group slightly. And it may be that some of the discordance we were seeing were that individuals had mild MCI, but were being scored as cognitively stable, and then would come to dementia. And some of these things that we're seeing may be due to limitations of seeing early cognitive change in Down syndrome. In terms of the different machine learning approaches that I'm using, I haven't specifically thought about whether the sort of cognitive aging machine learning approach that I'm using, whether that could determine MCI. I think the problem is if I don't have a gold standard, this person has MCI, it's very difficult to train the model to identify that. But I may be able to start looking at sort of using the MCRT and just looking at the age profile to see if there is a group that where the concordance fits better between the sort of cognitive age and the biological stage that we would expect. And if we're missing people in there that do have MCI that we're just not identifying as having MCI based on either MCRT alone or consensus diagnosis alone.
And just going on that is one other, so you've used the MCRT and we like that and it seems to be a very good test. If you either added on DSMSE or you added on other measures, do you get any more bang for your buck, or is it really MCRT does enough? And if I just got a simple, that kind of test, especially if, if companies are thinking about, do I do a composite or do I use this as a one single measure?
Yeah, so I mean, based on this data, it seemed that MCRT was doing really pretty well. I think with the machine learning approaches, they use everything that we have in ABCDS, so they take every subscore that we have. And as we look at that further, hopefully I'll be able to answer that in more detail, and I'll be able to potentially pull out some of the different weightings, what is really contributing to the cognitive age, and then maybe be able to answer that in a little more detail.
Thank you. We have one more question.
Yeah, so I had a question for Lita. In looking at the studies that you're looking at with the default mode network, there's also been work showing that those with autism also have differences in their connection in the default mode network. And so I was wondering if any of the studies that you looked at specifically looked at those with Down syndrome that have a dual diagnosis of autism, to see if there are any unique differences that also exist in that population. And also given that there's been some studies showing that more broadly those with intellectual disabilities may be at more risk for developing Alzheimer's as well. So just kind of thinking within the Down syndrome population for those who have a dual diagnosis, if there are some, potentially some interesting things within the default mode network that may be able to be picked up.
So I'll be honest, I don't know much about autism, but in my sample, in the pooled sample, there was a tiny value at a dual diagnosis. I remember there was one study, which is actually the one that was excluded because it wasn't a true resting state. They were looking at autism as well, but I'm not sure if it was Down syndrome and autism together. It just didn't meet the inclusion criteria. Sorry, didn't hear. Sorry, in my sample, in none of the studies, there were a dual diagnosis of Down syndrome and autism. There was one study, as I said, that was not a true resting state, that was looking also at autism. But I don't know if it was DS and ASD or just DS and then another sample with ASD. So I'm sorry. Was that the question, right?
Yeah.
Yeah. But that could be something to look into. Yeah.
Do we close it? Yeah.
Okay, we will stop here. Thank you everyone for your attendance.
And thank our
and
presenters
thank you to
again. Lunch is downstairs, correct? Yes, lunch is downstairs again. Thank you.
Thank you to you.
What your Fantastic.
Easy.
Easy.
I'm looking for the organizers to tell me, yes, I can. I just took the liberty. Maria, can I get started? Okay, I got approval. Someone's going to tell me what to do. I'll default to Maria. So, hello everybody, and welcome to this session. We are all extremely excited to have representation from Global South at this meeting so we can foster collaborations. I'm Sid O'Brien. I'm one of the moderators, and thank you for being here.
And I'm Natalia Vilor Tejedor, the second moderator from Barcelona Beta and Utrecht University. A pleasure to be here and to have the opportunity to moderate this session on this important topic, and to have this amazing group of speakers that will present and introduce very nice things that they are doing now in LATAM and in other places. So let's start with our first speaker, Agustín Ibáñez from the Global Brain Health Institute and Trinity College Dublin, that will present Down syndrome and Alzheimer's disease research topics.
Thank you so much, Sid, Nati. It's a great pleasure to be here. And I will talk about brain clocks and how we can use it to capture different burdens of different things. Those are our disclosures, especially thanks for the organizers, the Alzheimer's Association, that have been with the Red Lab Consortium for almost 10 years. So I will introduce the idea of brain clocks, what they can do, how they are important, then research on Down syndrome ID and autosomal ID, and how we have combined this to try to understand how the genetic risk impact in accelerated aging, association with transcriptomics, and association with multimodal phenotypes. And then I will try to bring some ideas for Latin America research on Down syndrome that we are very excited about that. So one simple way to measure accelerated aging is doing brain clocks. As you know, aging clocks measure the biological aging from organs or, systemic age or brain age. What you do is you train simple machine learning or a fancy deep learning model with data from neuroimaging in this case. You estimate the age, and then you have a gap between the chronological and the biological age. That means that there is people that looks younger or older. And then you can investigate what drives this difference, right? So we have been pushing this idea of neurosyndemics, that the environment, the body, really, really is important to understand the pace of aging. And we have been shown that geography, exposome, income, lifestyle modulate the brain clocks, right? So it's something that I will come back later on. But now what's happening with the internal genetical drivers of aging? Are they also modulating the aging clocks? And this is a wonderful idea that comes from Imer and Juan, and also a great computational neuroscientist from Venezuela, Carlos Coronel. So there are almost nothing with brain clocks in Down syndrome, just a few studies, very small samples, not well methodological designed. There is no comparison between genetics version of AD, classical like presenilin-1 mutation versus Down syndrome. They are not association with the deep phenotyping from plasma to centiloids or cognition. And moreover, there is no understanding of what are the underlying mechanisms or the biophysical mechanisms associated with these brain clocks. So this is what we wanted to do. And we have been working in computational models for both EEG, EMG, as well as fMRI to create not only robust models that can estimate the biological age, but also creating semi-empirical modeling to understand which mechanisms like excitatory-inhibitory balance or other neuromodulation effects, how can be associated with this estimation of the biological aging. So this is our design. We have a very large data set of 9,000 individuals because if you have a lot of subjects, you can create a normative model of your clocks, and that makes your model more robust for estimate the individual pace. And then comparing with the presenilin-1 mutation carriers and non-carriers from the family, from David Aguillon group in Colombia, as well as a large data set, 700 of Down syndrome. Thanks for Imar and Juan for bringing all these consortia together. including all the stage from preclinical to fully dementia symptoms. And then we connected with this deep phenotyping. So the first thing, we were able to estimate the aging as should be done. And we have this very nice gradient across different domains of imaging. So controls are followed by Alzheimer's. Sorry, for controls that followed by non-mutation carriers versus early asymptomatic Down, prodromal, and Down syndrome with dementia. So that means that they are an increased accelerated aging in the gradient that is expected, almost 20 years of accelerated aging in the Down syndrome with dementia. We have a very nice association with the BACS, the brain age gaps. We were able to find associations with cognitions, with centiloids, with p-tau217, and with NFL in Down syndrome that really reinforce this deep phenotyping, the role of the BACS to characterize these patients. We also, in each different domain, like EEG, fMRI, or EMG, we found the same effects with the data what we have, because it's not the data for all the conditions. Then another thing an easy criticism that we got from a reviewer, it's, okay, you are training on normative models, thousands of subjects, they are completely different of the brain of Down syndrome. So maybe it's just the deviation, right? So we trained the models with the brains of the Down syndrome and then tested all the others, and we have exactly the same effects. And then also we control it by the age bias. It's a very well-known effect in the clocks. So the people that is older have larger gaps. But the people that is Down syndrome in general are younger than, so even in that case, and even controlling the effect of age bias, we have exactly the same effects. They were replicated with the controlling for the age bias, all the results. And then with this biophysical model, we use a neural mass model and dynamical field model for EEG or EMG, as well as fMRI, to try to, build a model, then mapping the empirical functional connectivity data and understanding the underlying mechanisms. We explored the inhibitory and excitatory balance that is very well known that affects dementia. And what we found is there is an increased hypoexcitability that is very well known in dementia, but also more accentuated in Down syndrome. And impressively in asymptomatic carriers, there is the opposite effect. It's a hyperexcitability. And this has been very nicely shown in animal models. It's the first effect demonstrated across Down and presenilin mutations. And then we use transcriptomics to explore these areas of excitatory, inhibitory activity in the brain with the expression of different genes associated to dementia. So first of all, we found for presenilin asymptomatic carriers the expected tau and amyloid genes, of course. Then in sporadic Alzheimer, we have the expected modulation for more, large neurodegeneration, if you want. And in Down syndrome, we have the larger effects, not only, amyloid and tau, and lipid, but also neuroinflammation and immune response. That is, I think, is a very important way. We just use the last point, perturbation, in which we, in silico, understand by stimulating, using, cholinergic stimulation in global excitatory approach or focal stimulation. We have very nice results. This is crazy. I don't want you to look at this complex thing, but in a single word, what we found is two things. So the global excitatory cholinergic system trying to help, recovery or reduction in accelerated aging in both Down syndrome and Alzheimer's. And only focal inhibitory stimulation was useful in the carriers. That is consistent with the hyper-excitatory effect observed on that. The model also predicted the future, that this is important. I think this is very nice, and it was, again, a suggestion for the clinicians having really good ideas. So the estimated years of onset in presenilin mutations is happened in the BACS 10 years before. That is very nice and has been reported elsewhere. But in Down syndrome, it was almost 30 years old that we already identified accelerated aging in the participants. So I think that is very important. So the BACS showing a gradient of accelerated aging across the different neurodegenerative conditions. By far larger effects were observed in Down and in Down with dementia. higher accelerated aging was associated to lower cognition, higher amyloid burden, and high plasma. Biophysical models showing this shift from hyperexcitability to hypoexcitability, as well as potential therapeutic targets. So I will skip the other thing because I want to bring something new. So we have been using brain clocks to show quite the opposite effects, the effects of environment, from pollution to socio-political instability, right? So I think there is a huge opportunity for testing this Down syndrome in more diverse populations, where cumulative burden, cardiometabolic effects, even socioeconomic impacts or ancestry impacts can modulate the clocks. This is why I think that we need to create a consortium in Latin America for Down syndrome, and of course Africa and hopefully Asia. But also to understand resilience. We can identify, if accelerated aging is a hallmark of the patient that doesn't develop dementia. And also we are now creating a baseline 3 million simulation of individual biophysical models that will allow us to test biophysical twins. Like, in each participant, create a simulated subject and you can test different pharmacological modulation. This is a data set from Red Lab. It's going to be open when we get approval, and we hope that we can do something similar in Down syndrome. I think that in Latin America, from FLENI to Colombia, to Chile and Mexico, they are groups that are interested in doing the Down syndrome, and we hope we can contribute and work together for that. Special thanks for the invitation and for the Alzheimer's Association for this wonderful conference. My first time here. Thank you. Thank you.
Thank you. Wonderful talk. Our next speaker is Carolina Ochoa from the Brain Institute, Universidad Alfaro y Santiago, Chile. I can't even read from here. I need my glasses.
Well, I can know.
Okay, thank you very much. So I think 10 minutes, it's little time to tell you all I want to tell you about. So I'm going to go through a quick journey of what we do in BrainLab, specifically in genetics, and we can discuss further, right, if you want to know more details or other projects that we are conducting. So first, for the ones who have no previous knowledge about the Red Lab study, very short. So it's a case control study, multi-partner. That means that we have 13, 16 sites across 7 countries in Latin America. And the participants are about 4,000 participants. And those participants have been very comprehensively studied, evaluated. And we have I think I can go here. We have biomarkers data, so ATN proteins. We have a very extensive evaluation with neurocognitive assessments, different types of neuroimaging, genetics and epigenetics data, and also a very comprehensive assessment of social determinants of health and adverse early life experiences. Okay, that was very short. And then I'm going to introduce you to the topic that is important today. So you can see in this very, very nice paper that has been recently published by my colleagues from Red Lab, specifically in the genetics group. And in this paper, you can find the genetic characterization of the cohort and also some results regarding, for example, ancestry, which is a topic that I'm passionate about. So ancestry across some of the countries within Red Lab, we obtained the global ancestry through comparison with 1,000 genomes. And we can see here the admixture of our population. It's mainly, you can see here, mainly European ancestry, but also a big proportion of Amerindian ancestry. And in some countries, you can see here in violet color that there is an important proportion of African ancestry and some South Asian ancestries. So this is a very strong point of our cohort, and we really want to leverage that in our studies. in that paper, you can also find some of the first results regarding genetics, and you will see here the family studies, that it's also a very important point in red blood, and there are certain families with carriers, right, to study Mendelian inheritance. And interestingly, also in the near future, these families will be also followed on in time. Also, there are some results on the pathogenic variants analysis, where we could find already know, but also novel rare variants. And interestingly, some of these variants identified in some families. When we performed a local ancestry analysis, you can say that we obtained like maybe expected European specific variants, but also, for example, you can see here some Amerindian specific variants, which is very interesting because we are there looking into very specific local ancestry, not only global ancestry. Well, this is some work that I'm doing with my postdoc, Danilo Carmona, and then we performed like a standard GWAS, right, that you would, all of the people like working on genetics would do a GWAS, right? So we did a GWAS on Alzheimer's disease diagnosed versus controls, and we had like the strongest signals in the chromosome 19, where we could replicate previously reported genes associated with Alzheimer's disease. We can also here see some incipient signals, which of course we are expecting very soon to increase our sample size, at least double the participants. So we hope that we could also replicate the signals. But I was just saying that our population is very admixed, and that's a very particular characteristic of our cohort. So what we did next, we were interested then in studying this local ancestry of those variants. So you can see here in the first plot that that's the standard GWAS, right? But then we used this tool that it's called Tractor, that maybe you are familiar with. And what we do is to leverage the fact that all of us who are admixed, we have different tracts with it. We have like pieces in our DNA that come from different ancestries. So our DNA looks like a mosaic, like here. So we leverage that, and we can study not only person, let's say by person, but also the specific different ancestries within that person. And then we repeated our GWAS, but now it's an ancestry-informed GWAS. And we obtained, well, with the European ancestry, similar results, also for the Amerindian ancestry, but we could see some differences here that we want to look deeper into, because we obtained, for example, some variants with different directions from the Europeans to the Amerindians, so we need to look further into that. And for African ancestry, we see, for example, that we lose the wide significance of the signal. But this could mean that maybe for that locus, that ancestry is not relevant to AD, but could also be a power issue, right? So we also plan to improve those analyses in the future, very soon. We have also explored some genetics characteristics of ATM biomarkers, and I will start with the N. So we conducted in our preliminary results a GWAS in 40 brain regions. This is a standard GWAS. This is not an ancestry-informed GWAS. And interestingly, these results, all of our participants with available data so far were admixed Americans. So here we have no European or African participants. And we had, these are the 4 most relevant regions, and we did find significant variants in genes associated with neurodegeneration, neural plasticity, and synaptic signaling. This is also a work in progress, but if you want to know more about it, if you're going to the AAIC this year, you will see a poster that you can come and we can discuss this. Also, we explored some genetics analysis in relation to plasma biomarkers and ApoE4 dosage. What you can see here is the different proteins, and we did an analysis stratifying by ApoE4 dosage. So you can see these are crude comparisons, but you can see here clear differences among the different levels of proteins when you have 0 allele, 1 allele, or 2 alleles. And our results here are also consistent with the current evidence. And we also did that here. This subset of data is only for Amerindian ancestry. Then, continuing with the plasma biomarkers, Red Dot also has DNA methylation data. So we also leveraged that data, and we got some interesting findings. So here you can see the Manhattan plots of the EWAS analysis for p-tau and p-tau 217 and 181. And we found distinctive CpGs differentially methylated, and in an enrichment analysis for the p-tau associated signals, we found that they are annotated to genes related to cell death regulation, for example, apoptosis, neurodegenerative stress-specific pathways. While if we take a look at the results with beta amyloid biomarkers, we also had differentially methylated CpGs in our sample, and the enrichment analysis showed us more general processes, metabolic processes, oxidative stress, suggesting a more systematic aging-related signature. So this is very interesting because this could be in line with the beta amyloid cascade hypothesis. And that's my presentation. I wanted to make it short. Thank you very much to my colleagues in BrainLab, all the PIs participants in the Red Dot cohort, and also to our funders, the Alzheimer's Association, Bright Focus, etc., and our also partners from the GNA. Thank you very much.
Wonderful presentation, Carolina. Thank you so much. Our third speaker is Lucia Crivelli from Fleni, Argentina, and she will introduce some insights on Latin fingers.
Okay. Hello, everybody. Thank you. Thank you, the organizers for this conference, for inviting me for the first time. This is my first time in this wonderful conference. I will try to bring some insights about this very important Latin American initiative, trying to bring some knowledge about capacity building and how clinical trials, when they are leveraging the different local possibilities, can make a difference and can then be receptive to other clinical trials. So as we know, modified risk factors for dementia are very high. Worldwide, they account for 45% of the cases of dementia, but this number is even higher in low and middle income countries. But it's interesting to see how this number is different in the different countries in low and middle income regions. So this is a paper by Regina Paradela from Brazil, where she compares the potentially modifiable risk factors in different countries. And you can see that we have lower and higher numbers, but that there are common factors that are very important in a region, basically obesity, depression, and physical inactivity. But when you look within the region and you compare countries, and here I'm comparing the country with the lowest risk factors, which is Peru, and the highest risk factor, which is Chile, you can see the configuration of these risk factors also differ. So for example, in the country where you have the highest risk factors, hypertension is very high, but smoking is very low. And the opposite happens in Peru, where you have very high smoking risk. So this brings us attention that we should look into the region, and within the region, we should be respectful of the different local vulnerabilities. Also, when you look within one country, and this is a study by the group led by Claudia Suemoto in Brazil, you can see that within one country, the different regions have a different configuration and a different total number of modifiable risk factors. So here they compared different ethnicities and did not find differences, but they did find differences when they compared rich and poor regions within the Brazilian country. And the same happened in this study within Chile, where they found that the differences were not in the different regions, but between genders. So women in Chile have higher risk factors, mostly depending on physical inactivity and depression than men, 10 points difference. So this brings us to the knowledge of how important it is to be culturally respectful and to adapt the needs of the interventions and the needs of the research to the culture we are aiming to bring knowledge to. So what do multi-domain trials like Latin Fingers bring us to the table? This is the first or most important emblematic multi-domain trial. It was conducted more than 10 years ago in Finland. It's the original finger trial in which they compared two groups, a control group who received only regular health advice and a multi-domain intervention, tackling mostly nutrition, physical exercise, cognition, cardiovascular control, and socialization. And the results were impressive. There was a change in global cognition and a change in executive function, memory, and processing speed. This led to a very broad worldwide initiative. You must have heard about the Worldwide Fingers Initiative, which is an initiative that tries to bring an implementation, a harmonization, and a validation of this research to different countries around the world. I will talk a little bit about the pointer results. The US pointer is the trial member of the Worldwide Fingers, which was conducted, led by Dr. Lara Baker and supported by the Alzheimer's Association at the US and presented their results at AIC last year. The US pointer then is a multi-domain intervention, and they bring the same core components of the interventions to the table for the participants, but these interventions are not the same as the Finnish ones, but they are harmonized and they are validated to be culturally respectful and idiosyncratic for the US population. They have 30% of the, or more than 30% of their participants being underrepresented population within the US, and they have 5 centers participating. And the results, I can briefly describe this, but you should read the paper. The results they showed at the AAC last year show a very significant effect of the multi-domain intervention on the population at risk of dementia. They also found effects on global cognition and executive function. So finally, our Latin Finger trial, this is a multi-domain intervention too, but maybe the most important characteristic is that we will bring together leadership from 12 different countries in Latin America. So we have 12 different PIs that have been conducting this trial. The design of the trial, these are the PIs, and this is the group, because we have one PI, but we have a very big network of people working in this intervention. So these are meetings that we held at Colombia, at Argentina, at Brazil. We also had one in Cancun, in Mexico, and in Dominican Republic. Yearly, we meet in person at AIC, and then we have our own meeting. So Latin Fingers is a 2-year multi-domain intervention for people at risk of dementia, and we try to test first the feasibility of this, because it's the first multi-domain intervention across Latin America. So the first aim was to test if this was feasible and possible, and the secondary aim was efficacy. For this, what we had to do was to bring from Finland and to bring from the US the different the protocols and the design of the trial. And this we call the external harmonization. We harmonize our design to the Finnish original trial and to the US pointer. We work like sisters with the US pointer, trying to match our outcomes, our interventions, our population and eligibility criteria in order to make these studies comparable and to have like an American axis where we can collaborate together. What we did, so is this external harmonization with their criteria and with the Finnish criteria, but we also had to harmonize within Latin America, because as when I described the modifiable risk factors, each country has their own culture, their own customs, their own risks, so we cannot do the same in each of the countries. So what we did was to establish working groups in which we had one leader. For example, I was the leader for the outcomes. And Dr. Ana Vigil from Mexico, she's the leader for the nutritional intervention. And in every working group, the leader coordinated the negotiation and the discussions of the 12 different countries. So we got interventions that were had the participation of all of our centers. So, as I mentioned before, we have 5 interventions as the finger and as the pointer trial. I won't get too deep into this, and we have another group that has regular health advice. These are the outcomes. We have labs, cognitive assessment, physical exam, plasma and biobanking, MRI, and we have also APOE genotyping at the beginning of the trial. So this builds a very big biobank, which is in some way one of the results of the trial. This is the first longitudinal biobank in Latin America that has outcomes before and after the intervention. And we have behavioral, MRI, blood, and DNA samples. And as I told you before, we worked very hard in the trial harmonization. Maybe the most important things to say here is that we look for interventions to be to respect the core features of the US pointer and the Finnish initiative, but we also want them to be culturally respectful, affordable, and in that sense also feasible as public health policies eventually. So, for example, just to give one example, in the physical domain, in the physical intervention, what we did was to follow exactly the US pointer protocol with the frequency, the intensity, and the type of exercise, but we modified that exercise, and as you can see in the slides, we have on the top Mexico doing the exercise on the parks, because they are used to doing exercise outdoors. We have Costa Rica doing it on a gym. We have Chile doing it inside the hospital premises. So these are some of the characteristics of our population. We have a lot of women participating. Our ethnicity is mostly mestizo. 60% of our sample is at mixed. We will be presented the results which are embargoed at AAIC this year. So why does LATAM Fingers matter? Why do we think it's relevant? Because it's about capacity building. It's about people, systems, and it's about networks. It's about building and strengthening regions to be able to, as we mentioned at the beginning, to be able to then make research that's relevant for their own culture and relevant also for other cultures, because as we live in a global world, we know that Latinos are everywhere. So I will give you a very small example. This is an example of a PI. She's Lisette Tuque. She's from Ecuador. This is the first intervention trial she has ever participated in. She had only very basic research experience, and she joined our initiative because she had collaborated with some researchers before. Here she's training the personnel before the startup. So we had in-person meetings, and we also had Zoom meetings to train personnel. Here is what she did with some of the resources from the funding we got from the Alzheimer's Association, in which she built the infrastructure that was necessary to receive participants in her center to have the physical exercise done there, because gyms are very expensive, and they are mostly located in very rich regions. And she's located in a very suburban area, and after having many problems with people traveling to the high-income areas to get exercises in the gym, she solved the conflict building the gym close to the neighborhood in which participants are living. And this is her collaborations, which she had to do by herself, new collaborations internally with a laboratory and with an MRI company and her own team. So I think that this is an example of how when the trial design also includes the design of building local leadership. This can be very productive and can build the infrastructure for new trials like the one I will show you now. I will talk from risk to protection. I will talk now about a project we are leading together with Jorge Libre from Washington University, which is the Super Aging Project. It's a project on healthy aging, and it's regional. We are funded by the Alzheimer's Association for some of these countries already, and we are searching for funding for others. The importance of this is we are trying to follow a cohort of healthy agers and trying to identify who of those healthy agers are super agers. For this, we try to follow the Mesulam criteria. We are working closely with Emily Rogalski in this, trying to follow their criteria to detect or to identify who is a superager. But something similar to what we did with Latin finger is going on. As you saw there, most of the investigators in this are part of that network, so this is like a spin-off of that network. So what we found is that this criteria is not easily applicable in our region. When you look at the memory performance of a healthy, cognitively healthy and healthy individual in high-income countries, the numbers don't match with those in our regions. And not only that, but in our regions, in low and middle-income countries, we don't have many norms, many publications with really valid local norms for identifying normal performance. So, for example, in this unpublished work by Jorge, he looked at the 1066 cohort, and he looked at normal performance in that cohort, and he found that the normal performance in the memory test in the CERAD was quite lower than that expected in the same population in high-income countries. And the same happened in this paper by Dr. Caramelli about the age that is expected when you try to identify somebody who's superaging. You would think that age can be transferable, but that's not the case because life expectancy is different in low-income countries. So we change the criteria, we adapt the criteria to be sensitive to our own population. So what we are doing is we are working on pre-existing cohorts. Flenny has a very big aging cohort, so does the 1066. So these three countries that are beginning with the study are already rooted in those cohorts. And we will try to find in this population what is justifying superaging and what is explaining also normal aging. We will do biomarkers, and we are trying to find if superaging has to do with resistance or resilience, if biomarkers are positive or negative in the different two groups that will be divided. So my conclusions have to do a little bit with this, being dementia risk being multi-determined, and also that dementia prevention and the study of aging has to be culturally adapted. Also that Latin America and low and middle countries have not to be thought about as replication scenarios, but also as discovery scenarios. In that we have to be very careful in empowering leadership in our region, and also that we need to build capacity. So every trial, every opportunity to research has to leave behind something for the research community, the investigators to capitalize and to grow further. Thank you.
Thank you. Wonderful talk. Next will be Hernando Santamaria Garcia from Bogotá, Colombia.
Well, thank you so much. I'm so happy to be here. I did my PhD here, so this is a happy return for me. So I will try to convey you the idea that all the social factors that are associated with brain health disorders could be tracked by measuring different biological metrics. And in that case, I will present you some of our preliminary results by using epigenetics. Just to start with, when you see the global burden disease image about how the prevalence of dementia could grow up in the next years, you see a clear differences between the global north and the global south. And those increments in the global south are mainly based on or explained by the presence of non-communicable diseases and the presence of all the modifiable risk factors which have been studied by the Lancet Commission and Professor G. Livingstone. But not only the modifiable risk factors, also are important the social factors, the social disparity factors. When you see the case of Latin America, you could find that in that region we have high presence of poverty, high numbers of social exclusion. We have, for instance, in my country, in Colombia, we have more than 7 decades of armed conflict, but also we have high rates of low education, high rates of isolation. So we have a very cumulative, massive cumulative burden of social risk. And in that case, in our region, we also have high prevalence of non-communicable disease. We have prevalence of high rates of diabetes, high rates of stroke, high rates of hypertension. So we have like a strange mixed combination of social exclusion, but also we have problems with obesity. We have problems with cardiometabolic risk in general. That was one of our interests recently in an article published with my team and led by Agustin Ibanez too, and we tried to track which of those modifiable risk factors could impact in healthy aging in Latin America. And for our surprise, we detect that at the same time, even in the same range, the non-communicable disease impact in the healthy aging as the social factors do. As you can see, there's a lot of explanation of the healthy aging outcomes based on the factors like socioeconomic status, isolation, or education. That in our regions, education is a social factor, it's a social determinant of health. Even more, you could track, and that was our interest in here in this article led by my colleague and my friend Hernan Hernandez. He tried to create like an age estimation metrics based on non-communicable diseases and non-communicable risk factors. And he created like a biobehavioral clock. That biobehavioral clock was after estimated by each country and each continent. And we observed that some regions look younger or older based on those non-communicable factors, as is the case in Europe, like look younger compared to Latin American countries that look older, just because of their profile of non-communicable disorders. And that metric, that estimation metric of age based on non-communicable factors is also affected by a group of different exposomes, like country-level exposomes, like a level of inequality in the country, level of gender gap, level of political factors. So with that, I try to convey that age estimation metric could be a good metric to assess how those non-communicable factors impact in the prevalence of dementia, or in the prevalence of brain health disorders, but also how those country-level factors could also impact the presence of those factors. So we have a very specific burden of social factors and non-communicable factors in Latin America, but at the same time, as my colleague and friend David Aguillon showed you, we have in Latin America very specific ancestry and genetic factors associated with neurodegenerative disorders. We have one of the largest kindred families with presenilin 1, which is led by Dr. Lopera and Dr. Aguillon, but also we have very particular genetic and phenotypic associations with some mutations revealing different profiles of neurodegeneration and very particular and specific genotype and phenotype expressions. So considering that we have very particular social risk factors, very particular non-communicable patterns, and very particular ancestry and genetic factors, it's interesting to see how those social factors could be biologically embedded in some metrics like epigenetics. And that's our interest in this project that we are starting to develop, which is trying to assess the changes in expression of genes by using DNA methylation, which is one of the most used techniques in epigenetics, and try to see how those epigenetic marks could be impacted by social risk factors, but also how those factors could be a good metrics of brain health measures, could be good metrics of progression of disease, for instance, and other public health metrics. With epigenetics, as Agustin showed, it's also possible to generate clocks or estimation of age based on the profile of methylation across the genome. And you could detect if your patterns of non-communicable disease or your lifestyles could affect your biological age or your epigenetic age by just trying to generate that metric. So you could develop epigenetic clocks. You could also develop organ clocks based on the pattern of methylation across the genome. And you could also generate different analysis by using environmental wide association studies, something very similar to GWAS. It's just by using this kind of approach. And you could also implement like candidate genes procedures to detect specific biological pathways that could be affected by social or non-communicable patterns. So that was our interest, to try to see how individual social risk and country-level social risk could impact different metrics of epigenetics by using DNA methylation scores. And that is supported by the ADAD-1 that we developed with my friend and colleague Agustin Ibanez and our colleague Michael Corley from UCSD, with the support of NIH and Alzheimer's Association, of course, with all the platform built by Red Lab in Latin America. That was an interesting. Here I will present you very fast some of the preliminary results that we have by using this approach. DNA methylation metrics, and assessing how the social risk factors at different levels could impact those metrics. In that case, we try to assess how the socioeconomic status, social support, education could affect different groups with high social risk or low social risk in terms of epigenetic ages. And we observe that people with high social risk looks older than those that have low risk. And in particular, we created that metric, which is a metric, the epigenetic age acceleration metric, which is a metric of, as the name say, a metric of, it's a kind of clock. And we could say here, and we could see here that the different organ clocks, specifically brain clock, omic age clock, inflammation clock, liver clock, system age, and general epigenetic clock, are accelerated when you have a higher risk, high social risk in general. And this pattern is observed in the full sample, but also in particular is observed in dementia samples. We also tested some mediation analysis and see if the general pattern of psychosocial global risk factors could be, and the some epigenetic clues could mediate the association between that pattern of risk with some brain health measures, in this case, cognitive measures, and we observed that the epigenetics could be a mediator of the relationship between those social factors and the brain, of those social outcomes. We also track how those clocks could be referenced in cortical patterns, but also in functional mechanisms in the brain, and we observed that some of those patterns could be tracked also by those metrics. Here we have some specific clocks could be represented in cortical atrophy patterns in ID and FTD in our samples. And just to finalize with the last group of results, we are trying to develop some models of EWAS, environmental wide association studies. We are aware that we are underpowered in terms of sample size because we only have for now around 500 samples. But we started to try and control and generate the algorithms to run the EWAS studies for social adversity in Latin America, and here we have some preliminary results. This is the general description of the Manhattan plot for describing how different risk factors like socioeconomic status, social support, and integrated social risk score and education could be tracked across the genome. we implemented different models and strategy to control the inflation associated with the different tests that you implement with this kind of technique. And we control Bonferroni, and we generate very robust models to see the effect size with those associations. And just to summarize, we obtained this pattern, which is a hypermethylation pattern in the promoter of the WNT9B gene, which is a very, very interesting gene placed in the chromosome 17, very, very associated with those processes, neuronal survival, neuroplasticity, and reduced inflammation. So that pattern that we observe maybe could reflect why there are some of those consequences, like reduced neuronal survival, reduced DNA, and reduced other biological pathways, very, very associated with neuroplasticity that could explain a little bit the pattern of neurodegeneration that we observe in our populations. So just the concluding remarks, just to see that, and just to mention that brain health disorders show marked global disparities with higher prevalence in Global South, and individual and country-level social determinants may have explained those patterns. Biological factors and social exposomes interact and are better strategies to understand the complexity of the brain health disorders. And epigenetics is a good tool for capturing the biological embedding of the social adversity. Accelerated organ and epigenetic clocks are associated with high social risk in healthy aging and neurodegeneration, as shown by our data. Those clocks also predict brain health measures, which could be a good tool for implementing in clinical settings, in prevention settings, but also in public health strategies, because epigenetics is sensitive to capture some changes in those metrics. And results in environmental wire association studies also shown that a specific biological change in a specific biological pathway, the WNT9B pathway, which could be explained some of the neurodegenerative patterns that we observe in that population. So just to finalize, social disparities become biologically embedded with important clinical and public health consequences. This is our team. And I want to thank all of them and the team that we have in Colombia, a group of very younger and early career researchers, very talented and very happy to support those kind of initiatives. And just to say thank you to all the team of Red Lab, NIH, of course, Alzheimer's Association for all your support. And thank you so much, Barcelona, for having me.
Thank you so much, Fernando. Beautiful presentation, very incredible all the work that all of you are doing. I'm feeling very grateful to be here at the first row, seeing all these interesting results. So now we have time for some questions and discussion. And perhaps while you are thinking, and I will start by maybe some curiosities that I have when I was seeing your presentations. One is connected with sex differences. Lucia was showing some results, yes. But I was actually very curious on the brain age gap, because you show the global analysis. I'm not sure if you were also, and if you can explain a bit the difference that you found for men and women.
Thank you, Nadia. It's a great question, and indeed, yes, we have, in many studies, we have shown some bad news, like, the first study we did in brain clocks, we shown if you live in the south, you tend to have an older brain, right? And regarding your chronological age. If you have dementia, of course, but if you are female, also you have, accelerated aging in comparison with men. So we already shown this effect. Then when we use the biobehavioral age gaps, we included sex as a factor driving the clock. And in fact, it was associated, but even when it was associated, when we did stratified sex analysis, we have larger accelerated aging in female. I don't remember, I don't know if Hima or Juan are here, I don't think that we do gender results in the Down syndrome study, right? No. No, I don't think so. That is, but I don't know in epigenetics, probably you can complement that, right?
Yeah, so far, maybe you have conducted sex-specific analysis. In what I showed, I did not, yeah.
But the preliminary results that we have show differences based on gender, because females look accelerated in comparison to men. But the effect of the other social risk factors is even higher than that pattern. Yeah.
That's a very important result. In the Nature Medicine paper, we've shown that the effect of social disparity is larger than the effect of sex. You know, and this is also even larger than the effect of age, that is the larger one. So this is a great question for me. What means this risk for females? Is just sex? Is the conditions? Is the combination? So from environment to genetics, there are multiple factors that need to be.
Actually, I was very surprised with your model methylation, social factors, and then the covariate. You were including sex there as a covariate.
Yeah.
And I was thinking maybe you are removing some heterogeneity there. So, yeah, I don't know how you are interpreting this.
You know, this is a standard procedure to control the inflation of factors that could affect your results in epigenetic modeling. But I think that we could trying to control for other many factors and trying to produce different models by
and
females
do
and males.
Stratified analysis. The problem is the sample size is very preliminary, so
Yes.
You can try as a modifier of the effects.
Yes.
Sex as a modifier.
Well, one of the, continuing on this line of discussion, we know that in Down syndrome and in ADAD, there is a pretty significant age of onset variance amongst individuals. And when you link that back to the clinical trial discussions that were going on yesterday, I'm wondering if these social biological epigenetic clocks may help explain some of the age of onset differences, because especially Global South may actually be a unique opportunity to hone in on that from an intervention standpoint, like is starting up. And I'm curious about your thoughts on this.
You know, thank you for the question, but this is the tricky one, because you obtain the samples after the disease started. So in Latin America, you have a problem of reporting the symptoms. You have like a cultural protective environment. So you only go to the clinics when you are very affected. And maybe a good strategy to answer that question could be to have like longitudinal models, cohort models. But just to anticipate some of the works that we are doing in that way, with the samples that with presenilin-1 mutation, we observe that they also have differential patterns of epigenetics and affects the age onset. So maybe epigenetics could be a good metric for tracking those clinical measures too. Yeah.
Any questions?
Well, that's interesting. I was discussing yesterday with David Aguillon, who's here, the age of onset report. I think we talked about it, how it's like a paradox, because highly educated people tend to report the disease before people who don't have that knowledge. So you think that the clocks will be turning the other way around, that the disease will be evident in low educated people before, people with lower reserve before, but at the end, the people who are more educated tend to report the symptoms before them. So something to take into account.
Yes, and maybe just to add to that, although it's true that we get the epigenetic measures in our case after like the onset of the disease, we can also like consider that epigenetic and epigenetic clocks represent like all the exposures we have had through our lives. So it's the accumulation of exposures. Yes. So we can, although it's also changes like in time, but they are like there's this epigenetic memory, right? So it's like also we can see like a chronic exposure, and then the age of onset of it's also like let's say like a chronic effect, and then we not only can see epigenetic as the let's say, before the disease. Epigenetic can also be the consequence of the disease. So we also do not know the direction. We cannot right now measure directionality, but well, we can do some causal analysis, right? Some indeed randomization and stuff. But it's also good to see an epigenetic pattern of the age of onset.
This is interesting considering that you also have the internal exposome, and you could try to frame that as saying that when you have neurodegeneration, you are changing all your biological process, not only the deposit of proteins in brain, but also you have modifications in metabolism and inflammation process. So you have a kind of different exposure that could affect the epigenetics. So it's difficult to control for that, but it's interesting too. Yeah.
Well, given the expertise and the data shown on this panel and the individuals in this room and the collaborations, it sounds like there's some really wonderful opportunities to apply these methods in the Down syndrome cohorts that are available, as well as the ADAD cohorts that have data and longitudinal and all of this multimodal data, and you're already collaborating on some of these things. Sounds like this would be a wonderful opportunity, because we really, truly need to refine that age of onset for the clinical trials, especially if the therapeutics become available in this country. We really need to refine that, because we need to make sure we're getting the therapies to those in trials when they should be in the trials, and those to therapy when they should be on therapy.
Yes.
Can I say something about that? I think that multi-aging clocks in general that are a mess, from proteomics, epigenetics to biobehavioral or brain clocks, but you know, nowadays nobody agrees what is really aging. So I think that we really need to develop a multi-aging approach. And this is a unique opportunity to bring both vulnerability and resilience, right? So, and how we can do, we are fans of doing, you know, environmental factors, social factors, but biology and genetics are essential, and we need to connect this. So I think that multi-aging clocks really can connect both genetics and environmental drivers of age of onset, of burden of the disease, and even we could be used for monitoring the disease and the intervention. So I hope to convince the Down syndrome guys, and Imer should be around there, to develop something similar to what you are doing in Africa, but also with Latin America.
Well, and one of the other beautiful things about this component is these methods can be applied more broadly, and then those who need to be scanned can be scanned. So, one.
Yeah, we need that microphone.
Oh, we don't have a microphone. Can someone please bring El Jefe microphone? He is the boss.
Here in the top.
Here,
the
first row.
Here.
Thank you, Sid and Agustin. And I really like what Lucia was saying about building capacity. And I think that the grant that with Jason, I don't know if Jason is here, that he put a supplement to his R01 in the NIH. So if Jason, after me, you want to comment on that, I think it's the capacity building, because I think the problem with Down syndrome is oftentimes it's just they are neglected. And oftentimes it's just you just need a small group of investigators and clinicians that take care, and they can have a huge impact in the quality of life in taking care of these patients and everything. And so it's this capacity building that Lucia was referring to. So and on top of that, we could learn a lot of things. So I think that I couldn't agree more that with this infrastructure in place, it would be very, very easy, as we discussed yesterday, Agustin, to some of the centers in Red Lat. If you have, with very little investment, you could have huge scientific output and a meaningful change for many, many people living in those cities, because there are plenty of people with Down syndrome that are not seen. When we go to neurology conference here in Spain, the question is, if you're not treating people with Down syndrome, with Alzheimer's, who do you think will treat them? Here in Barcelona, it can be us, but in the rest of Spain, I mean, it has to be you. And sometimes you see some faces like stunned, that they didn't even think that Down syndrome was their thing. And if that Alzheimer's is not yours, who is going to take care of that? So yeah, I think building capacity is hugely important, and we'll learn a lot. So. Yeah. couldn't agree more.
Well, thank you so much. This is so important, and I think that FLENI, for instance, David Aguillon in Colombia, and other centers are already starting to do research in Down syndrome. Right? So we will have the emerging groups, and I think that we can try to convince the Fundación Pascual Maragall to helping us to build this Down approach in Latin America. Okay.
Yeah, because I think that's important. We are part of the R01 project from Jason Hassenhab. I think when we talk about capacity building, sometimes it's misunderstood as building infrastructure or buying components, or it has to do with building a culture too. It has to do with building leadership, with giving leaders independence. It has to do with researchers having the knowledge that they have to gain, and also capitalizing what the region already has. For example, in Argentina, there is a big Down syndrome community. Lucia Pertierra has been working in that community for a long time before this project came along. So she's leading the clinical core of the team. And you have to get to know the place in which you are going to work, the population with which you are going to work, and like leverage on the people who already have the experience and make them strong enough to take the risk to go into research sometimes, because these are, when we talk about LMICs, we are talking about a lot of possibilities and opportunities, but a lot of challenges in taking the risk of, for example, changing your clinical orientation to research one. No?
Indeed.
Hi, very fascinating presentations. I was just wondering how easy or how difficult will it be to implement these intervention studies into public healthcare in Latin America?
This is for you.
Into what? Public health care.
Well, that's the goal.
Yeah.
The
knowing
research,
like all the challenges.
Research is oriented in order to make a difference in the region. It has to be, so when we talked about harmonizing what was done in Finland and then being more similar to the US, it has to do with that. It has to do with thinking about something that's affordable, that is culturally relevant, and that is feasible as a public health policy eventually. Because if we see that there is change using this intervention, then we can make a difference that impacts what we saw at the beginning of the presentation with the modifiable risk factors. And being that we were flexible enough so we were respectful enough to follow the core features of the US pointer, but we were flexible enough to make the interventions relevant for our own populations in each of the different countries, then this will be friendly enough to be applicable as a public health policy if the results are positive.
Yeah. Are you working with the policymakers right now in Latin America?
Well, that's the aim. We have to see the results first, and that's the aim of our project is to work together with public health leaders in the different countries.
Perhaps also continuing with the discussion of the risk factors, Hernando, you mentioned or you show these models with the mediation using epigenetics as mediators. And I was thinking about our discussion this morning and yesterday on medication, which is, I think, a really important factor, or should be.
Yeah. Which one?
Medication.
Medication. Okay.
So how can we be sure that this is a risk factor or a mediator, or what is the role of medication there?
Well, you mean medication for different conditions, any conditions, and not necessarily medication for
not necessarily,
AD.
but also,
I mean, I was trying to see that some of those patients could receive the I don't know, donepezil or that kind of dementia drugs.
Yeah.
Mm-hmm. But most of them also have medications for cardiometabolic risk. Mm-hmm. We need to control that. I don't know exactly. I don't know exactly what is the role, but of course there's some reports showing that medication could modify the patterns of epigenetics too. We need to create those models controlling for,
And as a social factor, as we were discussing?
Well, that's interesting because we were discussing that in some ways you could have like a social metric could be how the population use some kind of drugs, for instance, sleep pills or painkillers or antidepressants. So in some ways, this is kind of social metric, and we are interested in see how those factors could be associated with some mental health outcomes, with some well-being measures, but at the population level at the moment. But of course, that kind of, the drugs could also impact it. It's interesting because it starts to be considered like a social determinant of health. Not only the kind of drugs that you use, but also the drugs that you could access, or the drugs that you could have options to be available or using that. In Latin America, you have problems to access for some drugs. You receive the generic forms of the drugs. So it's interesting, but for me, it's a new project. It's a very complex project, yeah.
Well, it sounds like there's a lot of opportunities, especially to expand, as Juan, we were discussing earlier, I know that's discussion that's going on, the Down syndrome work, especially as communities already working with populations. Obviously, the population's there, and the population is interested. So now it's what are the next steps and how do we move forward so that we can bring these sorts of interventions, sociocultural, behavioral, as well as pharmacological, to the Down syndrome population across Global South. So now we just got to figure out how to make it happen.
Yeah. Thank you, Sid.
Really remarkable presentations. Thank you for, hi, I'm here. I just wanted to make two comments. One is that I just am so thankful that we can do this and get paid to do it. And I think we owe our funders like a real gratitude, especially the courage they have to fund these new things, because we're really making a difference. We're not just searching for p-values and papers in Nature Medicine. And I remember having a conversation with Lucia a few years ago when she was first doing fingers. And I said, well, is it going to work? And you said, well, of course we all want it to, but I think you said the more important thing is that we're building this to make an impact, and we're changing the culture. So we really want it to work, but we're doing something broader. And we had the same excitement about moving our Down syndrome study into Africa a year and a half ago. Unfortunately, we ran into some obstacles with our current administration in our efforts, but we're still hopeful that that will happen. But it's just amazing what these little things that we can do, how they flow out to societies and into cultures. And I think the consequences of that are really immeasurable. So sorry for my sentimental journey, but that's all I wanted to say.
I would like to add something to your comment. It's not only, so it's true that it also has a social meaning, because interventions, they work. What's difficult is to spread the word, to communicate this knowledge to the participants, because one of the main problems is adherence to this program. So that's something that needs to come together with the science behind it, like how we communicate to the people. This really works. This is beneficial for you, but you need to keep it in time. So there are also a challenge is also to see whether the beneficial effect lasts on time. So some studies study, okay, after 3 months, 6 months, after a year of the intervention, do they still have an effect? Participants still do those exercises, for example, at home? Do they change the lifestyles? So it's also nice to measure if the knowledge of the person receiving the intervention and the habits changed through, before, right, and after the intervention. Because a lifestyle intervention, people need to, feel them, not only do them because they are enrolled in a trial, they need to believe in them because I need to keep doing them.
Yeah, I can comment on that, on our experience in fingers in 2 points. The first one was when we started recruiting participants. Some centers were drawn directly to high-income communities in their cities because they thought it would be easier. You know, this was also a feasibility trial, and we wanted to meet the milestones and to recruit participants. But then we found that people from low-income communities were much more receptive because they were grateful for this. In Latin America, you cannot get paid for participating in a clinical trial, so that was not part of the equation. They just wanted to have contact with the knowledge of how to take care of their brain, even if they were randomized in the control group.
Yeah.
They were happy to participate. And that difference, we talked about it at the AASC conference in which we had a panel on recruitment. That was a very nice surprise for us to know that low income and low educated people were very receptive. And then your second comment was about adherence. And what I can tell you is what we are doing in Latin Fingers, I didn't have time to comment it, is an extension study. So we've been funded for continuing evaluating these persons, but like open label, the two groups together. They have to do their own lives. They don't get the intervention after the intervention finished, after 2 years, and they get assessed every year in order to see if these trajectories are divergent after the intervention too. Because as you say, it's important for they to own the life change. No? So yes.
That's great.
Okay. Well, thank you for having us, and look forward to having a panel like this again next year.
Thank you. Thank you.
Will I ask her to turn her camera on?
Dorem, can you open your camera, please? Can you hear me? Sí. Dorem, I don't have your mic and your camera. Can you open, please? Okay, I can see you. Can you speak? ¿La oyes?
Hello.
We are ready.
Yes.
Okay.
Yes, I'm ready.
Dorian, sorry, this is Emer. Just letting you know, we'll be starting in like 2 minutes or something like that. Is that okay?
Okay, no problem. Thank you.
Thank you so much. Thank you.
Eso es. Ah, bueno, me equivoco. No pasa
nada.
Igual.
Bueno,
yo creo que aquí está viendo.
Hi, everyone. Thank you so much. So this next symposium is around making research participation meaningful with perspectives from our participants who are part of the DABNI study and also from family members. We are joined as also by Noreen from South Africa, who's the mother of a young man with Down syndrome. So when we were thinking about this session, it was really thinking about how and why the participation of people with Down syndrome in the development and the design of research is so important. And I think looking historically of some of the really unethical practices, including people with intellectual disability in the 50s, where children with intellectual disability were deliberately infected with hepatitis, and through that it led to the development of ethics committees. And then over a period of time, from around the 70s to the 90s, there was a real exclusion of people with intellectual disability of Down syndrome in a lot of clinical research, and there was a real overprotection that led to then unethical exclusion. And so I think in more recent years, there's been more of a balance and a real understanding of the real importance of including people with Down syndrome. And so in the session today, we're talking about PPI, which stands for public patient involvement. And this is where people, any population as part of a research study, are involved not only as passive participants, but are also actively involved in whether it's around the initial study design or reviewing information to the study to make sure that it's accessible, and then through to dissemination. And I think especially now as we're moving into clinical trials, we really need to consider this to make sure that the information that we're giving people is accessible, that people are understanding information, particularly around risks, and that in developing that information, we co-develop it with our partners and participants with Down syndrome. I think also this is an important reminder that as we're talking about the importance of including voices of people with Down syndrome, and that it is hugely important, that we also think about the voices that are often absent. And one of the most notable absences is the voices of people with Down syndrome across the Global South, and particularly across Africa. And so I think as a field, we need to consider how we're able to include these populations where from recent studies, it seems that across US and Europe, there's approximately 1 million people with Down syndrome, which means that there's approximately 5 million people with Down syndrome outside of the US and Europe, but that we know very little about their health comorbidity, about their aging trajectories, or even around life expectancy. So I think this is a really important area. I'm delighted to be joined by Doreen today to talk about some of her experiences of participating in research in South Africa. But first, would like to introduce our panel. So I might ask Antonio, would you introduce yourself?
Pues mira, a mí me pusieron como un PIT, quiero decir, para descubrir lo que es el Alzheimer.
Okay, so Antonio is saying that he is participating with us to discover about Alzheimer's. Antonio, cuéntanos un poquito qué cosas te gusta hacer en el día a día.
A mí lo que me gusta hacer todos los días es la tablet con la música.
So he's saying that what he likes to do in today is mostly the tablet and listening to music. Vale, ¿y qué edad tienes, Antonio?
55.
And he's 55 years old. Vale, muy bien. ¿Algo más que nos quieras contar de ti o pasamos al siguiente compañero?
Estoy con mi hermana siempre.
And he also likes being with around his brother all the time. Vale, muy bien.
El único que tengo.
He's his only brother. Vale, bueno.
Eloy, will you introduce yourself?
Eloy, si quieres contarnos quién eres, tu edad, qué cosas te gusta hacer y presentarte.
Is my name Eloy? Soc l'Eloi. Jo tinc 38 anys, visc a Vic amb el meu projecte de vida, amb la meva parella. I ara estic treballant a Creu Roja d'Osona i també estic treballant a Eurofilms com a ambaixador. I a més m'agrada, soc casteller i estudio a la universitat en anglesa.
Okay.
I a més soc periodista, periodisme, Èxit 21, la revista digital de la Fundació de la Síndrome de Down.
Okay. So he is 33 years old. He lives with his partner in Vic, and he has he works in 2 places. He works with the Red Cross, and he's also a castanyer, which is a Catalan community thing, and dancing and yeah, no, and sorry. And he does English classes at university, and yes, I think
journalism. And journalism.
Oh, and sorry, yes, and he's also involved in journalism.
Yeah, thank you.
Muy bien, gracias.
Ara la Montse et pots presentar, Montse?
Hola, bona tarda, soc Montserrat Vilarrasa Monclús, tinc 45 anys i estic vivint fa 12 anys que visc de forma independent i treballo en una empresa ordinària d'auxiliar administrativo.
Muy bien. So Montserrat is 42 years old. She has been living independently for the last 12 years, and she currently works as an administrative auxiliary in an enterprise.
Yeah. So, Montserrat, vols llegir això que havies preparat, que ens volies compartir amb nosaltres?
Sí, la Fundación Catalana Síndrome de Down estic una revista digital d'X21 igual que el meu company Eloi, i també estic a l'Assemblea de Drets Humans de Montserrat Trueta, i des del 2016 soc la representant del colectivo de la discapacidad intelectual en el Ayuntamiento de Barcelona.
Perfecto.
Y, perdón, también en el 2018 vayan a convidar una persona de la María Soledad Cristina Reyes, de la ONU, Nueva York, a firmar una declaración de los derechos de las mujeres, un documento del lideratge polític i públic.
Molt bé.
Perfecto. So Montserrat, as the same as Eloy, they both work in this electronic journal magazine where they write articles and they perform interviews. It's called X21. And she is also since 2016, she is the vocal of the collective of people with intellectual disabilities in the City Hall of Barcelona. And she was invited in 2018 to go with the UN to go to the New York and sign a paper on women's rights and leadership among the community. And I think this is all.
Super. De acuerdo. Thank you so much. And finally, Doreen, would you introduce yourself briefly?
Okay, hi everyone. My name is Doreen Gomba. I'm with my son, Mpho Gomba. He has Down syndrome. We are from South Africa in Johannesburg. He's 19 years old. He's attending school at Jiswa Training Center. Yeah.
Fantastic. Thank you so much, Doreen.
Thank you.
So we, with our participants, we have a few questions that we'd like to ask. And some of the PPI work that has been done across Europe over the last number of months was around the development of a Down syndrome adaptation for the finger study. And with that, we had a number of groups across all of Europe, advisors with Down syndrome in making those adaptations and also in supporting the development of information material around that. And we have, so within each country there was 2 to 3 advisors across each country. Within the Horizon 21 consortium, there's about 8 different languages. So we primarily had the advisory groups as within each separate country and then translated everything back to English. But we did have one cross-European Zoom call where everybody joined, and we translation we made it a little chaotic, but we primarily talked about music, and people shared music from their countries as a way of kind of building the community across Europe. And so we're really delighted today. We have 2 members of that advisory committee from Barcelona with us. So Eloy, can you tell us about your experience of taking part in the advisory group?
Eloy, ¿nos podrías contar cómo fue tu experiencia cuando nos ayudaste a diseñar mejor el estudio para la salud cerebral y sobre el ejercicio físico?
Jo crec i penso que és molt important i hem de ser oberts perquè vull que facin aquestes activitats i que facin que continuïn avançant la investigació i que facin més coses d'això. I a més vull ajudar-vos i vull col·laborar. I a més, l'any passat vàrem fer un vídeo Zoom amb persones amb síndrome de Down d'Europa i vàrem parlar del tema de la salut i del món de l'esport.
Perfect. So Eloy is telling us about how he thinks it is important to keep advancing in research and to do these collaborative experiences and to be able to say their voices. And he is telling us about the experience he had in the video Zoom that he shared with other people with Down syndrome, where they mainly talked about health and brain health and sports in daily time. Muy bien, muchas gracias, Eloy.
Ahora, Antonio, ¿sabes que hay algunas personas a las que no les gusta hacerse algunas pruebas, como por ejemplo la resonancia magnética, tú has estado participando en un estudio y te has hecho esta prueba y queríamos saber tu opinión sobre esto y cómo ha sido.
So we are asking Eloy how his experience being in an MRI was, because some individuals with Down syndrome or families and caregivers may be scared of it or may have difficulties. So we want to know his experience.
Yo lo que iba a decir, todo el mundo que somos personas civilizadas lo pueden hacer igual. A que seamos así, a que tengamos hijos de familia, ¿vale? El problema que tienen se lo pueden hacer cuando ellos, los padres lo digan.
Okay, so what he's saying is that any individual is able to perform an MRI, whether Down syndrome or not, and that, yes, he doesn't see any issue with it.
Yeah, the only issue he mentioned is that some of the parents don't allow their children or their families to participate because they think they cannot be able to go through the MRI. So that's the only limitation. It's like some parents don't allow us, and why? Okay. And now, Monse, tú has estado ayudándonos aquí para un estudio en concreto que permite entender si hay algunos mecanismos en el sueño que se pueda haber afectado o no, y has pasado aquí la noche, ¿no? Has venido aquí, te has hecho una prueba que se llama polisomnografía y nos gustaría saber cómo fue tu experiencia.
Yo creo que las pruebas positivas, he hecho la prueba del son, del reloj también y un TAC también.
¿Y qué tal fue dormir aquí?
Y analíticas también.
¿Y qué tal fue? ¿Cuando pudiste dormir o no?
Sí que he podido dormir. Pero la persona que estaba al lado, no mucho.
No.
Bueno.
Puede pasar.
So Monse participated in an R1 study that involves having a PSG recording and sleeping in our laboratory, and also wearing actigraphy, so 2 clocks around waists, and having a blood analytic. And we asked her about her experience, and she reminded us that she also had a CT scan, which we didn't mention, and that it was good for her, that she slept well, but that her caregiver, which was in a seat below, didn't sleep so well. Yes, that's it.
Yes.
Perfect.
So when we were doing this session, I thought it would be, I immediately thought of Doreen and her experience in South Africa, and I think there's a lot we can learn about how we can connect with either underserved or remote communities through an amazing initiative and WhatsApp group that Doreen has developed. So Doreen, would you explain about the WhatsApp group that you have across South Africa and how it supports parents and families of people with Down syndrome?
Oh, okay. Yeah, we have a WhatsApp group that is called Network Parent. It's across 9 provinces in South Africa. So our focus, main focus, is to become leaders in understanding our children with disabilities. And we also fight for their human rights, like e.g., right to education, right to equality, and also safety from abuse. And, you know, as we are gathered in that group, we are also learning from one another. Like each time there are challenges that we are facing in our communities, so we uplift one another, that when someone is mistreating you or is discriminating your child, what are the coping mechanisms that we can make to, you know, to teach our communities about our children, because they don't understand them. So the WhatsApp group is so helpful for us to communicate, even though we don't know each other, because we are so scattered, 9 provinces. We haven't met, but we always discuss on the WhatsApp group. So it's a helpful tool for us to meet each and every time when we have challenges. Thank you.
Thank you so much. And I know more recently we used the WhatsApp group. We were doing a research study across South Africa on healthcare and healthcare access and people with Down syndrome. And it was through the WhatsApp group that we were able to connect with a lot of families. So could you tell us about your experience in that research project and how families felt about engaging in research in Down syndrome?
Yeah, regarding the research, yeah, we met. It was so great experience meeting with different caregivers of children with Down syndrome, and most of them, you know, they were so happy when we were telling them about this research. Some of them, they felt that their voices are mattered, and also their insights can help them to shape better support their system, because the system, most of us, is failing us by not giving us enough schools. We don't have school here in South Africa. There are so few, so children are staying with their parents in their house. They are doing nothing. And as they are sitting at home doing nothing, their development, it's not developing well. So that's the problem that they are facing, most of the parents. When we were conducting this research with them, they were telling us that if we can get a school for them so that the children can develop, can show their abilities and the capability of what they can do. So because of they are not going to school, it's a challenge for them. So that was the experience that we felt, and we are trying to partner with Department of Education to get them placed, even though the waiting list, it's too long, because we have so many children that are not in school here in Johannesburg.
Yeah.
Thank you so much.
Thank you so much for that, Doreen.
Thank you.
I think we might open it up to everyone here. If anyone has any comments or questions for anyone on the panel.
¿Hay algún comentario en la sala o alguna de las familias quiere participar o añadir algo de lo que hemos dicho sobre vuestra experiencia o no? Bueno, nada. Vale, sí.
My question is, if you're considering participating in a research study, how would you want to learn more information about what is involved?
Os preguntan de forma colectiva, si cualquiera quiere contestar, si a la hora de cuando os pedimos si queréis participar en algún proyecto, ¿cómo os gusta recibir esa información? O si pensáis que falta que os digamos algo, o querríais saber más, o está bien así, o si tenéis alguna recomendación que darnos. Por ejemplo, cuando te dijimos si querías participar en el estudio del sueño, no sé si a lo mejor te parece bien como lo hicimos o querías recibir más información o distinta.
Bueno, yo creo que las informaciones son importantes. Estas investigaciones investigaciones es probable que las personas como nosotros podemos tener Alzheimer, por eso hacemos estas pruebas y estas cosas, y yo creo que es positivo.
Vale. Monse, cuando te propusimos participar en estas pruebas, el formato, cuando te dábamos la información, por ejemplo, podía ser un dibujo o un vídeo o hablar. ¿Cómo te gusta a ti que te expliquen el por qué es importante que te hagas estas pruebas? Sé que tenemos algunos vídeos que los podemos utilizar, ¿no? Por ejemplo, para que tú sepas cómo es una resonancia o esto. ¿Cómo prefieres?
Pues no sé, ahora...
De cualquier forma está bien. Vale, muy bien. O Antonio, por ejemplo, cuando te dijimos de hacer la resonancia, ¿más o menos crees que te informamos bien? ¿Sabías que tenías que estar tumbado en el tubo y que todo iba... ¿Y por qué se estaba haciendo?
Yo, como yo me relajo, me voy haciendo las cosas para no fallar en las preguntas que me hagan.
Muy bien. Vale, perfecto. So Montse was saying that she thinks she understood why it's important for her, as she knows that she could eventually develop the disease. Being an individual with Down syndrome, she understands the importance of investigation, and she thinks the information she received was okay. And Antonio, I asked him if he understood that he had to remain still in the MRI, and if he understood why, and he said, and for him it was okay, he just relaxed and he was calm.
Correction.
Perfecto.
Bona tarda, i yo volia donar las gracias, muchas gracias a Montse, a Eloi, a Antonio por su participación hoy. Me ha encantado escucharles y quiero darles las gracias porque participar en la investigación en Sant Pau, donde estéis, en el hospital en que estéis vinculados, nos ayuda a todas y a todos. No estamos hablando solo de síndrome de Down, sino estamos hablando de todas y todos, porque todos formamos un todo, ¿no? Y también gracias a las mamás y a los papás que participan en la investigación. Y a mí me gustaría hacerles una pregunta a Montse, a Eloi, a Antonio, también a Dorín, que sería: tenéis aquí a las mejores investigadoras, a los mejores investigadores, a personas muy influyentes que pueden cambiar el curso del mundo y del Alzheimer, ¿no? ¿Qué les pediríais? ¿Qué les pediríais a estas investigadoras, a estos investigadores, desde vuestra perspectiva? ¿Qué les pedís? Como si fuera una carta a los reyes. Muchísimas gracias por participar.
Me siento mejor. Yo me siento mejor las pruebas que me hagan. Porque me gustan para yo tener la memoria fresca.
Okay. So, oh, sorry. Sí, sí.
Y vocalizar las palabras.
Y estoy encantada que está aquí en San Pau, hace investigaciones y todo esto. Hago todas las pruebas que me digan, menos de la espalda, que tengo un 53% de desviación, pero por lo otro, bien.
Perfecto. So I'm going to translate this, and the question was if they had any questions for us as investigators and scientists and doctors, and they didn't have questions, but they said that they were. So Antonio said that he liked being tested and having his memory tested, and Montse said that she was happy to participate in investigation, everything except lumbar puncture, but because she has a bit of scoliosis in the back, and she doesn't want that. And yes. There was another question, yes.
Buenas tardes, soy la madre de Eloi y primero quisiera agradecer la colaboración de tantísima gente en esta investigación, porque creo que es un paso adelante que, además de investigar la síndrome de Down, pues investiguemos las cosas que les pueden producir cualquier enfermedad, que ya no tan solo es de la síndrome de Down, sino que es humana, pero que les puede afectar muchísimo a su manera de ser, a su manera de vivir y a su longevidad en la vida. Entonces, a favor de ellos, lo que quisiera pedir, que creo que ellos a lo mejor no se han acordado, es que uno de los principales problemas que tienen actualmente a la hora de poder colaborar o pedir ayuda en cualquier tema o en cualquier situación es la comprensión de lo que se está haciendo. No nos lo parece, pero a veces cuando hablamos en los medios de comunicación, en la televisión o en cualquier lugar, utilizamos términos muy complicados. Ellos tienen capacidad, seguro, de entender, de realizar, de pedir, de luchar. Las tienen todas las capacidades, pero les tenemos que dar a veces una pequeña ayuda para que sean capaces de comprenderlo, porque hablamos rápido, vamos rápido, vivimos rápido. Y ellos la rapidez no la entienden. O sea, son personas que van a su ritmo, que llegan a todos los sitios igual que nosotros, pero tardan un poquitín más. Entonces, a la hora de investigar, a la hora de pedirles su apoyo, su ayuda, que seguro que la agradecen muchísimo porque entienden que lo estamos haciendo por también una causa que que les toca directamente, tendríamos que tener conciencia de que necesitan información, que a veces ellos confían en que nosotros les decimos, vamos a hacer este proyecto, nos están invitando a que colaboremos con este tema. Y ellos, si nosotros, que somos sus referentes, les damos pie a que lo hagan, ellos tienen total confianza y lo hacen. Pero a veces no les contamos o no les sabemos contar o no les damos toda la información para que ellos puedan entender exactamente qué es lo que están haciendo y para qué sirve lo que están haciendo. Y vuelvo a repetir que solo necesitan un poquitín más de recepción a la hora de poder realizar con plena satisfacción y con plena conciencia cualquier cosa que se les pueda pedir.
Vale. So this is Eloy's mom, and she was saying how important it is to adapt the language, and so she firmly believes, as we do, that they are able to understand and get to everywhere as the same as non-Down syndrome individuals, but they might take a bit longer, so it is important to bear in mind as physicians and scientists to have adapted language and speak slower and be more patient, enable for them to fully understand what is happening and then engage even more than they already do. And this was her main message, I think. Yes.
Hola, gracias por hablar con nosotros. I have a question for Doreen. Kind of the same question that the gentleman here was asking about if you were approached about research, how would you want to be approached, and what would make you interested, and what might make you not interested. We're hoping to expand. We have a number of collaborators in different countries throughout the African continent, and just looking for some more insight.
Oh, okay. For me, if I'm taking, a chance to be in a research, especially with Down syndrome, I would firstly like to know what it's all about, to go through the questions, because whatever I will be looking at it's the question, and then regarding the question, I will check if it is relevant from what my challenges are, because it's pointless for me to take part in a research that is not relevant to my challenges. It will be just waste of time to take, part in that. And also, regarding other parents, when we are doing these researches, we ask for their permission first. We are not just doing it without their permission. Some of them, they will just tell you that they are not interested. And then others, they will participate in it. And then with other parents, they will want to know, what are we going to give them after doing this research? And we are not promising anything. We are just collecting the data, nothing more, nothing less. There's nothing that we have to promise them that after we have done this research, we will give you whatever, maybe a food parcel or whatever. So we are not telling them those, even though they are expecting that. So yeah, I will go through the questions and check how they are. Thank you.
Thank you. Yes. Okay, so I just wanted to thank you, the families, and of course you. And I think we don't have words really to say how it feels. It's like a privilege, I think, for us to perform research with this population. We learn a lot all the time. It's an ongoing learning all the time, because we want really to improve, and I think with that we can finish the session. So, lo que quería era daros las gracias a vosotros, a todas las familias, a todos los que participamos en este proceso en el que no hemos parado de aprender, en el que seguimos aprendiendo. Creo que es un privilegio para los que podemos trabajar haciendo investigación con personas con síndrome de Down, porque ellos nos enseñan todo el tiempo y a veces tienen unos puntos de vista que igual no se nos ocurren, y creo que esto es importante porque queremos seguir mejorando y nada, que realmente creo que con palabras no se puede expresar las gracias, porque todo lo que estamos haciendo es gracias a vosotros y por vosotros.
Okay.
Hola. Sí. Hola. Hello. So we are finalizing the program, and so now it's awards time. So I'm extremely happy to start with the Lifetime Achievement Award in Down syndrome. And this year's recipient has been Ira Lott. And it's a true privilege to introduce him. Dr. Lott has had a truly extraordinary career, one that has helped define the field that brings us together today. He trained in medicine at the Ohio State University College of Medicine, completed his residencies in pediatrics, neurology, and child's neurology at the Massachusetts General Hospital, and went on to the University of California, Irvine, where he served for many years in major leadership roles, including Director of Child Neurology, Chair of Pediatrics from 1992 to 2000, and Associate Dean for Clinical Neuroscience from 2002 to 2008. He's now Emeritus Professor of Pediatrics and Neurology at UC Irvine. But beyond these distinguished titles, what truly defines Ira's legacy is that he helped create and shape an entire field, and he did that decades ago. He was one of the pioneers who recognized that Down syndrome is one of the most informative human models for Alzheimer's disease. And studying aging in people with Down syndrome could provide unique insights into the biology, timing, and clinical expression of Alzheimer's disease more broadly. That vision helped lay the scientific foundation for what we now know as Down syndrome-associated Alzheimer's disease. At UC Irvine, he was instrumental in building the research structures that made this work possible, helping to establish one of the landmark longitudinal programs in the study of aging and Alzheimer's disease, one that now has served as a template for all ADRCs in the US that want to build a Down syndrome program. His contributions were not only conceptual, but deeply translational. He helped move the field into an era of therapeutic research, including early treatment studies with donepezil and a later randomized trial with antioxidant therapies. Even when results were modest, those studies were extraordinary because they showed that rigorous clinical trials in adults with Down syndrome were both possible and necessary. Another essential part of Ira's legacy is mentorship. He has not only made seminal contributions himself, but has helped shape many of the investigators who have carried the field forward. Among those names, Liz Head stands especially prominently. The scientific lineage that Ira helped build is one of the clearest signs of a true lifetime achievement. And I would also like to add a personal note. I had the privilege of visiting Ira at UC Irvine and sharing dinner with him and his wife, and more recently of seeing him in Barcelona with his daughter's family. Those moments made me appreciate even more a facet of Ira that is just as important as his academic legacy, his human one. Beyond the scientist, beyond the leader, beyond the pioneer, he's a deeply warm, generous, thoughtful, and kind person. Those of us who have had the privilege to know him personally know that he has enriched this community every bit as much as his scholarship. So today, we honor a remarkable investigator, a pioneer, and a mentor. It's a true honor to present this Lifetime Achievement Award to Dr. Ira Lott, who will be joining us via Zoom. So, Dr. Lott. Dr. Lott, can you speak? Maybe he's frozen? No. Can someone help us? Ira? Can you try to speak? We cannot hear it. Pre-recorded. Fantastic.
Was so important in getting Down syndrome program started in Catalonia and a model for the rest of Europe. Foundation now carried on by Kathy Trias. So I want to make a couple of comments about my bio just to set the stage. I finished training in pediatric neurology, child neurology, 1974, and that included a 2-year stint at the Bethesda Naval Hospital during the Vietnam War. Give you some idea how old I am. And the first job out of training was under Dr. Hugo Mosier to head up a newly formed research unit, clinical research unit, at the Eunice Kennedy Shriver Center. Mrs. Shriver insisted, as only Eunice could, that a research center named for her had to be where services were rendered to people with intellectual disability. So the Shriver Center was built on the grounds of what was then called the Walter E. Fernald State School. Fernald School is the oldest publicly funded institution in the Western Hemisphere. It was founded in 1848, about 18 years before Langdon Down described the phenotype of his disorder. And like many institutions in the world, it underwent progress and agonies. In the 1970s and 80s, with deinstitutionalization, the population declined from over 2,500 residential patients to about 500 or 600 today. So it was at this clinical research unit that I began to see people with Down syndrome who lived at Fernald, who were undergoing a catastrophic cognitive decline. Now, we knew in those days that Alzheimer's disease was certainly a possibility, but none of us knew very much about it. So we took care of these patients, tried to understand in terms of their neurological status and co-occurring conditions, what was going on. We had residents that joined us. One of them is Dr. Flo Lai, who made a decision as a child neurologist to work in the area of Down syndrome and who still practices at Mass General. 1983, I was recruited to UC Irvine. And in 1985, I went down from the campus, from the medical center to the campus to meet with Dr. Carl Kottman. Dr. Kottman, who sadly passed away in 2024, was one of the leaders in the neurobiology of Alzheimer research, one of the most cited investigators in history. At that meeting in 1985, I didn't know much about Alzheimer's, and he knew very little about Down syndrome. But Carl was a visionary. He could see where the field was going 10 years in advance. And so Down syndrome became part of the fledgling interests in the Alzheimer's Disease Research Center in Irvine. Today, Down syndrome has its own core, I believe the first in the network. And I would estimate now that over 100 people at Irvine, through many departments, are working on some aspect of Down syndrome. As a postdoctoral student, Liz Head made a decision to work in Down syndrome, and I think we all know what a major contribution she has made. So I want to talk to you in the next few minutes about some of the neurological observations. And the first is 2 case studies about outliers. Outliers are of interest to neurologists and to pediatricians because in studying them, you often learn about more basic questions that they raise. So the first instance is a 66-year-old woman with Down syndrome who remained cognitively stable despite abundant AD neuropathology. She had stable cognitive batteries, but elevated CSF biomarkers, abnormal amyloid and tau PET scans, increased plasma AD biomarkers, and then with the privilege of studying her brain, intermediate AD pathology. So what is going on here? Is this some kind of a epigenetic variation in the in APP? Is resilience, environmental enrichment? This example forms what in the Alzheimer literature is now called no dementia Alzheimer neuropathology. And there's a group in Europe who is thinking that these people are preserved in their cognitive status by autophagy, which means that there are metabolic processes getting rid of the tangles and the other toxic elements. Anyway, this case brings those questions to the fore. Next slide, please. Second case I had dealings with personally, 66-year-old man with Down syndrome. And by the way, we have permission to show you photographs, people with Down syndrome, that I'm going to talk about. This 66-year-old man came to our Alzheimer's screening clinic. Sorry I'm late, Dr. Lott, but I was finishing up the cash receipts of the day program. I'm thinking to myself, 66-year-old man doing cash receipts? A bit unusual. Then he said, please do not discuss my medical situation with my brother. I said, of course. May I ask why? He said, because my brother and I do not share the same philosophy of life. So I thought, okay, that's it. We've got to look at his chromosomes. Next slide, please. So chromosomes were examined by Julie Korenberg's group. And what this man had was a deleted segment of chromosome 21 that contained APP. Next slide. We studied him for 7 years. And at that time, we were using very different neurocognitive tests than today. But what I'd like to show you is in the second line, the results of his tests. And in the third line, controls from Down syndrome. Not many, it was a very small study. But look at how they contrast with the last line, people who are about the same age but have dementia. So there's absolutely no evidence of dementia clinically or in neuropsychological testing. Next slide. Next, please. So here is a partial trisomy patient, APP disomic, phenotype consistent with trisomy 21. We had our geneticists go over him very carefully. Stable neurocognitive tests, 2 negative PIB scans, no AD biomarker increase. And interesting, he had hypertension and a remote myocardial infarction. And we very rarely see hypertension and MIs and evidence of atherosclerosis in full trisomy. So it's been interesting from a number of standpoints. Next slide. We were privileged to examine his brain at birth, and on the left you see superior frontal lobe from the individual with partial trisomy at age 72, and on the right, what a full trisomy with AD dementia looks like 3 decades earlier. So this was just normal aging with no evidence of AD. Next slide. So I think this case indicates that triplication of APP is necessary for the occurrence of dementia and Down syndrome. And in this way is similar to a report by V.J. Prasher in the Annals of Neurology in 1998. Not exactly the same, but same conclusion. So there's much more going on, of course, than triplication of APP, but the case does provide a singular example of an outlier. Okay, next slide, please. Now, there are many people at this conference, including ourselves, that contributed to a Down syndrome module, which was established in the National Alzheimer Coordinating Center database, NAC, and that is called the Uniform Data Set. The module was established in 2020. And in addition to the longitudinal cohort in NAC, the other special cohorts comprise frontotemporal dementia, Lewy body disease, and now Down syndrome. So one of the questions is whether the neuropsychiatric measures in Down syndrome can align with those with the UDS. There are many aspects of the module that cannot overlap because of the baseline intellectual disability. But Dr. Christy Hom, who's a pediatric neuropsychologist in our group, worked with David Seltzer, who is head of the longitudinal core of our ADRC, to explore one of those measures. Next slide. So the severe impairment battery was a paper we published way back in 2016 that showed some alignment. The problem is the SIB is no longer part of the uniform data set. So our more recent study used a neuropsychiatric battery that is included in the UDS. Next slide. So that is the neuropsychiatric inventory. NPI is a validated performance-based interview designed to assess behavioral and psychological symptoms in Alzheimer's disease, part of the UDS. The NPI is among the tools used to establish baseline severity of psychiatric symptoms in reporting clinical trials. This is a paper just published by J.O. Cummings as part of the consort approach to clinical trials. Next slide. So you see here in the comparison between Down syndrome and older adults, 396 individuals from our ADAD-DS population and over 32,000 in NAC of older participants. And you can see the similarity in the onset of neuropsychiatric symptoms. Next slide. The NPI IDA presence by Down syndrome and older adults cohorts cognitive status at baseline, and is divided into cognitively stable, MCI, and dementia. And then at the bottom on the x-axis are all the different components of the psychiatric manifestation. I'm not going to dwell on this slide other to show that there is similarity. So we feel the NPI is a test that will show psychiatric symptoms in alignment between Down syndrome and older cohorts in NAC. Next slide. Now, these psychiatric symptoms raise questions of frontal lobe circuitry. Frontal lobe is one of the major brain areas involved in neuropsychiatric symptoms in Down syndrome. Obviously not the only area, but one of the major ones. Recently, it's been shown, not by our group, another group, that there's a changing proteomic signature in the frontal lobe over the lifespan of Down syndrome. So it's an area where a number of toxic things happened. Now, what does this mean for the neurological exam in the office? And how can inform clinicians. And I would suggest in two ways. Next slide. One is in the area of primitive or frontal lobe release reflexes, as they're called. And you see them listed there. These are reflexes which are normal in children with Down syndrome, as well as other infants. But by the time the infant is 9 or 10 years old, these reflexes are inhibited, usually by the frontal lobe. And we find very frequently that there are one or two that have limited clinical significance. We don't think that they mean very much. But when they start to pile up, and you see more than 4 abnormal reflexes, Jordan Harp has found that they're highly linked to cognitive and social symptom decline. So this is an easy test in the office that can be done to perhaps implicate clinical manifestation of frontal lobe circuitry problems. Next. The other is gait dyspraxia and relation to frontal lobe. So dyspraxia is in gait, difficulty in executing normal gait in the absence of weakness or other motor abnormalities. There's no spasticity, there's no weakness, there's no peripheral nerve damage. But people with Down syndrome have a problem with gait, very common in our experience in the early stage of dementia. And the history is so repetitive as to be stereotyped. Caregivers will say, well, he has started to hesitate when he comes to a crack in the sidewalk, or hesitate when the carpet changes to the wood floor, or hesitate at the first step. It's almost like there's a magnet tying his feet to the floor. And in fact, in the general population, gait dyspraxia has been called magnetic gait abnormality. And I do believe it indicates frontal lobe dysfunction and is common in Down syndrome as well as AD in the general population. Next slide. Now I'd like to turn to a different subject for a moment, and I'm going to throw the questions out. Are there pediatric precedents for Alzheimer's disease in children with Down syndrome? As a pediatric neurologist, we are interested in this longitudinal view that people with Down syndrome provide from infancy to aging, development and aging. So what do we know about it? Well, the first step is to note what the brain is like from the very beginning. And we have been having conversations with Renata Bartasaghi, very well known investigator at the University of Bologna, in regard to brain dysgenesis in Down syndrome. What she has found, along with other investigators, in fetal brain, there's a reduced proliferation rate of neuroprogenitor cells. Fetal time, fewer neurons, more astrocytes. Infantile, reduction dendritic branching in spines. Infantile, impaired axonal myelination. And then from childhood on, impaired white matter connectivity. So this, if you will, is the neuropathological phenotype with which people with Down syndrome go out into the world and needs to be considered as we evaluate them through the lifespan and for aging. Next slide. So this is a schematic diagram from Dr. Bartosagi's work. You can see brain size is reduced at Down syndrome, euploid on the left, Down syndrome on the right, smaller cerebellum. The neuroprogenitor cells are decreased. The neurogenesis is decreased, but astrogenesis, gliogenesis is increased. Some research have called this the gliogenic shift in the brain of infants, fetal infants with Down syndrome. And you can see the result in the final part of the cartoon: decreased dendrites, decreased connections, fewer neurons. So this is the kind of basic phenotype from where people with Down syndrome start out. Next slide. Now, do we have any other evidence that something is going on related to Alzheimer's disease in the childhood period in Down syndrome? So Mark Mapstone, who is a biomarker specialist, and Dr. Toitio, who is a pediatric neurologist heading up a Down syndrome clinic at our children's hospital, have been generating data on plasma amyloid across lifespan. Next slide. The participant characteristics: total of 440 participants with Down syndrome recruited from our Children's Hospital of Orange County, ADAD-DSAD, peripheral blood, using Samoa platform. The mean age of the pediatric study is 10.9 years. Adults mean 46.2. And you see the sex, ethnicity, and controlling for obesity and BMI, which are different in the Down syndrome group. So this data has just been generated. It's in progress, hasn't been submitted, but Mark should go ahead and show it. Next slide. So here is plasma amyloid. AB40, pretty low in children with Down syndrome. But look at plasma AB 1 to 42. Very high in the 6 to 17-year-old age bracket. And it looks just about as high as in 55-year-old people with Down syndrome. So what is it doing? I mean, is it just sitting around inert? Or is it having some effect? We know that children with Down syndrome are not making plaques and tangles. There are a whole other series of neurodegenerative biomarkers that Mapstone has studied, and that data is still coming out, but it looks like they're abnormal, but at a later time than this early childhood elevated plasma amyloid. Next slide. So Jorge Basiglio is a neuroscientist at Irvine who's worked in this area for many years. He's written some seminal papers on mitochondrial dysfunction in Down syndrome. He puts it together in a way that I find interesting. The top embryonic, neonatal, infantile, adolescent, adult, and increased reactive oxygen species and mitochondrial dysfunction from the embryonic period. Cellular senescence starts very early in childhood and Down syndrome. amyloid pathology comes in the infantile period, nerve growth factor dysmetabolism, and progressive inflammation, microglial alterations, and then in adult cerebrovascular pathology, amyloid cortical change beginning, and then classical tau pathology. So all of these things are going on, and is plasma amyloid somehow worsening some of these metabolic processes? I don't know the answer to that. I hope it will be addressed in future research. And I would just say finally about this, inadvertently, we have established two silos in regard to this issue and Down syndrome. The people who know most about Down syndrome are pediatricians, developmental pediatricians, child neurologists, but they don't necessarily know much about Alzheimer's disease. The people who see adults with Down syndrome suspected dementia are gerontologists, Alzheimer researchers. And so my view is we need to bridge this field and to make children part of future investigations looking at the lifespan issues in Down syndrome. Next slide. So I want to finish up with 4 suggestions about future directions, and one for Taya and I have been talking about this. Juan's a co-author, and the chapter on future directions for a book I'm writing called Navigating the Neurology of People with Down Syndrome, a perspective for the general healthcare professional. So you see them listed. Next slide. So precision medicine for Down syndrome, this is bandied about a great deal now, aims to customize the diagnosis, treatment, prevention of disease through the interplay of genes, environment, and lifestyle. This is going to be powered through AI and machine learning with some more comprehensive algorithms. Now, the thing that makes this attractive to me is that it's going to move practitioners away from one size fits all. Practitioners don't necessarily see a lot of people each year with Down syndrome. And when they do, they tend to make conclusions. For example, we get people with Down syndrome in their 40s, 50s, 60s referred into our clinic from the community. Well, he must have Alzheimer's, so he's on donepezil, memantine, and then we start to look at him and we find that he's getting deaf. That's why he's confused. That's why he's irritable. So one size doesn't fit all. And I think precision medicine will go away from that concept. Secondly, wearable sensors. These are small electronic devices which can collect, process, transport data to a healthcare facility. I'm sorry, I'm not quite done with that. They're used for monitoring sleep, motor problems, digital markers. And research opportunities include measures of comfort, safety, accuracy, data security when it needs to be done. The next slide. How to deal with comorbidities. We do them one by one. There is a comorbidity index that has been used in many areas of medicine, but not in Down syndrome. It's a statistical scoring system which quantifies co-occurring conditions to a single numerical value. And we like to explore this as perhaps having predictive value for dementia onset. Next slide. Finally, overcoming barriers for clinical trials. And you can see what we have listed here. I want to talk just about compliance for a moment. Do people take the drugs that they are prescribed, including investigative drugs? Next slide. So years ago, we looked at anti-epileptic drugs, very important for people with Down syndrome and for people with ID. And this is what we found in terms of cognitive decline. The major cause in this group, it was a very big group, it was about 897 patients, were seizure status. So next slide. So what we found about anti-epileptic drug compliance in community-based individuals with ID, not all Down syndrome, 793, 30-month compliance protocol. Bottom line, if you live semi-independently or in a family home, those living in those circumstances missed anti-epileptic drugs 30% of the time compared to those living in a group home. So that's about what happens in the United States with medications in general. So I think in clinical trials, particularly when they involve taking a medication at home, we need to be very cognizant about compliance. Next slide. Yeah. Okay. Yeah, I want to make some brief acknowledgments. Eric Duran has been my senior coordinator for 27 years. This year, he won the Healthcare Professional Spotlight Award for the state of California Regional Center System. Alicia Hernandez, 26 years, she's been executive assistant and very good as a family liaison. She's absolutely crucial in research. Next slide. Here is our current coordinator team. Next. We've had 35 years, back one. 35 years of continuous NI support, mainly for the National Institute of Aging, Shriver Center Institute for Child Health, Human Development. Very grateful to advocacy groups. In the United States, they've been in the halls of Congress for 3 decades. And our local grassroots Down Syndrome Association of Orange County started the Down Syndrome Clinic at our Children's Hospital. Next. And most importantly, to people with Down syndrome and their families for dedication, loyalty to our programs. Next. And I put this slide in just to remind us in our archives of why we're all here. 50-year-old high-functioning man with catastrophic physical and cognitive decline due to Alzheimer's, being comforted by 2 of his friends. And this is not staged, this is a real photograph. And it emphasizes the empathy and interest in others that people with Down syndrome have. And how to preserve that is a major goal of our research and your research. Next slide. So to end on a more optimistic note, people with Down syndrome teach us about other people with Down syndrome. They teach us about issues in the general population. And in the end, people with Down syndrome teach us about ourselves. Thank you.
Is he in Zoom? Ira, are you listening to us? I think we have an issue that when we speak, I think that you're silenced, and I guess that the audio channels can hold both, maybe. Well, in any case, if you're listening to us, Ira, thank you very much. It was truly inspiring. We know that you had a previous compromise that you couldn't come. I believe you're in the East Coast. You told us, and just for let you know that we have a small detail. No, that it's a book for Gaudí, as we said yesterday. Is the centenary of his death, and so we had a book from Gaudí and a small plaque signaling the Lifetime Achievement Award. And again, congratulations, and it's a true privilege to give you this award. Thank you very much for all your work.
So now we are. It's a true honor to present the Life Achievement Award in Autosomal Dominant Alzheimer's Disease to Professor Sir John Hardy from the University College of London. Professor Hardy's groundbreaking work in neurodegenerative diseases, and especially his research into the genetic basis of Alzheimer's disease, has marked a pivotal moment in biomedical science. Through landmark discoveries done by his group, including the identification of the first Alzheimer's causing gene mutation and the formulation of the amyloid cascade hypothesis, Professor Hardy has fundamentally changed our understanding of neurodegenerative diseases mechanisms and has paved the way for new therapeutic strategies. While there are awards that simply honor their recipients, in this instance, it's the acceptance by Professor Hardy and his presence here today that truly elevates the committee and these occasions. On behalf of the steering committee, we offer our heartfelt recognition and gratitude for his legacy, his transformative discoveries, and the enduring influence he has on the field. Please join me in a warm and enthusiastic round of applause to honor Professor Sir John Hardy.
Thank you, Liz.
Okay. Happy for you?
Yeah, thanks. Just before I start, I want to say how pleasant it is and how inspiring it is for me to come to this meeting, because one of the things that I have felt frustrated about previously was how little attention Down syndrome got, and how that was now costing Down syndrome patients opportunities in terms of getting access to clinical trials and so on. And I think coming to this meeting, and this is the third one I've been to, I can see that this meeting is actually central to the positive changes that have come about. So I really think this is a meeting that, well, you guys can be especially proud of, and we can be grateful for coming. So thank you. It's very nice, of course, to get an award. Now, I'm going to be fast here, because I don't want to talk so much about the past. I think we've still got to campaign, and we heard that actually in the session, especially from South America. But it's true everywhere. We still have to campaign. Now, there are treatments, as we know, and I'll go over them very quickly. for Alzheimer's disease. They're expensive and difficult to give, but they are real treatments. And we have to campaign to make sure that your patients, your Down syndrome patients, have access to them. And this is as much a political campaign as it is a scientific campaign. So that's the background to this. I'm going to go through the first part of the slides very quickly because, you know, we all know the history really. So let's, whoops. Perhaps someone else should do the slides. It's not responding to, this is the pathology. Plaques in the middle, bottom. Here is the congophilic angiopathy, bottom right, which we ignored, but actually has come back to bite us because this is the underlying pathology that the ARIA is caused by. So this is the pathology. This is really a really important paper from Campion in France. And it was mentioned indirectly mentioned by Dr. Lott as well, really, which is that people with the APP duplication are the people who get Alzheimer's disease. And those individuals, as he said, like VP Prasha published, which don't have the APP but are otherwise trisomic, don't. So really, Down syndrome is where Alzheimer's disease started. And the next thing I think, and you should think about this when you think about campaigning, is that actually Alzheimer's disease is generally a fatal disorder for Down syndrome. When you do the balance of whether to treat or not in cancer, this is the type of equation that one always has in one's mind. Is on the balance of the situation, is it better to treat? Well, in Down syndrome, if we don't treat, then we have a very bad outcome. So I think that this needs to be shouted from the hilltops to our regulators. What could be more important in an individual than actually preventing their death with a treatment? So this is something which is, although you guys know it, and Dr. Lott just spoke about it, but you guys all know it, but the general public does not yet know it, the regulators do not yet know it, and sadly, neither do the authorities which decide whether drugs get reimbursed or not. There's still campaigning to do about this. So when we formulated the amyloid hypothesis, and of course it was first formulated really by Glenner from his work on Down syndrome, when we formulated it, people said, okay, we believe that amyloid is what causes early onset disease, but we don't necessarily, and that's very rare, we don't necessarily believe that it's what's involved in late onset disease, and we geneticists couldn't answer that question, but now we can. We now know what are the genes and what predisposes to late onset disease. This is the paper on the bottom left from Leslie Jones was the first to make this point explicit, and that is the vast majority of risk genes, the vast majority of risk genes for late onset disease are involved in amyloid clearance. They're actually microglial genes involved in lipid metabolism, and it took us a while to understand why. Now we know also that the genetics of Alzheimer's disease is a two-stage genetics. There's the genetics of amyloid deposition. The genetics of amyloid deposition is, in general, the biggest risk easily is ApoE. But the risk of dementia in that context is independent of ApoE, and it's in fact dependent nearly entirely on the microglial response. So we now know a two-stage process: amyloid deposition, dementia in that context. This is largely ApoE dependent, and this is largely microglial dependent. Now we know there's a lot of argument, and I'm going to talk about it briefly, about whether these drugs work, and the answer, you know, when we see the drug trials, and they've been mentioned, we've seen the lecanemab and the donanemab trial, but much more important than this trial data really is the real data from real clinical operations. This is the data from Washington University, and they see that it is really an effective treatment in their dementia cases. I've seen similar data from the Mayo, and they have reached the same conclusion. And in a much smaller sample, I've seen the same at Queen Square, where we're already now involved in the amyloid treatments. So these are treatments which work. They require monitoring, but they work. This is the pattern, though, and this is work that I showed last time I was here, but from Tony Holland and Shahid Zaham's group, looking when does amyloid start to deposit in the brain of Alzheimer's sufferers, and here is that really well documented now. We now know that this deposition starts, if you like, in the late 30s. So we know if we wanted to start to try and treat with anti-amyloid drugs in Down syndrome, we know what age to treat with. And there is a belief, and it's a belief, not yet a fact, that if we could treat early, we would get less, we would possibly have a greater efficacy, but we would also likely have a less side effects from ARIA, because there would be less amyloid for the antibodies to hit. So we know what to do. And this is the data from the Cambridge study of amyloid deposition, which has been discussed here before. So dementia in older adults is almost universal. From a diagnostic point of view, one of the problems that was really bedeviled the general Alzheimer drug trials was that the trials, until there was good biomarkers, had diagnostic accuracy rates of only about 70%, which meant that about 30% of individuals who went into the trials did not have Alzheimer pathology. Obviously, if you've got bad diagnostics, that's weighting a clinical trial against yourself. In contrast, in Down syndrome, if you're hitting cognitive decline in the 50s, it's almost certainly one does not have a diagnostic issue. And that's something I think that the drug companies and you guys can take on board to push trials in Down syndrome individuals. Now, I'm often asked, why did anti-amyloid drugs take so long to work? I was getting emails in the 2010s, 2012s, when we had a series of anti-amyloid trials which didn't work. And I would get, they were friendly emails, but, well, reasonably friendly. They would say, you know, how does it feel to have slowed Alzheimer's research up for 20 years with these stupid anti-amyloid trials? When are you going to give up? So for me, when the first trials worked, believe me, that was a relief more than anything. Anyway, I'll just talk about why do these first trials look so hopeful in mice and yet not translate? The answer is that we misinterpreted them. These first trials, and this isn't the only one, what they did, these antibodies did, was prevent further deposition. If the deposition was there, they did not suck, if you like, the amyloid out of the brain. So these early trials, well, I'm going to come back to the idea of statins, but they were like giving a statin during a heart attack. Right drug, but too late. That's what these, in general, that was the other problem besides poor diagnoses in these early trials. ARIA, what's causing ARIA? Well, what's causing ARIA is that pathology we didn't take any notice of, the blood vessel pathology. Almost certainly it's caused by the antibodies hitting the blood vessel at high concentration and causing an inflammatory reaction. So some of these, as I've been mentioning, but I'll just leave this slide up, I think that some of the problems that have been had with the trials, problems would not have been had with Down syndrome. And so I really think it's great that you're getting some of the drug companies at least now to start testing drugs with appropriate care, of course, in Down syndrome. So I think this is something that we should be campaigning for. But the breakthrough for me, I'm going to come back to the last point, p-tau 217. I'm going to come to the breakthrough point. This is the paper that really woke me up to the problems we've been having with clinical trials. It's from Eric Karen and Bart de Strooper, and he said, no, they said, what you need to do is not prevent amyloid deposition, you need that the drugs suck amyloid out of the brain. And this was their theoretical slide, published before any of the successful trials. And you can see there that they suggest on the left-hand side that, let's see if this, oh yeah, there we go. That here you give the antibody, it reduces amyloid a long way, and then you run the clinical trial long enough to have time to see a separation between control and placebo and treated. So this really was the prediction they made, and sure enough, this prediction is accurate. And this is the real data, and the real data is interesting, both the positive and the negative real data. These are the trials which have been approved, donanemab here, lecanemab, both of these have been approved. Gantenerumab, which I'll come back to later, failed. And you can see now, when you look at the amyloid reduction, you can see why it failed. It didn't reduce amyloid fast enough or far enough. But actually, aducanumab is really an interesting outcome. For a drug to be accepted by the FDA, you typically have to have 2 successful trials, independent. And actually, aducanumab was really the exception, because it had 1 successful trial and 1 trial which was not statistically significant, and there was a huge fight, it was in all the newspapers, about whether this trial should have led to an approval. And I have to say, the Alzheimer's Association in the States really went out on a limb to back the approval and got a lot of heat for it, actually, so they really deserve some credit. But actually, now we see the data, we understand this was the positive trial and this was the negative trial. So it fits with the Karen and de Strooper model. So I think this is important. I think this is important not only for the drugs themselves, it is important for that, of course, but it's also important because it teaches us what we need to do. If we have an anti-amyloid drug, which we think is removing amyloid, and we can't hit these curves, we know that it isn't going to work. So it really resets the agenda in terms of running clinical trials. And I'm going to go on about that a bit more. Here's the lecanemab data, removing amyloid. Here's the lecanemab data, clinical outcome. You can see it's 2 things. It's worked. There's just no question that it's worked. There have been a number of people arguing that it hasn't worked. I don't think this is a valid argument. It has worked. Second point, though, it hasn't stopped disease. It has not stopped, or rather, it has not stopped decline. We need to understand what is the basis of this residual decline. And this is an area of research that really is important over the next period. Now, there's a separate question, which is a political question. No question it has worked. And what's happened in the UK, and in Europe, there's a continuing fight about it. But here in the UK, it's been approved, but it hasn't been approved for reimbursement. And that has led to a very, very unusual outcome. And it's very unusual in the UK. I don't know of any other situations where this has happened, where the patients, private patients, are being given the drug on their own dollar, but NHS patients cannot get the drug. There's a second part to that, though, and that is a big part of the cost of giving the treatment is the monitoring and the dealing with side effects. And what happens if a private patient has serious side effects? Well, they're told to go to the NHS. So we've come to a situation where private patients are getting the drug if they wish, but half the price is being, if you like, paid for by the NHS. But NHS patients are not getting the drug. And of course, I don't think Down's patients are either, and that's something to campaign for. So this is, you can see that this is really a difficult issue. The other point is when they're making the decisions about cost. Those decisions about cost historically have been to do with lost income. So you were, in other words, when you were balancing whether it was, in inverted commas, worth treating somebody, what you were doing is you were balancing lost income versus cost of drug. That's what the actual... Now this, of course, means that Down syndrome individuals are going to do badly in that equation, really, but also elderly people do badly in that equation. So really, people are gradually waking up to this, if you like, loaded assessment into a reimbursement. Now, I wanted to come back to things. One of the things I wanted to leave you with as an idea is we have treatments. They're not great. We're discussing the problems now. But I think every year from now on, the treatments are going to get better. Every year. And the treatments are going to get better in a number of ways. Number one way, we're going to get better at deciding which patients are going to benefit. That's going to happen. Number two way, we're going to understand the ARIA better and better. The clinicians already know that ARIA is usually after the first or the second infusion. So maybe we can relax the safety monitoring after the first, second or third infusion. Third thing is that they're now doing subcutaneous infusions, and that clearly makes the whole process cheaper and easier to organize. So those are ways, but of course are we going to get better drugs? And maybe this is the first better drug. This is trontinemab. It's been only published as a phase 2, and what it is the gantenerumab active site tied to transferrin. And so the transferrin shuttles, it's amazing. It's amazing. I would never have believed it would work, but it shuttles the active site of gantenerumab into the brain, and that means the drug can be given at much lower concentrations, which should, and this is only a phase 2 data, should massively reduce the ARIA risk. So we can see that this type of drug, and this isn't the only one, is coming down the pike. We need to answer, these are the questions, the research questions we're now being set. What is the underlying, what's the cause of the residual decline? You know, the amyloid has gone from those brains and yet they're still declining. Is it in some way amyloid dependent or is it amyloid independent? Will we get better with earlier treatments to reduce ARIA? Those trials are going on right now, and I've just come back from the ADAD-DSAD, and I was hoping to hear about this, but we didn't hear much. There's going to be an AAIC meeting in London. Maybe we'll hear from that in a couple of months whether the earlier treatment has less ARIA. Can we get better this way? But we're always going to be facing the problem of long-term trials. Let's just fantasize that trontinemab is going to be a great drug. The trouble is, if we know now what it does, but even if we know now what it does, we'll only get it into the clinic in maybe 5 to 10 years' time. The whole process of proving a drug works takes so long that we can't get faster, and we need to get faster. Our current patients need it. You know, we need to speed the process through. And I think that the biomarkers are a way of speeding it through. And what you're looking at here is a p-tau. And across several different dimensions. P-tau goes up selectively. This is from our, these samples were collected by our Turkish colleagues and run by Amanda Hesselgrave and Henrik Zetterberg. And the p-taus go up selectively in amyloid depositing disease. But better than that, when the amyloid is sucked out of the brain, this is lecanemab data, when amyloid is sucked out of the brain, p-tau responds. So p-tau is a marker of amyloid deposition, but it's also a marker of amyloid removal. And if p-tau is a marker of amyloid removal, we should use it as the primary outcome. And we need to convince, and this is a political and political campaign, really. We need to convince the regulatory authorities, and this is the first stage, for me anyway, of convincing the regulatory authorities, is convincing you. That's stage one. The next stage, maybe you need to be talking to the Alzheimer's and the Down societies, and also, of course, to your local clinicians and so on. It's a campaign. So I think we should be using the plasma p-tau as the outcome measure in phase 3 clinical trials from now on. And of course, I realize that that's something that's often difficult. I spoke to somebody earlier in the day who said that in Sweden, nobody believes biomarkers anyway. That was not quite what they said, but especially not Henrik Zetterberg, let me just say. but it's a campaign. So, and I'll just remind you of the history of statins. There was the pathologists and then the geneticists said cholesterol was bad news for heart attacks. Actually, starting with the pathologists in the 1930s, geneticists got into the act in the 1970s, but it was the pathologists and then geneticists said cholesterol was bad in heart disease. And then Endo in Japan showed that statins reduced cholesterol. He had an early statin that he got. I think he got it from a seaweed. It was really a biological. He got from a seaweed which reduced cholesterol, and so that was 1975. And then statin, because this convincing worked and the regulatory authorities were convinced that cholesterol was bad for heart disease, then statin was approved simply as a cholesterol-lowering drug in 1987. And it was only shown to be clinically useful in heart disease in 1994, and only shown to be clinically useful against stroke in about 1998. So that by having statin, having cholesterol as the target, saved at least 7 years in terms of the approval process. And 7 years is a long time. And we have to fight the same battle to get our colleagues and then the regulators to believe that p-taus are going to be a basic target for Alzheimer's in both the general population and Down syndrome. So it's a campaign, and we need to campaign now. Thanks very much. And thanks again. I really, yeah. Oh, pictures. Oh, God.
Yeah, to make it fast, we have more awards. Like last year, we want to recognize some early career scientists, and we'll have Maria Carmona to announce the award. So, Maria, are you here?
Okay, hello, thank you very much for staying here. I'm glad to announce the awards for early career researchers that have been kindly founded by the Jérôme Lejeune Foundation. And there are 3 poster awards, 1 communication award, and 1 early career research award. I will communicate each of the 3 categories by separate, and please, the awardees come here to take a picture. And I hope you are all still there. Otherwise, we will send you or them an email. So the 3 poster awards, the decision was quite difficult because all the posters were really nice, and all the communications, as you saw, were also, the level was really high. And also the early career researcher was not easy to decide, but the 3 poster awardees are Lucía Pertierra, Luis Echeverri, and Sofía Gasó. Please come. No tengo nada para hablar, no, gracias. Luis is not here? Okay. So, da igual. So that's the funniest. No, we will send you an email with the details of the prize. We don't have anything in material to give you now, but those are good news. This is the funniest part of the conference, I think. So the Oral Communication Award goes to Natalie Edwards. But I think she left. So and finally, the Early Career Research Award goes. I'm glad to congratulate Lizzie Flores. Hola, una foto. So that's it. That's it.
So now we'll be closing the conference, and we'd like to start with an announcement by Natalie Ryan, if you want to come.
Okay, so I think first of all, actually, I just want to say a huge thank you to Juan and Raquel, not just for today, but for the last 3 years of absolutely incredible conferences. I've learned so much at each of these, as I'm sure you have, and I just have seen firsthand and been involved in so many collaborations, new projects. I think everyone who's been at these conferences has recognised how special they are, and so thank you for creating them. And I also want to thank Maria and the Alzheimer's Association for recognising how important they are for our field as well, and committing to supporting their ongoing continuation. And Juan and Raquel have been really clear that they feel that the conference does need to move to new locations. It needs to, have new chapters. I've been told this is not forever, but for next year, it's going to be my honour to welcome the conference to London. So I really hope to see many of you there. Thank you.
Well, thank you. Since I don't have a microphone, I'll come up here. And thank you, Natalie, for that introduction to London. And we're very happy to continue to support and to collaborate with the current steering committee and the chairs who've done such an amazing job. This is my first time, but my team has been coming, and they have said how amazing this meeting was and that I had to come and see it for myself. So I'm really, really proud of that. And I think the collaborations that I hear have already been fostered for 2-3 years now, and hopefully will continue, can also be fostered further by a collaboration between the Fundación Pascual Maragall and the Alzheimer's Association. We've launched a co-funding program, so you should take a look at it and see if there are opportunities that might fit you for, again, cross-country, cross-the-pond collaborations. Our goal is really very similar to the Pascual Maragall Foundation, which is just to support the field to ensure that work gets done as fast as possible. And I know that from what I've heard these past 2 days, everything that you do is a labor of love. And for us, that's what we want to continue to support. So thank you all, and please apply for funding. Thank you.
So can you put the slides that we prepared? Just a note, we're going to change location and also the timing of the year. So it will be in the fall. I don't know if you mentioned it, but it's going to be, so save the date for next year in the fall, not in the spring. Do we have... Okay, so I guess I'll start by thanking all the I'll leave that to Raquel. I'll give you the numbers. I'll give you the numbers of the conference, so you know how we organize it. To remind you, you've been here, so no surprise. And I'll give you a few numbers. So we had one keynote that David gave yesterday, was beautiful. 2 Lifetime Achievement Awards. So congratulations again. 4 oral sessions. That's the abstracts that you submit, and you submitted through this year as a new mechanism through the Alzheimer's Association webpage. And so basically we had a lot of reviewers in this room, and also we used some of the Alzheimer's Association reviewers in their system. And we had 4 symposia that were by invitations. We had 16 oral presentations, 31 speakers, 38 posters, and a lot of time, I guess, to mingle. I think that's the key of the success of this conference, so that we can have a lot of time to discuss. I think the most important part takes after the presentation, when we have the discussions in the coffees at night. I think that's the really important part of this conference. And this is just a few numbers. So we had 231 participants. I have to say we were a bit nervous, because we had 184 in person, now not, but at the beginning 184, and this room fits 190, so we are close to the limit of in person for this room. Half of them, approximately, came from Spain, 44 from the US, but you see that we had 28 countries coming to the conference, and I think that speaks to the interest that this conference has had in only 3 years. And I think that this year we managed to get some travel awards, thanks to the Alzheimer's Association, thanks to UNT from Sid O'Brien, and thanks to the Jérôme Lejeune. But still, it's a kind of poor conference. We would love to support more early career investigators, but still, I think that is a success. So we're very proud of that. And as I said, the other thing that we want to recognize is always the early career investigators. I'm not going to repeat it because she just went there. And maybe I leave this to Raquel to close and thank the people that put the money.
It has been really a great pleasure to welcome you all here in Barcelona again. Thank you very much for coming. A special thanks to the members of the steering committee, the scientific committee, moderators and presenters for making this conference possible. Thank you again to the Alzheimer Association, the Barcelona Alzheimer Hub, the Institute for Translation Research in Texas, the Fundación Jerome Lejeune, the T21 Research Foundation, the Fundació Catalana Síndrome de Down for their support. We wish to express also, again, our gratitude to our sponsor, Lily and Al Nilan, and the National Down Syndrome Society for the generous support. And thank you also to Rosa Roda and Mireia Cañadas from Support Surveys in the back and the technical support. And Ana Bono. And also the people from the Sant Pau and the Clinic Barcelona who have helped us to organize this meeting. So I hope you have enjoyed the meeting and have a safe trip back home. See you in London next year.
In committee, we have a photo, if you don't mind. The rest of you, safe travels.